Hi, James.
Hello.
Hello. I don't think neither you or Giuliano are recording at the moment, or Mateus neither if he's meant to.
Yep, I just noticed.
Hi, James.
Hello.
I don't think neither you or Giuliano are recording at the moment.
Juliano, have you been posting the links to the Zooms, the Zooms in the CompuCell Twitter feed?
I have not.
We need to do that.
Yep.
You need the link.
No, I can find it easily enough.
And then one other thing, Giuliano, if you can. I should have done this earlier, which is I should have drawn attention to these documents for people to prepare for the hackathon.
Mm-hmm.
So if you could post those in the, maybe it's better I do it. Here, let me get that. Sorry. Boy, I'm behind again. So Giuliano, I've made you co-host. You should have been post anyway.
Yeah, I actually was host for a while, and then I guess when you joined, I got
How to change the Okay, Mateus, do you have co-host privileges? You should for recording. I've uploaded a lot of videos, but there's some missing ones and we need to trim them and edit them.
Yep.
Great.
think what I'm gonna try to do is create chapters on the live streams that are in your channel already, James. Maybe that will make the process of cutting in them up afterwards easier.
Whatever you feel works for you to do it. Okay. Yeah, thank you for joining us. I hope this has been at least a little bit helpful. I know it's not always as organized as it could be, but I know you're going to be working with this now for a couple of years, so I hope it just helps you get started. TJ, you're ready to do MaBoSS?
As ready as I'll ever be.
Do you have were you able to do a workaround for the Windows issue?
Not one that I would trust.
Because it looked, when I did it, if I installed 4.2.4 and then reinstalled 4.2.5, it worked.
Yeah, I saw that. So my thought on that is that when we get to the hackathon, for those who want to use that functionality on their project, we can do a quick overview on those details and things like that. To get through the rest of today, I don't think that we'll be using libRoadrunner or Antimony for the rest of the modules, but if we do, I would strongly suggest the instructor just send everyone to the NanoHub tool. That's what I'll be doing for MaBoSS, to skip the extra installation work for the local installs. Just to keep us all focused on what's important, and I don't, I mean, it may just be my impression of the hackathon, but I feel like it's maybe a little more okay for, going off with people who want to fix that package on their local install and working with them on it. What do you think about that?
I'm sorry, I had to drop out. So you say that again. I'm sorry.
I don't think that there are any more modules that will be using that functionality for today, but if someone is planning on using it that I'm just not thinking of, I would just urge them to send the local install people to the NanoHub tool for that module. That's already what I'll be doing today for MaBoSS, because I don't want to spend the time walking people through getting MaBoSS added into their CompuCell installation. Yeah, be happy to do that during the hackathon, but it's not something that's generally applicable to everyone. So I'd like to just skip it if at all possible.
Okay. Okay. So at the moment, I don't know how many people will join us in the next few minutes. Feel free to chat, and we'll get started in a few minutes. If there are any questions about yesterday, now's a good time to ask them. Okay, I see. Oh, Gilberto, welcome back.
Hi, James. You too. uh,
We'll wait just another minute or two and then get started. I think people are getting tired, so I suspect people will sign in later, probably.
yes, Friday is always like that. I was teaching today.
What are you teaching?
Initial electromagnetism for an engineering course.
That was never my favorite topic. Let's see, there's a question here in the chat. If we want to model immune response in cancer with a pure ODE model, you suggest using CompuCell and antimony, or their tools are better suited. If you're going to just do ODEs, I would suggest using tellurium and antimony are very good. There are a lot of other modeling packages of various kinds that you might want to think about, but tellurium and antimony are great. Tellurium, I would run in the Jupyter Notebooks installation version of it. And that has the advantage compared to running inside of CompuCell that it runs a lot faster. And you have more plotting options and things like that. If you want to do very complex models, a lot of people like Cell Designer. Some people like Copazi. Copazi is slow, but has a lot of nice features, especially for PBPK modeling. Has a lot of support for that. They all use the SBML model exchange backend, so that you can go back and forth from one to the other. We didn't show you, but if you look in the cheat sheet, you can convert SBML to antimony and backwards. So the problem with SBML is it's an exchange language. It's like PostScript. It's not something that you want to read by hand. And so if you're going to write SBML models, you have to have an editor of some kind to do it. The Cell Designer is a graphical user, a graphical layout tool, has nice features for drawing networks, these kinds of things. But it's a lot more complicated to use than antimony and tellurium. So if you're not going to go into spatial models, then I wouldn't run it inside of CompuCell, because you have the overhead of the spatialization. That's a very good question. The advantage of writing the models in something like SBML or antimony, though, is that if you do it in Copazi or the Jupyter Notebook, Tellurium, or one of these other languages, you can pull them in to CompuCell and run them without changing them. And so there's no reason you have to choose. You get both. Great question. Any other questions? So what do you think, TJ? I'll wait another minute. I guess people are still signing up. Please feel free to ask any questions. TJ, go ahead if you have something you want to say.
I was just going to ask, you meant concerning the pre-module discussion, or if we're actually getting started with our first module of the day.
I was just going to do the orientation. If you're rearing to go, I think we have to wait till 10:30 to start formally.
Yeah, that would be my preference.
Okay. No, I meant just to discuss presenting the agenda for today.
Sure. Yeah, I'd say it'd be okay to start. We may have to do some repetition for those who will join us.
Okay. Okay. Okay, well, welcome back, everybody. Thank you for coming back. Today is day 5, the last formal class day of the workshop. I hope we've caught up with some of the things that were issues before. We're still working on this issue with the Windows release 4.2.5 for Tellurium and antimony. If you're having trouble with that, we really ask you to use the NanoHub installation, which definitely works for today. If you don't want to do that, you can roll back to 4.2.4, but please don't do that until after the first module, because the module you need is in 4.2.5 and not in 4.2.4. I need to remind you that these sessions are all being live streamed and recorded, so please be aware of that. We are slipped by one module, so we're going to begin with module 4.4 from yesterday on doing Boolean network models inside of CompuCell. It's a brand new functionality of CompuCell in 4.2.5. We were very excited about it, which is why we rushed 4.2.5 out. Maybe we're a little bit too enthusiastic doing that. And then we'll have Julio Belmonte giving 2 lectures on links, epithelial tissues, and convergent extension, which is a very important component of CompuCell. And you'll see how to use those in the context of multicellular, of subcellular compartment models too, which are quite useful for building more elaborate biomechanical simulations. And then we'll close the session with Jim Sluka talking about how to run parameter scans in CompuCell, do data exporting and file sharing, and some things about how to design your models to be optimally shareable and efficient. So that's the agenda. 10 to 10:30 is our usual introduction. 10:30 to noon will be TJ speaking on MaBoSS. Julio will be speaking 12:15 to 1:45, and then again 2:15 to 3:45. And Jim Sluka will round us out at 4 to 5:30. Then we'll have the usual time for questions and comments at the end. I wanted to draw your attention to something that we mentioned a couple of times, but perhaps should have been more put more emphasis on, and that is that in the student materials, there are a number of documents and exercise sheets that could be very helpful for people, especially if you haven't done this kind of modeling before, in terms of developing your model ideas for the hackathon. There is a very short document called Hackathon Preparation, Thinking About Model Strategies, which gives you a first stab at how to define a model that makes sense in CompuCell. There's also a longer document which goes into quite a bit more detail about how to think about constructing a model. How do you define what a model is trying to do? How do you define the components of the model? How do you lay it out so that it's functional? That's called a workflow for developing mechanism-based computational biology models. And I strongly urge people to read that document. It's only a couple of pages, and it will be useful to you here. And if you're going to be doing computational biology, I think it'll be useful to you no matter what kind of computational biology you're doing. Lastly, there is a worksheet based on that workflow for developing mechanism-based computational models, which tries to lay out a series of questions to define those models. And I'd strongly, again, urge people, if you're very far along, you don't need it, probably. Even if you are far along, it's a good way of organizing the information that you've thought about. And so I'd strongly urge people to look at those and try to go through those. I put the links to them in the chat, and we'll put them in again. Giuliano can put them in again. For the hackathon Zoom is a different Zoom link from the one we've been using. So please remember to register. We'll start at 10 a.m. Tomorrow. I would ask people to go over, I know you've done it before, but for people who have not, joined a team yet, it would be very helpful to have another lightning 30-second presentation of your idea so that people looking for teams can join up. It will take about half an hour to an hour from 10 to 11 to get the team set up. If you have a team name, descriptive team name, please send that to me so I can do a breakout rooms for your team that are descriptive. The last time I used the name of the person who was identified as the lead for the team, but that doesn't help very much for people who don't know the subject. So please give me a short descriptive name. I don't remember what the character limit is in Zoom, but it's not very long for your project. And we'll do that quick check-in on Saturday morning. Then we'll have a work session. In the middle of the day, we'll have a quick check-in again, optionally for people who want it. We'll ask people at the end of the day to do a very brief lightning presentation of what they've done. And we'll follow the same format the second day, except that we won't have we can have a check-in in the morning rather than a repeat of project names. And we'll do a slightly longer presentation at the end of the day. So again, for Hackathon Day 1 tomorrow, 10 a.m. It doesn't have to run to 11, but please do help people looking for hackathon teams to join. We'll use the hackathon Slack. We'll meet for about 2 hours initially to get going. I encourage people who haven't started on their projects to use the preparatory material I've just alluded to before. We'll have as many of our team as possible available. Not everybody will be available at all the time, but we'll do our best to support you. And we'll ask people to report around 5 p.m. So that we know where we are. Okay, that really was what I had on the agenda for today. Now, if there are any other questions or comments, we've got a few minutes for those. There was another question. Yes, go ahead, please.
Oh, so I was going to ask about a model that I know that has been previously built, which is a model of the Polonaise gastrulation movements in chick embryos. I think it was 2010 that it was built. The code is actually available, like with the paper, but it comes kind of packaged with pots, and I was wondering if you have the code available, not packaged with pots, so that I could see what has been previously done, so that if I try and build something, I'm building something slightly different at least.
So boy, this is the paper with Case Ware, right?
Yeah. Vassiev is the first author.
Right. Well, because there are two. There's one with Bhakti, and then there was one where it was help.
I'm not sure. I'm not familiar with it. With another one.
Okay, so there were two papers that came out. Bhakti wrote his own software.
Okay.
And so he didn't use CompuCell. And so I don't know the model structure in detail. Ariel Balter also did a paper on that. Maybe we're talking about the same paper. There were two that came out nearly at the same time. But I wouldn't worry too much. Those papers were pretty, first they're old. So I doubt the code from either one really runs. If it runs, it'd be nice, but I'm impressed if it does. But secondly, I think that one can do a lot more now. In particular, if you were going to do CompuCell now, the Convergent Extension that Julio will show us today would be very important. In principle, you could do something in 3D where you actually have the cells leave the epiblast and move down. And I think you could do more in terms of collective migration. You could use some of the ideas that Giuliano showed at the very beginning of persistent movement, which are probably important.
I'm certainly looking forward to the Convergent Extension. So is that effectively modeling intercalation of cells?
That's right. The Convergent Extension is one kind of intercalation. They're different. I used to argue a lot with, I shouldn't probably say this on recording. I used to argue a lot with Claudio Stern, because in the textbooks, intercalation is described as a mechanism of development. But intercalation is a result of cells doing specific things. It's not a mechanism by itself. It's a result.
Okay.
And we used to argue about this a lot. In the end, it came around to my way of thinking, which is not always easy with Claudia. And so one of the questions with intercalation is, how are the cells exerting forces? When you're in an epithelium, it doesn't look like the cells can exert forces very easily, except very locally. Now, in fact, on the basal surface, they're doing things that are more long range than it looks when you look from the apical surface. So Julio's methods were designed to model essentially germ band elongation in convergent extension. And that's almost certainly, depending on the organism you're working with. If you're in zebrafish, that's almost certainly important in early development. With chick, because the embryo is under so much tension, it's not so obvious that mechanism is there. And you still face the question of what is giving the polarity of the convergence. You have to have something that organizes a planar polarity like mechanism, whether it's actually planar polarity pathway or something else. Julio's plan was hoping to talk about planar polarity models today. I don't know if he's going to get to it, given the time limits. I think one thing this course has taught me is that we really need to have an advanced course where we cover some of these ideas more. A week is a long time to ask people to be here, but it's actually not that much time to introduce these ideas, just because the biology we're covering is complicated, and so there are a lot of things that have to be discussed. But I would love to see those. That initial paper with Ariel and that we did and Case, we really hoped we'd follow up on that. And then, as usual, Ariel went on to another job, and we never did a follow-up paper. So I'd love to see that work get refreshed, and I think there's a lot still to do there.
Okay, cool. Thank you.
Okay, any other questions, comments?
Actually, I had a question.
Sure.
And it may come up in today's examples, but I'm not sure. So one of the things we've looked at a lot is how cells organize in groups, primarily based on cell surface properties. But it seems like CompuCell now has another additional mode where you have elasticity or some, like from the center of mass of the cells, some like springs holding them and pulling them together. How do these two ways of bringing cells together interact? Do they need to be done separately? Can they be combined? And do we have examples where we can better understand the other mode, where the center of cells sort of elastic connection with each other dominates what happens?
Right. So every modeling methodology has advantages and disadvantages. The biological reality is that cells have surfaces and they have actin cortex, and therefore that the molecular species on the cell membrane are not freely diffusing, but are localized. Now, vertex models, which we showed at the very beginning very briefly, are good because if you have differential contractility along certain surfaces, if you're in an epithelial sheet and you have differential contractility along certain directions, the models are very good at doing that. And because the edges in quotation marks of the domain are objects, namely links, you can attach things to them. And so the Drosophila people love that because that's something that they need a lot. If you're in a center model, the cell doesn't have any volume to attach anything to. It doesn't have a surface to attach anything to. And so if you want to then implement those features, you can do it, but it's rather awkward. You have to do a lot of hard coding of things to do it. In CompuCell, volume is defined. The voxels inside a cell exist. So you can attach things to those, but the surface of a cell is not defined. There is no membrane, explicit membrane in CompuCell. The methods that Julio will show today, I hope, are a way of getting around that to some extent, but you still don't have the mechanical representation of the cell surface the way you would if you actually triangulated a cell and used the equivalent of a solids modeling package, where each surface is divided into little triangles that represent that surface. And that means that you have to do a little more work to represent some of these phenomenologically, these things like junctional adhesions or particular kinds of contractility. Now, the links that CompuCell uses are a way of doing some of that. But as you pointed out, they have to anchor to the center of something that's a cell-like object. It doesn't have to be a cell as a whole. If I take my cell and divide it into lots of subcompartments, then I can anchor onto each one of those subcompartments. And so if I really want to build a cell which has an explicit membrane, I could do that by defining a cell object and then defining lots of small membrane objects around the perimeter of the cell, and then linking them together to actually make an elastic membrane.
I see.
Now, in 2 dimensions, that's not unreasonable. It works pretty well and it's reasonably fast. If you try to do that in 3D, you're reinventing MaBoSS software package, and it takes a lot of work and it's very time consuming.
Yeah.
But last year, we did have somebody who did a nice model where they put small blobs of material around the surface of a large cell, and then did local contraction, attached local membrane protein densities to it. And that works. You can't simulate a lot of cells at once because you're expanding basically the trade-off is like with a variable power microscope. You go to high resolution, you don't see as much in your field of view.
Yeah.
But you can do that.
Okay.
And so that what the epithelial sheet model does is it makes a compromise. It divides the cells into 3 compartments: apical, medial, lateral, and basal. And then it says, okay, I'm going to link across from the apical compartments, like to stick to other apical compartments, medial lateral to medial lateral, basal to basal. Then you can define it to apical contractility. In 2D, that works very easily. In 3D, it's a little more complicated to make it work because instead of having 2 neighbors, you have 6 or 7 neighbors around the cell. But these are all, it's possible to do it and it does work. So in the simulations Julio did of polycystic kidney disease, he used quite detailed models of apical contraction. So it's possible to do that. The question is always, what is it you're trying to reproduce biologically, and how much detail do you need to reproduce it? And definitely in some of these cases you do. So I hope that answers the question.
Yeah.
So for example, for rosette formation.
Yeah, that's exactly what I'm focused on.
Yeah. Right. And I think Priyam, who I think worked with you last year a bit, is planning. She said she's very busy. She's a postdoc at Duke now, in an experimental lab. But she said she would try to join for an hour or 2 tomorrow morning to help continue this project. But definitely those rosettes are very similar to the ectopic cell-bite simulation that Julio built with Claudio Stern. And so that's a good, if you're in 2D, it's relatively straightforward to do. If you do it in 3D, you could do it, but it's a little more work.
Yeah.
Thanks. I hope that answers the. Okay. Any other questions? Wow, there are a lot of comments. There was a question: how would you recommend modeling an evolving virus in CompuCell? Well, I guess the question then is, what aspect of the virus do you want to model? If you're going to be modeling DNA changes, actually single base mutations, CompuCell isn't so good, probably. If you want to model things like ease of infecting cells, you could definitely do that. we tend to, when we do viral infection, we tend to use fields for it. And of course, the problem with the field is that the field is all the same. It assumes all the viral particles are the same. But you could build a model with explicit small, be more like a bacterial infection model, where you actually have small blobs of particles that move around and that represent the virus, and they could have different properties associated with them. Escape mutation and coevolution with hosts. Good question. But one of the problems with any kind of evolutionary model is that to make it realistic, you need a lot of agents, really a lot. And so when we do things like model evolution in cancer, what we typically do is we only model a few hundred or a few thousand cells instead of 10 to the 7 cells. And then in order to have things happen, you have to turn up the rate of mutation and evolution so that you see realistic rates of phenotypic drift in that smaller population. So those models tend to be interesting and suggestive, but it's hard to replicate. The problem with anything to do with, for example, cancer evolution is it starts with a single cell. And if it's a single cell out of 10 to the 7th, that's a difficult problem. I don't think any modeling framework yet so far can really address that problem without making some compromises. Now, the idea of using a different chemical field for each virus strain, that would be what you might be able to do would actually be to use well, if you don't care about diffusion, it's not so bad. You could maybe use a vector field where you can store multiple pieces of information in the same site. But that's a good question. We'd have to think about that. Modeling explicit strains of viruses is not an easy thing to do in a spatial model. And if somebody knows somebody who's done it successfully, I'd be quite interested in seeing that. If you have an example where it's not been done that you think would be interesting, let us know and we'll see what we can do. Are there any demos for today? Yes, there are plenty of demos. Please download the demos. Julio's not on the call yet, so please, but I think they should be uploaded. So again, the module numbers would be 4.4, 5.1, 5.2, 5.3. Yes, so if you want to talk more about modeling interactions of hosts and pathogens, that would be great. I'd be happy to do that. We certainly have worked on problems that are similar to that in the past and helped people out. Okay. Any other questions before I turn it over to TJ for to begin the presentations? Okay, well, I hope things go well. I'll turn it over to TJ for the MaBoSS.
All right. Well, hello, everyone. Good morning to all of you who are in comparable time zones to where we are. And let's do a quick couple of technical checks. Can you hear me okay?
Yeah.
Great. And can you see my screen?
Yeah.
Great. All right. Well, I'm very happy to be back with you again to start off our last day of the week. It's been an outstanding week and probably quite an exhausting one for everyone involved. But we certainly, as James has already expressed, appreciate you hanging in there, sticking it out, sort of drinking from the fire hose, as they say, with working through all of the content that we've blasted through during the week. We are going to continue in that trend to start the day today with doing 90 minutes of a very new functionality, as James said, which is doing Boolean network modeling in CompuCell3D using MaBoSS. Of course, we have the chat for questions, the buttons as well for moving along or slowing down, so on and so forth. So please feel free to use that to provide feedback as we move through this. We are going to spend the next hour and a half surveying what exactly is a Boolean network model in the first place, and what that looks like in the simulator that is integrated into CompuCell3D, which is called MaBoSS. We're going to look at doing basic MaBoSS modeling and simulations in CompuCell, in particular focusing on taking Boolean network models and using them in a multicellular context with cells that can move and interact with each other. And we're going to do all of that within the context of building a simulation together. So we're going to do as we've been doing. We're going to start in Twedit. We're going to work through the wizard and build a simulation from scratch, and sort of work through some of the basic modeling ideas and some of the functionality that's provided both by CompuCell and by MaBoSS itself, and sort of learn those basic ideas in practice. So I have to provide a sort of starting set of caveats for this module. As James has said, and I've already said, this is very new functionality. This is very new content and modeling capabilities for many of us, for us instructors. This, as James said in the pre sort of introductory discussion beginning at 10 a.m., this functionality was released just a few weeks ago in the newest release of CompuCell, which is to say that all of you are the first group of people who have ever done any sort of training on this content. So I feel particularly privileged to be delivering this talk and to see how this progresses and see how this is received among all of you, see how this experience goes from an instructing standpoint. And with that also, though, comes the burden of that we have not really done any training within, say, the Biocomplexity Institute, in that while I have done demos of this new functionality with the various members of our group, to my knowledge, unless someone has gone off and done some self-study, I am the only person currently on the planet who actually knows how to use this thing. And so, while all of the instructors are available throughout this module for support with everything that, we've been supporting with, if you're having issues with syntax errors or something crashing or so on and so forth, everyone is still here available to help with all of that. But when it comes to the actual details of using MaBoSS in CompuCell, currently there is only me. Again, to my knowledge, if an instructor has gone off and learned this and hasn't told me, please speak up, because otherwise we're going to have to operate on the assumption that when it comes to the actual MaBoSS functionality within CompuCell, we'll have to keep all of the support within this main room and during the talk. The exercise that we'll do is in part very simple due to this, to try to keep this very basic, keep everyone on track, stick to the fundamentals and not do anything fancy. There is documentation available to this effect. I'm going to put this link in the chat that's available on my screen right now. I'll point out that this documentation, while written, has not been formally approved in our review process. So if that speaks at all to how new this capability is, Here is the link in the chat. So you can refer to that for all of the capabilities that are currently available in CompuCell. That documentation is subject to change at any point during the review process, of course. And this is also capability that's under ongoing development. So, you know, new things may pop up from time to time in the near future, but you can refer to that for now. The Tarun asked which folder in the Google Drive has today's materials. So this will be, we'll grab the link to the root directory for the student materials. That's now in the chat. The slides for today are available under, I believe it's slides and then day 4. The final, I guess what you could call a solution to the exercise that we'll build as well. That's also available. So if we get lost or just totally derailed, or you have a power outage and get behind, it's totally fine. The end product of what we'll build is also available for your review, and you can find those. Yes, Giuliano has a link in the chat for the slides. That's now in the chat as well. Thank you, Giuliano. And lastly, and then we can move on to actually diving into this thing. I am not an expert in Boolean dynamics. I certainly have my math papers, but my background, at least in applied mathematics, is more in mechanics, dynamics, and then of course agent-based modeling. But the world of Boolean is very new to me. So I know that there are people who are attending today and who have been here this week who have been working more in this realm mathematically. And so we may come across questions that I just certainly can't answer because I'm still learning. And so for those of you who are more familiar with the mathematical details of these models, feel free to speak up in the chat and answer where I cannot. We certainly welcome that. Now, with all that in mind, let's actually do this. So a Boolean network model. We're just going to start with the pictures here. So look at this diagram here on the right. This is a network model describing the cell cycle. And if I'd not told you that this was a modeling paper, you might assume that this was something that was inferred from experimental data, in that someone could have set up experiments in their wet lab and actually inferred these relationships through various knockouts and so on and so forth, when in fact this is actually from a modeling paper. This is from a modeling paper using MaBoSS. And the whole premise of Boolean network modeling works with these ideas of Boolean networks. So in a Boolean network model, biological objects, say something like the amount of a species, like we spent lots of time with Josh and James considering yesterday, doing the ODE modeling using antimony and Roadrunner. The Boolean models are not so much concerned with what the exact value of, say, a species in a cell is so much, but more so interested in things that can be described in more of a Boolean state. Something like being activated with, say, a gene, or some sort of species that, say, is above some threshold to where it starts to begin to have an effect in some signaling pathway. Boolean network models consider biological objects in these terms. Something is either high, or say true, or say 1, or something is false, it has a state that's false. It's either low or 0, you know, or off. And each of those objects in a Boolean network then takes the form of being a node within that network. Those nodes being depicted here in the network in the top right on the cell cycle model. They're just, you know, these little ovals. They have their names. Those are the objects of the network. Each of them is a node, and then the network is connected through these various interactions. Is there a question? Okay, just wanted to check in real quick. So the network is defined with interactions between those various nodes. This consists of defining the logic of a node in that certain nodes can either promote or inhibit the activation of other nodes. And this is written in the typical form. So you can see self-inhibition here of cyclin E just by this notation here. And then interactions, say also inhibition by cyclin A, so on and so forth. And so the Boolean network models will actually explicitly write the interactions of the various nodes through logical statements. That something will change its state one way or another based on some certain set of logical, or some certain set of conditions that are written as a logical statement. And so to that effect, then the events in a network, the dynamics, is described as stochastic state changes, where during integration, rather than say describing the next quantity of some variable in an ODE model, instead what occurs is integration will go through and consider those interactions and determine, say, the probability of a node changing a state based on the interactions that's defined for it according to the states of all of the nodes that it interacts with. And that generates things like this. So this is a result from this cell cycle model work. And a network can then be described as something like binary. So you can arrange all of the nodes in a network in a particular order and then just write some sort of binary sequence that describes all of the nodes of the network as either being true or false in the form of 1 or 0. And you get all sorts of interesting things like what you could only hope for with a model on the cell cycle, that you get some sort of cycle where the network is moving through some sort of cyclic dynamics through the state space. And that looks like just a sequence of just a binary moving around in a loop like this. Boolean network models are very nice in that you can actually construct images like this, where you can go through and actually define every possible state in a network. And then you can define all of the possible transitions from one state to the next. You can do things like isolate what states are not achievable in the state space that's constructed by a network. Some people here, I'm sure, could tell you all sorts of other interesting things. I'm not going to dwell on it because I am not at all qualified to tell you all of the great things about Boolean networks. Instead, what I'd like to do is take this then into the realm of doing Boolean network modeling in CompuCell in a multicellular context. So when we
lots and lots of stamina.
David, can I ask you to mute real quick?
Olympic team trials in Eugene a few
Sorry,
weeks
we got a little bit of noise here. Thank you very much. So when doing Boolean network modeling in CompuCell, we get a lot of additional modeling capability, and we have the opportunity to investigate lots of very interesting phenomena, in particular, because when doing Boolean network modeling, it's certainly possible to, say, construct a network that considers, say, multiple cells. So we may be interested in considering the interactions between two different cells, each of which has a particular network running. What's great about this sort of framework is that we can have something like what we've been doing yesterday with the ODE modeling. We can have, say, a Boolean network model described in each individual cell. We can then go through and define the interactions between those networks, intercellular interactions that affect those networks and those networks contribute to. But in a dynamic context. So cells move, cells' environments change, and they affect their environments. And so having the capability of being able to model those interactions in a dynamic microenvironment, and to be able to define the contributions, the emergent contributions in this case, of the Boolean network models on the changes in the microenvironment. That capability is here presented now in CompuCell and readily available. And so we can do lots of interesting things like that. In this case, this is something like a coupled model of the cell cycle model that we saw on the previous slide, plus delta notch modeling. So coupling those two via intercellular interactions, and then using that in a simulation of wound healing. So we're actually going to build one of the aspects of this model shown here. Particularly, we're going to focus on the delta notch side of this modeling project. And you can see sort of what this looks like. Wouldn't pay too much attention to this for now, because we're actually going to go through and do this. But hopefully by now this may look somewhat familiar. So you can do things like still looping over our cell list, every cell in the inventory during the simulation, and actually grab the MaBoSS simulation that's attached to a particular cell that we had, say, attached to the cell previously in our start function, and then go through and actually relate the various states and model parameters of the MaBoSS simulation for that cell to other things that we've been working with in CompuCell, like neighborhood conditions, concentrations locally around the cell that we may be simulating with diffusion solvers. Yeah, this is what we're going to do for the rest of our time in this module. So to sort of get started on this, MaBoSS has its own simulation and model specification, just like CompuCell. So we have our cell list, we have our way of getting our field secretors and accessing our fields, and so on and so forth, describing the size of our lattice. MaBoSS has its own specification that CompuCell supports. So we'll create or specify MaBoSS models in the same way that one would do so just using MaBoSS. MaBoSS is its own simulator. It's its own standalone package. I can't remember if I put the link in the chat to their project page, so let me just do that now. Here's a link to the project page for MaBoSS, so you can get their software through their website. And you can do all this sort of modeling and a lot more than what CompuCell currently supports using their standalone package. But the basic model specifications are the same. And this is what it looks like. So for a MaBoSS simulation, there are there are exactly two sets of specifications that are used to define a MaBoSS simulation. The first being a description of the network of a model. And that looks like this little code snippet that's shown here on the bottom left. And then there's a second one that is a simulation configuration. And that's shown here on the bottom right. Now, in the case of the network, you can see a sort of representative example here. So the network specification will go through and define every node of the network, and then define all the particularities of that node. In this case, defining something like a formal expression for the logic of that node that defines its transition, and then writing the rate at which the node state can change, whether going from false to true or from true to false. Those rates can be explicitly defined and even independently, and can be parameterized according to other expressions that are written in the description for that node. It can be cross-referencing from one node to the next. So you can write, say, something like the logic of cyclin A being dependent on the node state of cyclin B, things like that. Now, in the case of the simulation configuration, this is sort of defining everything else that goes along with a MaBoSS simulation. Some of the specification in what I would call MaBoSS proper is not particularly relevant to its integration in CompuCell, in that some things that are specified in a MaBoSS simulation and the standalone package will define things like the duration of a simulation. In the integrated form, CompuCell controls that, in which case the integrated version of MaBoSS will just simply ignore those specifications. More importantly, for our work today, the simulation configuration defines things like the initial state of each node, which you can see a pretty lengthy list. This is again for the cell cycle model, writing things like that the initial state, which is written as I state of, say, cyclin D is 1, stating that cyclin D in the simulation will start as true. There's also the ability to, say, define model parameters that can be used to describe the rate of transitions of various nodes, and lots of other things. That all is specified in the simulation configuration. Don't get too caught up on the details for now, because we'll actually go through and do this. But most importantly, 2 specifications. One describes the network itself, and the other describes all of the numerical particularities and initial conditions, so on and so forth. Now, MaBoSS is currently an extended feature of CompuCell3D. So we've recently launched a new set of libraries for CompuCell that we call CC3D Extensions. And this is the home for projects like what we're surveying today, in that it's software developed by some other group or some computing technology that someone else develops that we like so much and think would be so relevant to the modeling community that we've decided to write support for it in one form or another within CompuCell. The MaBoSS plugin is part of the CC3D Extensions library, in which case, for those of you who have the local install, you may find that MaBoSS is not actually built in to your installation, and that's intentional. Now, there's some additional work that goes into actually getting the extensions library. I've outlined the steps here for your reference, not to go through it now, but rather for your reference, if you'd like to go through and actually get MaBoSS into your local installation on your own. Of course, I'd be happy to help with that. So if you have any issues, reach out to me. You have my email. Rather, the NanoHub tool has MaBoSS automatically built into that installation. And so what I'd like to do for today is that for those of you who've been using a local installation, if you could migrate over to the NanoHub tool, if only just for this module, that would go a long way in expediting the process with spending as much time as possible on actually learning how to do this new modeling capability together in a learning environment. For those of you who may not have the link stored for the NanoHub tool, there's the link in the chat. So if anything, for the next hour or so, I'd ask that those of you who are using the local install would indulge me and migrate over to the NanoHub tool. We've got a couple more minutes before we actually start doing anything, so feel free to listen to me in the background for those of you who haven't actually gone through and set up a registered account there. Now, the exercise that we're going to do from here on out is a poll going, I'll say. Go ahead and leave that running. We'll continue to talk. So we're going to start on this exercise, and as I've already said, we're going to work on doing something like delta notch modeling. Yesterday we did delta notch modeling with Josh using ODE modeling, writing an antimony model and defining interactions between a cell and its neighbors and how that affects the delta notch model running inside a particular cell. We're going to do something very similar to that for the next
Hello. Looks like TJ's web connection may have gone down for a second. Please be patient. I'm sure he'll try to connect. Let me see if he sends me a message about it. Give me 1 second to try to see what's going on. Okay, TJ's internet is down. He is trying to get it back. Jim, are you on the call?
Yes, I'm here.
If TJ can't get restarted in the next 5 minutes, could you possibly take over and present now?
I'm
If
really
you're not ready,
Yeah.
okay. In that case, that's fine. I just wanted to check as a backup. There's a question about demos ready to download. Let me see if Julio is here. Julio isn't here yet, so I can't ask that question. Juliano, do you know? Let me check on the demos while we're waiting. Is there anything else anybody wants to talk about as a group? Since a lot of people have joined since this morning, maybe I can just reshare those slides that I was talking about for the hackathon. This is the era of Zoom. I just wanted to say tomorrow for the hackathon, we start at 10 a.m. Eastern Daylight Time. There's a different link, a Zoom link for the hackathon. That link has been shared in the chat. Juliano can share it again. I'd ask people to go over their projects one more time very quickly at the beginning to help out people who are looking for teams. I'm also asking everybody to send me my email, or you put it, the best thing to do actually, put it in that hackathon document. In that shared hackathon document, if you have a team, give me a brief descriptive name for the team so that when I create the breakout rooms, people can find you, okay? That would be very helpful. And I also thought that for people, especially if you're not far along in your model development process, I strongly encourage you to read the very short one-pager called Thinking About Modeling Strategies, which may help you orient if you haven't done this kind of thing before. And then there is a more elaborate document which you can walk through that will help you define your model and goals called Workflow for Developing Mechanism-Based Computational Biology Models. I strongly recommend that. And then there is a worksheet based on that, which you can fill out to begin to flesh out your model. So I think that could be helpful for people. Somebody says, Polly says, I asked for these, how the hackathon work. Is there, would one person be on NanoHub and screen sharing, or is there a way for the team to work on different parts of the same simulation? That's a good point. There's TJ back. At the moment, there is no Google Docs for these simulations. Actually, if you we've been teaching modeling, CompuCell modeling using Twit. One advantage of using the Git editors is that they do allow you to do shared model development. The disadvantage of using the Git editors is that they don't have the code snippets, and they don't have all of the support that you get for specifically CompuCell modeling. And so if you're an experienced Git person and you want to use the Git editor and develop your models in Git, that's fine. But TJ might have a few words to say on that. But he's back, so I should let him take the session back.
My sincerest apologies. I had my first internet dropout right in the middle of a talk, so I appreciate all of your patience. Maybe it's time for me to get a new internet service provider. For now, let's not dwell on it and let's just get back going. I will happily weigh in on the pros and cons of using something like PyCharm versus Twedit, and be happy to discuss some of the internal topics that we've discussed in the development of CompuCell, say maybe after we conclude with this module, or maybe in private chat or during the hackathon. So it's a very interesting topic that's near and dear to my heart as both a modeler and as a software developer. So, but I digress on that. For now, let's get back to the Boolean modeling. I'm gonna blast past all of this surveying and say we're gonna build a delta notch model, and we're gonna do that using a Boolean network model. So let's go ahead and dive right into that. We're gonna build something like what we see here with the patterning of blue, red, and green cells. We're gonna do that sort of visualization as well. So let's go ahead and start right in. If everyone could move over to Twedit. I'm gonna get Twedit loaded up for me as well. I'm just gonna start with a blank project. I'm gonna split the screen. And let's just go through this together. So let's start by creating a new simulation. So we'll go to in Twedit CC3D project, and then new project. And let's give our simulation a name. I'll remind you, no spaces and no starting with numbers. So keep it all letters is your safest bet. I'm going to name my simulation MaBoSS Delta Notch, like so. So you've got your valid name in the simulation name field, and specified a place to save it. I already have mine set up. Let's click next. And what we will build for this module will be a two-dimensional sheet. And so our simulation will be 2D, and we're going to cover the entire simulation domain with a layer of cells. We're going to do all of our modeling and all of our simulating just within this 2D sheet. So let's do a periodic simulation domain. Our cells are going to be about 7 by 7 by 1. So let's set our X and Y dimensions to 98. Z dimension can just be 1 since we're doing 2 dimensions. So X and Y dimensions of 98 and Z equals to 1. And let's set up our periodic boundary conditions. So for X and Y, we're going to select periodic for the boundary conditions. And then everything else we can just leave as default. So X and Y dimensions of 98, boundary conditions of periodic for the X and Y boundaries. Now let's click next. We're going to create a couple of different cell types here in the CompuCell sense. And we're going to use those to represent the CompuCell sort of interpretation of our Boolean network model state. And so we're going to do this in a very straightforward way and just give some cell type names that correspond to the state of our delta or notch network nodes. And so we'll give some cell types. Let's do a cell type called inactive. And let's add that. Let's do one named delta. We'll add that. I'm capitalizing my names. It's not important so long as you're consistent throughout your specification. Let's name one notch. And let's add that. And then one more, we'll call it both. And let's add that. And this is to mean something like expressing delta, expressing notch, expressing both, or in the case of inactive, expressing nothing. So if you have your cell types, inactive, delta, notch, and both, then let's click next. We're not going to do any chemical diffusion in this simulation, so we can just skip past the specification of chemical fields. So let's select next again. And we've got a couple of plugins that we'll need for this simulation. We will do a volume constraint. So in the middle column for constraints and forces, just select volume flex. We won't do any specification in Python on the basis of cells, so we don't need volume local flex. We'll just use volume flex. In the left-hand column, under cell behaviors, I'm going to do just very basic contact modeling. So under adhesion, you can just select contact. And then lastly, we will use one additional tracker besides the center of mass that's enabled by default. We're going to do some neighbor counting. So under the cellular property trackers, we'll also select cell neighbors. Again, if you make some mistakes here, no problem. Everything we're doing right here is completely reversible. This is just building the simulation for us. So no worries if you miss something here. All of the instructors are taking note of which ones are active, and we can get those set up should you find that one of them is not available. All right, so contact, volume flex, and cell neighbors. Let's click next. And we're done. So let's select finish. Can double click the item in the CC3D project pane, and we have our simulation set up. Now let's start to configure some of the particularities. So again, we're going to have a 2-dimensional sheet, and we're going to have that cover the entire domain. Our domain was 98 by 98 along the X and Y dimensions respectively, and then no considerations along the Z dimension. So in this case, let's move over to the XML script, and let's go all the way to the bottom. Should find things that look consistent with what you specified in Wizard. Cell type plugin, center of mass, neighbor tracker, contact, and lastly, the uniform initializer. Now let's go to what should be around line 93, and let's specify the box that will define where our cells will be seated, just again, going to cover the entire domain. In this case, for 98 by 98, our box min will be the lower left-hand corner of the domain, which is the coordinate x equals 0, y equals 0, and z equals 0. And then we're going to fill our simulation domain all the way to the opposite corner, and that'll be for box max with a coordinate of 98. X equals 98, y equals 98, and z equals 1. So our box is from 0, 0, 0 to 98, 98, 1. And then lastly, we're going to initialize our cells as all being in the inactive state. We will set up our MaBoSS simulations to reflect that, and that nothing will be active. And so in the case of the CompuCell side of this simulation, we'll go to what should be around line 77 in your simulation script for the XML should be for types, and we'll remove what Twedit populated for us with all of the cell types that we specified, and instead just initialize this aggregate with inactive cells. 0, 0, 0 to 98, 98, 1, and only with the inactive type. Now let's go ahead and save and then continue on. We are done with the XML, so let's move on to our Stepables Python script. Should be the third tab along the top row in your Twedit GUI. We're going to start by defining the MaBoSS specification. So again, MaBoSS simulations are specified with a network specification and then a simulation configuration. And we're going to start by defining our network. And our network will consist of 3 nodes. And we'll walk through this as we write our models. So we're going to implement these like we implemented antimony models when working with Josh yesterday. We're going to do this with multi-line strings. So let's go up to the very top of our Stepables script. Should be around line 3. And let's just make a little space. Let's just hit return a few times. We'll just make a little bit of workspace here. So at around line 4, make some space between the import statement and our Stepable class. And we're going to start by declaring just the start of a multi-line string. This will be the network specification. Let's go ahead and name this multi-line string BND underscore STR. You want to use a different name, feel free to do so. Just make sure you do so consistently throughout your specification. I'm going to name mine BND underscore STR, and go ahead and start to declare this. And again, this will be a multi-line string, which is a set of triple quotes. You can use single or double quotes so long as it's the same in the pair. I'm going to use double quotes, so a pair of triple quotes, and let's go ahead and declare just our nodes. And as a reminder, nodes in a network specification for MaBoSS just takes the form like what's shown here on the right-hand side of my screen. I'm going to do this with you. So to declare a node, we first just write node. That's a keyword. And then the next thing we'll enter is the name of the node. In this case, we'll call it delta, no caps, just all lowercase delta. And then we can return and then just start a pair of brackets, curly brackets. And we can return that a couple of times. So we have node, the name of our node, in this case delta, and then just a pair of curly brackets with a little bit of space in between, and everything that defines what goes on with this node, at least in its internal description, will all go inside this set of curly brackets. Now let's do the same for two more nodes. The other nodes in our network, one will be notch, as you may have guessed. So after our set of curly brackets, let's return a couple of times and let's declare a notch node. So we'll do this by typing node and then notch. Again, you're free to name them however you please. I'm going to use all lowercase on this side of the specification. So node, notch, and let's go ahead and place a set of curly brackets again to do some specification for our node notch. And the third node in our network will be something to do with the intracellular particularities of delta notch signaling. And we'll just call this NICD. Those who are more knowledgeable on this signaling pathway are welcome to elaborate on all the nuances of NICD. We're going to blast past that for now, for the sake of actually being able to do this in time and say that it's important enough that it found its way into this model. So let's do a node for NICD. We'll just type node NICD. I'm doing all lowercase, and then one more pair of curly brackets. So our network has three nodes, delta, notch, and NICD. Declared it in a multi-line string. And as maybe some of you might guess who were very enthusiastic about yesterday's sessions with Josh, chances are at some point we're going to end up feeding that to some predefined CompuCell function to create some MaBoSS networks. For now, let's actually fill some of this out. So each of these nodes can define, again, things like logical statements that dictate things like the MaBoSS transition rates, whether from a low state to a high state, or from a high state to a low state. We're going to do this at least to the extent that we can actually write some expressions and then use those in other expressions within the specification for a node. In this case, we're going to write some very basic logical expressions. And in each of those nodes, we're going to do that by assignment to a node variable. This is a variable that's particular to the node, not to the entire simulation. And for each of those node variables, we're just going to call them logic. Now, the evaluated value of logic we'll use in describing the rates for the individual nodes. But for all three nodes, we're going to write a logical expression. We're going to name that statement logic. And in this case, we're going to write the following expression. So we'll say that delta will only go high if both notch and NICD are both low. And I'm going to rely on some of the experience with Josh yesterday on delta notch signaling and try and get us through this. So we're going to say that for the case of delta, delta will go high when both notch and NICD are low. And we can write that with a logical expression like as follows. So we can say logic equals and not notch. Not being written with just an exclamation mark. We use the names of the variables in this sense, they're actually referring to the state of those nodes when logic is evaluated. So when I say exclamation notch, what I'm actually saying is not the state of notch. And likewise, to do that for both not notch and not NICD, we can do double ampersand and then exclamation NICD. This says logic evaluates to not notch and not NICD. Now let's jump down to our third node, NICD. And we're going to declare logic for this node as well. So again, this is not the same as logic defined in Delta. This is only particular to NICD. And in this case, we're going to say that the logic for NICD is just equal to notch, which is that the logical expression for NICD just evaluates to being the same as the state of notch. And then lastly, we're going to write one for notch. And notch is special, and hence I've reserved it for last, in that notch will use a variable that we will actually specify on the CompuCell side of this simulation. And what we will do is we will use a variable that we'll refer to as delta underscore ext. So this is a variable that we'll actually set in the CompuCell side of these simulations, and we'll use that variable as set by the CompuCell side in governing the transitions of the notch state in the Boolean network. So in this case, let's declare another logic for notch. And in this case, we'll say that logic just evaluates to the variable delta ext as specified by the CompuCell side of the simulation if delta ext is greater than 0. This is also a logical statement, just like not notch and not NICD, or just like notch. In this case, we'll say that logic is equal to delta ext, and we can tell MaBoSS that delta ext is a parameter by first writing a $sign before naming the variable. In this case, $, delta underscore ext, and then make it a logical statement greater than 0.0. Now you'll notice I'm terminating all of these statements with semicolons. That is due to that this is all using C++ syntax, not Python syntax. So in general, in writing these expressions for the MaBoSS specification, you'll want to terminate all of your lines with semicolons. Question, can you repeat why the $sign is needed? Yes, so the $sign tells MaBoSS that what's been named here, in this case, delta underscore ext, that this is a model parameter. And this is a parameter that we'll actually specify a value for in the second half of the MaBoSS specification, which is the simulation configuration. But it's also something that we'll be able to specify in CompuCell as well. And we'll be able to change the value of delta ext at every simulation step. So was there another question? All right. So $sign tells MaBoSS that delta ext is a model parameter. It's also something that CompuCell will be able to pick up, that we'll be able to change in our step function. For now, we'll leave it at that and say that the logic for notch is dependent on whether or not delta ext is greater than 0. So let's continue. And now we'll define the second part of the node specification, and that is writing the rate of transitions from false to true or low to high, and also what's something like a downward rate, which is the rate of transition from high to low. or from true to false. And MaBoSS will understand these with the variables rate underscore up for the transition from false to true, or rate underscore down for the transition from true to false, high to low, 1 to 0. And so we can write these for each of our nodes. Let's start with delta. So we will have an additional variable that we'll define in our simulation configuration. And this will be something like a constant rate that will be applicable to some of the nodes in our network. And that parameter will be just called fast. So we're going to refer to another model parameter that we'll define the value of momentarily. And the name of that model parameter will be fast. So we'll also refer to this one with a $sign before it because it's a model parameter. Now let's start by defining our rate up and rate down, upward and downward rates, transition rates for delta. So let's go up to what should be around line 7 and return, and let's start by defining rate underscore up. And rate underscore up, again, is the transition rate when going from false to true for the state of this node. And in this case, we're going to say that the upward rate is fast, but only when the value of logic, as defined in the line before, is only equal to fast when logic is true. So the upward rate for delta will be equal to fast only when logic is true. Otherwise, it'll be 0. And we can do that as follows. So in general, we can write expressions that accomplish this by first writing the condition for the logical statement. In this case, logic. And we can refer to logic in this sense as follows. So we can first say rate up will be governed by some condition, in this case logic. And then we'll write the possible values based on the outcome of this condition. So when logic is true, we'll have a certain value that's assigned to rate up. And when logic is false, we'll have a different value that's assigned to rate up. And in this case, and in general, we have the form. There's a template here written here in the bottom left of the slide. And the form is to first write the condition and then follow it with a question mark. And that tells the code to first evaluate the condition. And if the condition is evaluated to be true, use the first value that follows the question mark. And if the condition is false, use the second value, where the first and second values are separated by a colon. So when the condition is true, rate up will take the first value that follows the question mark. And when condition is false, rate up will take the value of the second value that follows the colon. Okay, so let's do that here. And in this case, we'll refer to our variable fast, or our parameter fast that we've not actually defined yet. It's okay, we'll define that in a moment. Just believe me when I say that we will. We're going to say that when logic is true, rate up will be equal to fast. And when logic is false, rate up will be equal to 0. So when logic is true, rate up will be equal to our parameter fast, whatever it is when we define it. And when logic is false, rate up will be 0. Now let's write the rate for going from a state of true to false, a downward rate. And again, this is called rate underscore down for the node delta. And we'll say in this case that when logic is true, the downward rate is 0. And likewise, when logic is false, the downward rate is equal to fast. What this is essentially implementing is that when logic is evaluated to true, the state of delta will go high. And when logic is false, the state of delta will go false. And that's what we're accomplishing here. So rate underscore down equals, let's again use logic as a condition. And when logic is true, our downward rate will be 0. And likewise, when logic is false, our downward rate will be equal to our currently unspecified parameter fast. Now, we'll do similar things for our notch and NICD nodes. For our notch node, in this case, let's also define our rate up and rate down for notch. I'm going to move a little faster now. Let's do for notch rate underscore up for our upward rate equals, and we're going to use the logic for this node as a condition. We'll say at logic. And in this case, rather than using fast, I'm going to make the upward rate equal to our other model parameter, delta underscore ext. It's a question about the syntax here. So for those who've used MaBoSS more than me, feel free to correct me. But in this case, logic here is being referred to as a node parameter, whereas the $ here is referring to a simulation, a configuration parameter. Yeah, most importantly that here for, like the case here with fast is not updated every simulation step, whereas here logic is updated at every evaluation for these transitions. Okay, so let's continue on. For rate up, we're going to say that when logic is true, our upward rate will be delta ext, our other model parameter. And when logic is false, we'll say that the upward rate will be 0. Probably ask, what are the possible states of the node? True and false. For mixtures of rates and so on and so forth, lots of complexity having to do with defining the transition rates, still working within the context of true and false. So these logical statements, transition rates and all of that can be arbitrarily complex according to the specification of the network. We're still working within the context of true and false states. And yes, the values here for upward and downward rates, this refers to something like the probability of a state change on the node for an integration step. So yes, that's exactly right. Values do refer to the likelihood of a state change. Okay, now for notch, we said if the logic is evaluated to true, that the upward rate would be our model parameter delta ext, and if it's false, it'll be 0. Likewise for rate underscore down. Let's keep this one simple and also refer to our logic and say if logic is true, then our downward rate is 0. And if logic is false, then our downward rate for notch will be fast. Question in the chat, does indentation matter? No. Someone please correct me if I'm wrong, but no. Yeah, so this can all be done like that as well. Yeah. Okay, so when logic is false, or I'm sorry, when logic is true, rate down will be 0. When logic is false, rate down will be fast. So when logic is true, notch will tend to go high. And when logic is false, notch will tend to go false. Now, let's finish this with NICD. And NICD will have the same rates as delta. So for NICD's rate underscore up, we'll also refer to this node's logic, and we'll say that if logic is true, rate up will be equal to our parameter fast. And if logic evaluates to false, our rate up will be equal to 0. And likewise for NICD, rate down, say that if logic is true, our downward rate will be 0. And if logic is false, then our downward rate will be fast. So when logic is true, NICD will tend to go to true, and when logic is false, NICD will tend to go to false. All right, let's move right along. Let's very quickly, so we're running out of time at an alarming rate, start with the second specification of a MaBoSS simulation, which is the simulation configuration. Let's do this in another multi-line string. So below our network description, let's declare a second multi-line string. In this case, we're going to call it CFG underscore STR for configuration string. And we'll also do a multi-line string here. So a pair of triple quotes. And now we're just going to provide our model parameters that we've been working with, but have not declared, as well as the initial states of our network. And in this case, for the model parameters, we can again refer to them first with the $, and then say, let's declare fast. So our model parameter fast, we'll write $, fast, and let's declare that equal to 10. And for our second model parameter, which again will be set in CompuCell quite a lot, we'll initialize that delta underscore ext, excuse me. We'll just initialize that as equal to 0. Again, terminating all lines with semicolons. So fast is equal to 10, and delta ext is equal to 0. Second of which we'll be setting in CompuCell quite a lot. Now let's declare the initial state of our network. We can do this with something like an attribute, like we've been working with in CompuCell, where the name of the attribute for an initial state is called iState. And let's just declare all the initial states of our nodes to be false, or in this case, we'll just write a 0. We can do this as follows. So the initial state for delta would be delta.iState. And if we want to initialize that as false, we can just write that delta.iState is equal to 0. Again, that's referring to the initial state of the node delta. And we can do the same for notch and NICD. Let's also declare those as false. So let's write notch.iState equals 0. and nicd.istate equal to 0. So our fast parameter is equal to 10, our delta ext is equal to 0. All three initial states written as istate equal to 0. It's going to conclude all of these specifications, so whatever space you may have left before the beginning of the steppable, you can just go ahead and remove that. Maybe have something like what I have here on my screen. I'm going to move onward very quickly. We have a couple more things to accomplish here. First, we're going to do a little bit of work with the random library, in particular with doing some random or pseudo-random number generator seeding. So let's go up to the top, and just after our import statement should be around line 2. You can go to the end of what should be around line 2 where the import statement is, and return a couple of times. And let's go ahead and grab the import, or let's do the import of the random library. We can do that by just typing import random. We'll use that here momentarily. Actually, we'll use that right now. So what we will do is do a little bit of seeding of the random number generator, which is a way of making sure that every time we run a simulation, we'll get a different set of random numbers generated by the random number generators behind MaBoSS. If you're interested in the particularities, we can discuss that after this module. For now, believe me when I say that you can generate a different set of random numbers by below the import statement, returning a couple of lines and just typing random.seed. Call that as a function, no arguments, random.seed. Now, while you're doing that, also, we're going to declare one parameter that's particular to MaBoSS itself, which is describing the amount of time that elapses every time a state transition is considered. And when I say a state transition, I mean every possible state transition once. So every node that has a state transition defined on it, each of those will be considered. And the amount of time that that corresponds to in simulation is referred to as the time tick. Now, while we're up here at the top of our script, let's go ahead and declare a value for our time tick. In this case, let's give it a name. So this model parameter will hold the value of the time tick for our simulations. In this case, we'll call our time tick time_tick, and that will be equal to 0.2. Without specification of the actual units. This could be minutes, seconds, hours, months, probably not for a delta notch model, but in general, we're being intentionally vague here. We'll just say that time tick is equal to 0.2. We'll use that to configure our MaBoSS simulations, which I believe we'll be getting to next. So imported random, we called random.seed as a function with no arguments. That'll seed the random generator with the time on your computer, and then time tick equal to 2. Giuliano asks, do we need to seed, call random.seed? In the case of CompuCell, no. In the case of MaBoSS, yes. We may go through and add in similar functionality to CompuCell. CompuCell will do something similar on its own. That functionality is not written into the MaBoSS integration in CompuCell currently. So for now, we're just going to call random.seed. Okay, let's continue on. And let's go actually attach some MaBoSS simulations to some cells. So we've got our network. We can now build some actual MaBoSS model instances with our network and our simulation configuration. Let's do that similarly to how we did when we were working with Josh yesterday. And we're going to do that by using a function that should look similar to what was used with antimony model attachment to cells. In this case, we're going to use a steppable function that's called add MaBoSS to cell. So we had add antimony to cell. We have also add SBML to cell. We have add cellML to cell. Now we also have add MaBoSS to cell. And that function has quite a few arguments, just like with adding an antimony model. Those are all listed here. We'll keep those as a reference on the next slide while we're actually doing this, and we'll talk through it, which there are actually more arguments available than what are listed here, and you can refer to the documentation that was provided. A link to was provided in the chat earlier. So for now, let's just go through it very quickly. Describe a cell to attach to, give the model a name, just like an antimony. Pass our strings for our network specification and our configuration, and then we can define things like the amount of time for an integration step, specify the value of our time tick, and provide, say, something like a seed for the random number generator. So we're going to do that here now by moving to our start function. So if you've been working closely with me, this should be somewhere around line 50. I'm going to go to the comment that's automatically populated in the start function for our steppable. So let's go to the end of that string and return. We're going to work in the start function, and let's just do a loop over every cell in our simulation. And for every one of those cells, we're going to attach one MaBoSS model. So let's do that. For those of you who like the code snippets, the item is under CC3D Python, then visit, and then which one is it? All cells. I'm just going to type it. For cell in self dot cell underscore list. Iterating over every cell in simulation, hit return. Twedit should add an indent for you. And we're just going to call self dot add MaBoSS to cell. So for cell in self dot cell list, we're going to call on this cell self dot add MaBoSS to cell. Let's go ahead and start our arguments. The first argument that we'll pass is the cell to attach the MaBoSS simulation to. We can do that by typing cell equals cell. The first cell being the keyword, the name of the keyword argument, and then the second being the value, which is the cell that we're iterating over. Second argument, going to specify the name of the model by which we can refer to the model later when we do actual interactions with it. In this case, the keyword is model underscore name, and let's give it a name that's a string delta underscore notch. So model underscore name equals the string delta notch. That's the name of the model. We use that to get this model later. And now let's pass our network specification and our configuration. So third argument, to pass the network description, BND underscore STR is the keyword. We can just type equals our actual network specification string, which I just so happen to have named the exact same as the keyword, BND underscore STR. So that's passing the description of the network. Let's do the same for now the configuration. This will be CFG underscore STR equals the multi-line string for our configuration, which just so happens to have been named CFG underscore STR. And then lastly, specify three more arguments. The first being the amount of time that corresponds to stepping the model one time. That is the keyword time underscore step. So this is the amount of time per integration step, not necessarily the same as the time tick. We'll say time step equals, and let's just keep things simple and declare it the same as our time tick. now we'll pass the actual value of the time tick, which also just so happens to have the keyword of time_tick. And let's pass our time tick, which we declared to have the same name, time_tick. And then lastly, we're just going to generate a random seed for each simulation that's created. We can do that with the final argument called seed. And we're just going to generate a random integer from 0 to 1 billion. And we can do that with a function defined in the random library. And that function is randint, RANDINT. It's a little snippet right here for your reference. This will return a random integer between 0 and 1 billion. Excuse me. So we'll say that seed is equal to random.randint from 0. I'm going to cast 1e9 to an int. And then complete the arguments for the function, randint. And then complete the arguments for self.add MaBoSS to cell. So loop over every cell, attach a MaBoSS model, and I have the name delta_notch. It has our network and configuration strings. We specified a time step equal to our time tick. We've also specified a time tick equal to our time tick. And then we're seeding each MaBoSS simulation with a randomly selected integer between 0 and 1 billion. Okay, let's continue. I'm going to blast through these very quickly. So here there's a select list of ways to be able to interact with every MaBoSS model that's been created in CompuCell. So this is not something that's true for, say, MaBoSS proper. This is true for the actual implementation that's integrated within CompuCell. So for example, just like how we yesterday went through accessing SBML models that have been created from antimony model strings, we would type something like cell.sbml dot, and then specify the name of the model that we would like to interact with. The same is true for MaBoSS, except that now, in the case for MaBoSS network models, we type cell.MaBoSS dot, and then the name of our model, where the name of the model corresponds to what's passed here during creation of the model in the first place. We've been using delta_notch. So now to get the delta notch model attached to a cell, we can write cell.MaBoSS.delta_notch. That will return the MaBoSS simulation that was created by this call. Now with those MaBoSS model instances, we can interact with them in a lot of different ways. We're just going to look at a few today. But again, the documentation describe all of the other different ways that you can interact with those models. In the case of actually getting one of the nodes that we've defined, we have our delta, notch, and NICD nodes. If we'd like to be able to get one of those nodes and interact with it individually, we can refer to it like a dictionary on the actual MaBoSS model that we retrieved. So if we write cell.MaBoSS.myModel to get a MaBoSS model named myModel, we can get a node named NICD in that network by just using the name of the node as a key when accessing like a dictionary. And we'll go through a little bit more to do with that, but for now we can also set things like model parameters like our delta ext, or also our model parameter called fast that we specified in MaBoSS. We can also access those going a little bit further into some of the things that are defined on the MaBoSS model per se. And in this case, for interacting with the model parameters, we can use another dictionary type accessing method. In this case, if we have some cell.maboss.myModel for some MaBoSS model named myModel, we can get the name of any model parameter defined by typing .network.symbolTable. So in the case for our delta ext, we'll write something like cell.maboss.delta-notch.network.symbolTable, and then specify a key equal to delta ext. Now, if you use the code snippet, you may have some additional arguments that were generated for you, like having to do with discrete time. You can remove all of the other arguments that have not been specified here. We're just going to use the defaults. Discrete time in that case actually refers to some of the various integrators that are supported by MaBoSS. By default, discrete time is set to false in the case of the CompuCell implementation, and what that refers to is using a stochastic Gillespie solver when doing integration. Whereas if you were to pass discrete time equals true, then you would do deterministic integration. Okay, and then lastly, before we get back to typing, we had our self.timestep SBML to do automatic integration of every SBML model in simulation. There's a corresponding method for MaBoSS called self.timestep MaBoSS, also called with no arguments. So self.timestep_MaBoSS, no arguments. All right, now let's get to some coding very quickly. We don't eat up all of Julio's time for the next two modules. Let's head down to our step function and let's actually do this. So what we will do is we will go through every cell and we want to define the interactions of the delta notch models between individual cells that are in contact with each other. Delta notch signaling is contact mediated. So we'll do a loop over every cell in simulation. And then we'll look at every neighbor of every cell and we'll count all of the common surface area that a particular cell has with neighbors when the neighbors are delta expressing. In terms of the network model, every neighbor that has a delta node with a true state. And so let's do that. So if you use the TWEDIC code generator, you may already have a cell list started for you. We're going to use that. So step function is going to start with for self and self dot. And let's return. And let's start by just getting our delta notch network model for this cell. And we can do that by, say, let's declare the name of our variable MaBoSS underscore model and say that is equal to, in this case, it'll be cell.maboss dot, referring to the model name, delta underscore notch. So that is the MaBoSS model created by this call for this cell. And now we will do a summation over all of our neighbors for this particular cell. Our end goal being to actually set the MaBoSS model parameter that we defined earlier, the one called delta underscore ext. What we are doing here is we are calculating the current value of delta ext in our network model, and we're going to cast that as being in terms of neighborhood conditions of this cell, where delta ext will be the summation of the surface area that this cell has with every neighboring cell whose delta node state is high or true. And so let's do that. So let's name our summation variable. We'll just set it to 0 for now. We'll call it num underscore delta underscore ext. And we'll initialize that as being equal to 0. We're going to add to that as we do a loop over the neighbors of the cell. Now let's do a loop over the cell list or the neighbors of this cell. We can do that with Twedit in CC3D Python, visit, and cell neighbors. I'm going to write it manually. I'm going to write 4, and we get a neighbor cell object and the common surface area returned by this call self.get cell neighbor data list when passing our cell as an argument. This will return every neighbor and the corresponding common surface area with that neighbor that corresponds to this cell that's been passed. Now, the neighbors that can be returned includes the medium, which has no MaBoSS model. And so we don't want to do anything with the medium because it doesn't have a MaBoSS model to work with, the medium being represented with none in CompuCell. So in this case, we can only consider actual cells by just typing if neighbor. And then once we're in this conditional that's only looking at the actual cells in the neighbor list, now let's get the actual MaBoSS model of the neighbor cell. In which case we can do something like neighbor underscore MaBoSS underscore model equals. And we'll access it the same way as we got the MaBoSS model for the cell that we're currently considering. And we can do that by typing neighbor.MaBoSS.delta underscore notch, referring to the name of the model. So this is the MaBoSS model of the cell we're considering. This is the MaBoSS model of the neighbor that we're considering for the cell we're considering. And all we'll do is just count the common surface area, add that into our running summation for this cell, but only if the neighbor's delta state is true, and we can do that just by casting that state to 1 or to an int. Now we can get the state of a node as an attribute just called state. So again, to get the node of this neighbor, in particular the delta node, we can write neighbor MaBoSS model, and then like a dictionary, just pass the name of the node that we would like as a key. That is the delta node of this MaBoSS model. Now, if we'd like the state of that node, the current state, we can just type .state, just like that. So the delta node of the neighbor MaBoSS model, get its state. And we want to add the common surface area to our running summation if the delta state is true, in which case we can do something fancy like this. Let's just cast it to an int. If the delta node state is true, then casting it to an int will make it equal to 1. Likewise, if it's false, it'll cast to an int of 0. We can just multiply by the common surface area for this particular cell, which is returned in the iterator. And now let's just add that to our running sum, which was num delta ext. And we can just then say num delta ext plus equals, and then int of the state of the delta node in the neighbor MaBoSS model times the common surface area. Can they be neighbors and the CSA be 0? Question in the chat. No, no, they cannot. But of course, the neighbor's state for the delta node could be false. So that's the feasible way here that nothing is actually being added to our running sum for this particular neighbor. We look at a neighbor that's not expressing delta. Its node state for delta is false. But no, the common surface area will not be 0. Okay, so we have our sum. Now, after our sum, let's get out of this neighbor loop and get back to our cell loop so we can do a return after what on my screen is around line 72. Let's reduce our level of indentation back to alignment for the cell loop. And now the last thing that we'll do here is add that to the delta ext parameter for this MaBoSS model. And the delta ext is used when determining the logic of the notch node and also determining the upward rate of the notch node. And so we're going to set the notch or the delta ext model or model parameter for this network according to what this sum turned out to be. So this will be different for every cell. And so every MaBoSS model, every one attached to each individual cell could have a totally different value. And that's all according to the neighbor conditions of the cell. So let's do that. Let's start by referring to our MaBoSS model for our cell, MaBoSS model. And now, as a reminder, we can access the model parameters for a MaBoSS model by typing .network .symbol _ table. So the MaBoSS model .network .symbol _ table, and then like a dictionary, refer to the name of the parameter that we would like to do something with. In this case, it would be a string for delta _ ext. So that is the delta ext that corresponds to this MaBoSS model. And maybe you guessed it, we're going to set it to the summation that we just calculated. In this case, we're going to cast this to a float because MaBoSS will use these model parameters as floats. Just going to keep it safe here and just do an explicit cast to make sure we get no mysterious crashes. So let's say equals float and then num delta ext. All right, we're running way behind, so I'm just going to keep pushing through this so we can actually see this happen, if anything, so that everyone can just see it work on mine. We can work together after this module if you're more interested in diving into the details of this. We're just going to blast right through this very quickly. Next thing that we need to do here, so after we've updated the configuration model parameters, we're going to start a new line and go back to the level of indentation for the method for step. I'm just going to time step the MaBoSS models that have been created for this simulation. Again, we can do that with self.timestep _ MaBoSS. Call that with no arguments. That's like time step SBML. And now the next thing we're going to do, this is to finish, is to update the type of our cells based on the new state of our network. So we've updated the parameters of the network based on local conditions in CompuCell. We've integrated the MaBoSS models with those new parameters, and now we're going to have some feedback back from the network back into CompuCell by updating the types of the CompuCell3D cell types, and that's going to be governed by the state of the network for each cell. We'll do this by starting another loop over all cells or cell in self.cellList. The first thing that we'll do is we're going to get the MaBoSS model for this cell again. So we can say MaBoSS model equals cell.MaBoSS dot delta notch. That's our delta notch MaBoSS model for this particular cell. Now let's go ahead and get the actual state of both the delta and notch nodes for this MaBoSS model. Let's name them delta and notch accordingly. We're going to get the actual states, not the nodes themselves. We're just going to name them as delta and notch to keep this intuitive. So let's say, for example, delta, which will be the state of delta, equals, and that'll be our MaBoSS model. Let's get the delta node, again, accessed like a dictionary, using the name of the node as a key. And let's get the state, which again is dot state. So delta is equal to the state of the delta node of this cell's MaBoSS model. Likewise, the notch state is equal to MaBoSS model notch, like a dictionary, dot state. And maybe you guessed it from the very beginning of our time together this morning. I'm going to update the inactive delta, notch, or both types based on these two states. Inactive corresponding to when both notch and delta are false. Delta corresponding to when only delta is high. Notch, likewise, only when the notch state is high, and both corresponding to when both are high. And we can very quickly write that as follows. If delta, and then in this case, either notch can be high or low. If notch is low, just to say if not notch, then cell.type equals self.delta. Otherwise, cell.type equals self.both. So if delta is high and notch is low, then the type is delta. If delta is high and notch is high, cell type becomes both. Then if only notch is high, so this is in the case of notch but not delta, by virtue of the elif, then the cell's type is equal to self.notch. And lastly, by virtue of the else, now we're referring to when both delta and notch are low, we get cell.type equals self.inactive. So the CompuCell cell types change based on the current state of the network. That completes all of this with only negative 21 minutes to spare. Let's go ahead and try this out. You can open this in Player. Likely you'll have some errors and some bugs. I fully expect to find some in mine. But once we get that all figured out, then we'll have a working simulation. So go ahead and give everyone just a couple of minutes, work through your tracebacks, reach out to an instructor or just enter anything into the chat if you have some sort of issue. And of course, refer to the code that I have up on my screen for those of you who may need a little bit more time while I'm getting this loaded up into my local installation of Player. All right, give everyone another couple of minutes. I'm going to test mine on one of my other monitors so as to not interfere with anyone who may be using my code as a reference. And I'm going to debug mine as well. All right, so for the sake of time and getting to move on to work with Julio some more, I'm going to move my player over to my main monitor. And if you got through your debugging in time, then great. Maybe you'll get something like what I have. If you didn't, no worries. We can work together in the private chat on the break or in the hackathon or after hours, no problem. My apologies for this taking an obscenely long amount of time, but let's see this go. So I've already queued this up, and in my case, I've got inactive cells shown as blue. I've stepped this once, so this is beginning in the initial state. And in my case, when my cells type goes to delta, which was the case that corresponds to when only the delta node state of that cell is high, those will be shown as green. Red for likewise sort of downstream when only the notch state is true. And then they'll show as black in the transient state of both the notch and delta states being high. So let's see if it works. So we would expect from something like a delta notch model, like we went through yesterday with Josh, we'll get some sort of patterning, and what do you know, there it is. So we got delta expressing cells resolving contacts with other delta cells, where inevitably one cell or the other must switch their state. And so you'll see that here, except that in this case, you can do lots of interesting things where you say include things like migration that will bring two delta expressing cells into contact with each other, whether by collective cell migration or just plain motility, totally random. You see them have to resolve these sorts of scenarios. And so you'll see that where you have these very quick little switches to being inactive. Occasionally you'll see one where both states are high, in which case it'll show as black. But you see this sort of robustness where you can get this somewhat dynamic, but mostly stable pattern for delta notch signaling. Okay, James, I'm going to turn it back over to you now. Thank you all very much for bearing with me on this. Again, it was a pleasure getting to show all of you this very new, very cool work. And I very much look forward to being able to work with those of you who might be interested in this for your hackathon project.
Okay, thank you, TJ. Appreciate, of course, we lost some little time because of the internet problem. So you didn't run over quite as much as you think, I think. Please do fill out the end of module questionnaire. Appreciate it. And why don't we take a 10-minute break and be back here at 12:35 for Julio. Are you here, Julio? Yes, Julio's here for Julio's presentation. He's going to show us about links and compartmental cells and epithelia and these kinds of things, which I think people are quite interested in as well. How many people were able to get the MaBoSS demo to work? Just as a curiosity, maybe a show of hands. It's good. At least one person got it to work. That's good. Yes. Yes, so multiple people were able to do it. That's great. I have to say I haven't tried it yet, so I need to go back and listen to your lecture again and do it myself. Okay, good. Well, let's give ourselves a little bit more time. Let's say 12:40. We'll start back. All right. Okay. see everybody back at 12. Do fill out the questionnaire.
No,
Hello. I think it's time for us to start our class. Okay. I see that there are two hands raised. Please ask your questions. Farnoosh and Tarumendu, do you guys have questions?
I think that may be a leftover from the previous session.
Okay.
And if you could just remind people, please give the module number and say we're recording, that would be great.
Yeah. So, okay. So we will start now module number 5.2. Okay. And remember, you are recording this lecture for you to see later. And we are also live streaming this to YouTube. Okay. So if everyone is ready, we can start. So today we'll be in this module we'll be learning about links. So CompuCell allows you to add links between the cells and exert forces, and that can be used to drive many different kind of processes that cannot be accomplished with what we have seen before. So remember the Zoom logistics. You can ask to the lecture to go faster or slower, and I'll be just like before. I'll be asking you to say yes or no, or send message in the chat and make this interactive. Okay. All right. So the motivation here is based on a biological process called convergent extension of tissues, right? And I use that to do an intro into FPP links, and then we work from simple examples of how that work. They're not, in the simple examples, they're not really biological relevant, but only show how you can use those links in the CompuCell simulations. That second to last item there is a mistake. I will not be doing a cell crawling with links, but I will just go after the simple examples direct to modeling convergent extension. Okay, so let's now make a brief review of development. Okay, so on the left is the life cycle of the Xenopus levis frog, and on the right the life cycle of the zebrafish. They are both modeling organisms that's frequently used in many labs to study development. Each one has its own advantages, and in yeah, there's a hand raised. Please ask your question. No? Okay. So let's continue. And what you can see here, and well, and then you can also see the life cycle of the humans, right? So in all of these cases, we start with one cell, and then we grow up to be a full organism. That was true for us. That was true for the frogs. That was true for the fish, and that's mostly true for all multicellular organisms. Okay. And one of the key characteristics that mark development is that in all those cases we start with something that's symmetrical, and looks like a rounded shape, and eventually there is some step there where the tissue actually loses its spherical symmetry and becomes elongated, right? Okay, so we are not just a bigger ball; we actually have an aspect ratio, right? We have a height and So usually this process is, in order this to happen, the tissue must start to converge in a given axis and elongate in another axis. So there must be some kind of differentiation in the way the tissue grows, or the tissue should be remodeled to be elongated in one direction, usually in the anterior to posterior direction, and shorter in the other directions. Okay. This can be achieved by differential growth, or can be actually an active process where the cells pull one another in order for this to happen. Okay. So here is a very nice video showing the whole life cycle, or most of it, of a zebrafish embryo. Here you see the cell membranes label and also the nuclei in yellow. And if you watch this video over time, you can see first there is the epibole, the whole yolk is, and now you have convergent extension. You see that the cells are moving inwards, and once they move inwards, well, because they have a volume, they start to shuffle in this direction and thing grows. And then other things happen as well, like the notochord formation, this formation of the somites right here, and then the tissue continue growing, developing. And there are other places in the Xenopus development where you have other instances of convergent extension, but the very first one that happens more or less here is maybe the most visible. You can clearly see the cells going towards the midline and then elongating the tissue like in the other direction. Of course, you cannot really see this elongation very much in this video because it goes out of the range where the microscope can see, but it's happening there. Okay, so now one of the questions that we can ask, okay, well, so this is the precise definition of convergent extension, is the process by which the cells start to interdigitate with one another along a preferred direction, and when they do this, they, well, converge in one direction while extending in the other direction, right? So this happens in Xenopus, this happens in zebrafish, notochord formation, as you can see here. This can also happen in a 3D situation where the tissue converge in one direction and extend in another, and then the other axis can either be converging or extending, depending on exactly the mechanism. So this happens at the beginning of epibole in the Xenopus development. Sorry, Xenopus, no, the zebrafish development. And it happens in many other instances, even in tubules that is growing, this convergent extension process can happen. So one of the questions that we can have is how those cells are doing this, okay, and how we can model that, because many of you may have interest in, okay, this actually happened in my system, and I would like to model this, okay. So there are basically two ways which tissue can converge and extend depending on the type of cells you are dealing with and the organs that you're dealing with. One of them, which is the first one that was discovered, and that was a work pioneered by Ray Keller in University of Virginia, is that he found out that the cells in the Xenopus embryo, they start to intercalate, and right after blastophore formation, and those cells intercalate by actually crawling on each other, right, exerting forces and pulling themselves along the same direction, and that leads to the, well, conversion in one direction and extension in the other. Another completely different way to intercalate a group of cells happens when the cells are in a more epithelial-like phenotype, and this is a process very well characterized in the Drosophila embryo, where the cells have, well, they already tile very nicely the tissue, so it's a confluent and nicely packed, and then through some signaling mechanism, the cells at all the cell borders at specific direction start to have an enrichment of myosin, they start to constrict, and this constriction of that specific bordering leads to this T1 transitions, where you go from here to a 4 vertices seen together, and then as the tissue relaxes, it expands and becomes a border in the other direction, where before those two cells were touching each other, they contract their boundary this leads to this four-way junction, and then the tissue relax, and now these other two cells that were not touched before are seeing each other, and those two cells at the beginning lose connection with one another. Okay, and yes, T1 transition. Okay, and if that happens, if many cells, this leads to the conversion of the tissue in one direction and extension of the tissue in the other direction. Okay. Okay. So this is a illustration of how that happens in Drosophila. This is all the cells are marked, and as the things happens, you can see that the cells of the same color start to lose connection between themselves, and the cells of the other color start to interdigitate, and over time this leads to the extension of the tissue in one direction and conversion of the tissue in the other direction. Okay. So another way that the other process is by the cells actually crawling one another, and that's is a little bit more messy, right? Because the cells are constantly pulling one another, so it's harder to follow. But basically, the process is what is as illustrated here. The cells are initially, they don't have polarization, then they start to have protrusions. Some signal tells them to organize their protrusions and the crawling activity along a specific direction. And then they ask the cells pull on one another in a specific direction. This leads to the conversion of the tissue in one direction and again extension in the other. Okay, so some cells may actually be doing the both processes at the same time. That's the work by John Wallingford recently that showed that cells in the xenons actually can do both processes at the same time, and one reinforces the other. And in the video that you see below, you see how dynamic this process is. In red is the cell boundary, and in green is atomizing cytoskeleton of the cell that's actually constantly being assembled and disassembled and using to generate the force that pull the cells in a given direction. Okay, these things also happens in 3D environments, and this work that's done by James Sharp. So as the limb of a chicken develops, the process by the way the limb actually extends outside the body, right, is not driven by differential growth, but actually by active cell intercalation of those mesenchymal cells that make the limb bud mesenchyme. And so here you can see all the nuclei in blue, and some cells labeled in green in the outline. And you can see that they don't have it's clearly not the T1 transition mechanism, right? The cells are actually crawling one another and pulling one another. Okay, and this is happening in a 3D environment, and as this happens, the tissue converge in both in the anterior posterior direction and also in the medial lateral direction, in the dorsal ventral direction, and that leads to the extension the other direction, which is the medial lateral, or the limb is extending in this way. Okay, so there have been many models of convergent extension, especially for this mechanism of T1 transitions. The first one, it was actually proposed by James Glazier and his students Zajac and Jones in 2003, where the cells just have instead of have the same contact energy uniform distributed around all the pixels, the cells actually have more a differential contact energy depending on where they are in their periphery. Okay. What's the time scale of that movie? Which movie? This one here? 10 minutes, if that's the one that you asking, Lorenzo. Okay. Okay. Okay, so continue, and then there have been many other models, mostly done in vertex kind of models. And the only model that has been proposed to do this cell crawling version of Convergent Extension was actually one that, well, that me and James and Maciek did together, where we are not actually modeling the cells crawling one another explicitly, but you are only modeling the net result of this activity, which is, well, when the cells is extending filipode and pulling one another, what's effectively happening is that the cells are creating a link between the center of mass of each one and pushing them together. Effectively, that's what's happening. So, of course, this is a simplification of what's going on, but this can be done in CompuCell and is done using those links, plugin FPP links that you'll be playing today. And when you place if you do that with just one cell, the cell does not work. That does not work very well, right? The idea is that the cells will be pushing their neighbors on the right and pushing those neighbors on the left. But when all those cells in the tissue does the same kind of movement, that leads to the conversion of the whole tissue in one direction and consequently to the extension of the tissue in the perpendicular direction. Okay, so this is the same simulation on the left and the right, but on the right you only see the cell outlines. The resolution is not very good, I apologize, but you can see the model in action. Okay. So here, so this is the model we'll be creating by the end of this module, and we can also test other things, like ask questions like, okay, what if the orientation of the cells is not completely aligned, but a little bit random distributed, or what happens if you have other types of cells in your model that either you cannot make a pull or that do not pull the others but can be pulled. Okay, so this is two outcomes. So here's when the blue cells are refractive and cannot be pulled, and if you have enough active cells that actually can integrate with one another, that can drive the whole tissue to converge and extend. Of course, that those blue cells are only kind of lying behind on the side. And if you have only a subgroup of cells that can actually pull on the others, and the red cells can be pulled but they don't actually participate in the process, you can still drive the conversion extension of the tissue, even though you have only 10% of the cells that actually does the participating actively in the process. Okay, so this was actually very surprising for us that did this model. We are not aware that you need so little cells to be actually active to drive the whole process to work. Okay, so this is the plan of action for today. After this motivation, we will do a brief overview of how links work in CompuCell3D. We will do some simple exercise to illustrate how to add those in practice, okay, from the XML level. Another set of simple exercises to manipulate links from the Python code, okay, and then we're gonna try to do our final project of the model, which actually create a simple convergent extension model in CompuCell3D. Any questions before we proceed? No? Then let's go. So this is how the links work in CompuCell. Okay. So basically it creates a spring between the center of masses of two adjacent cells. Okay. That line here between those cells, you can view that as a spring. Okay, I sorry didn't have time to put a spring there. And the energy term that actually connects the two center of masses is just, it has this familiar form where it just basically D is the current distance between those two cells. There is a target distance or the ideal resting length of the spring, And then there is another term here which says how strong is the spring. So if the cells are already at the target distance between themselves, the spring exerts no forces. As soon as they are too close or too far away, the spring restores the cells back to their target distance. Okay. So it has the exact same for the volume constraint, surface constraint, length constraint. It's just that now what is actually being minimized is the distance of the cells up to some ideal distance that they should have. And then every spin flip, every pixel copy that deviates the center of mass away from the ideal distance between those two cells will be penalized. Okay? And every spin flip that drives them towards that ideal distance between the two cell center of masses is favored. Okay. Does it make sense? Okay, so when you create a model and you want to add those links to your model, the thing that you have to select in the wizard is this focal point plasticity option here under cellular behaviors. Okay. I'll be uploading the solution for all our exercises after this module. Okay, I have not uploaded anything there yet. The other thing is that when you are using this plugin, is usually a good idea to also have the cell neighbors selected, especially when you want to actually manipulate those links from the Python code. Okay, because you have to find which cells you're touching and create a link between them or delete a link from them. Okay, so this is how the plugin looks inside the XML code. Okay, the name of the plugin is Focal Point Plasticity, or FPP, as we refer to them, and it has here, of course, a bunch of parameters. One of the parameters is lambda here, which will tell how strong the constraint is, and the target distance, which say should be the ideal distance between those two cell pairs. Okay. The other things that is very important is that there will be one setting of parameters for each possible pair of cell types. So in this simulation, is all the cells that are of type my cell and my cell will have this kind of links. Okay, if you have another types of cells, cell A, cell B, cell C, the XML code will automatically generate one of these things here for AA, AB, BB. Okay, so all possible combinations will have its own set of parameters. Okay. The other thing is that you can also define a maximum distance that the cells may have. Okay, so if you have those two things that are linked, but from other forces in your simulation, the cells are being driven apart, you can specify through this parameter here when this link will break. Usually if the cell is being pulled on not too much, the link breaks and there's no more FPP link or spring constraint between those two cells. Okay, and the other thing that this thing allows us to specify is the maximum number of links of specific cell type. Okay, so this is meaning that only each cell can only make one connection with another cell of the same type. If you make this two, then the cells can actually have two links connecting two cells at the same time. Okay, so you can specify that in the code as well. Does this make sense so far? The other things that, yes, thank you for answering. But you can see that there are other parameters here, okay? Just like this activation energy and this neighboring order. And you may wonder, we will not be using this for this tutorial and this module, but just to briefly explain how they work. They are basically used to inform CompuCell what should be the criteria to create a new link. Okay, so if the cells come together, right, they maybe form a new link. Okay, but when you create a new link, you have a problem because before having a new link, there was no spring energy between the cells, and as soon as you create a new link between those cells that just touch together, this energy will be well, we will increase the energy of our system, right? So so the act of creation a new link after a spin flip increase the energy of your system, so it's likely not accepted by the normal Potts dynamics. Okay. So in order to actually make this actually possible, you have to add this activation energy where if you create a link, you have the new energy of the link itself minus this energy to make it happen. Okay, so this activation energy is a facilitator for the creation of new links. Okay, so usually use a negative, that's why it's a negative number, and that's why you usually add that. Okay, but usually 50 is enough number to allow the creation of links. We'll be just using that as default and do not worry about that anymore. Okay. The other thing is that you have this neighboring order, which tells how close the two cells have to be to each other before you attempt to create a new link. Okay. Stephen asked if you can make all these parameters specific for cell pairs. Yes, you can, and that you'll be doing, and you need to do that for tomorrow's Convergent Extension, and we'll be by the end of the module, you'll be talking about how to do this. Okay, yes, that can be done from the Python level. You can specify a different link between each different cell type parts. You can add links, delete links, modify the links. Okay, you can do all this sort of fun stuff. Oh, how many leaves are Neo-Hookean? Is there a way to add the parameters to model Neo-Hookean dynamics? There is a way to modify the this formula here from the XML level. I do not know. For example, you can, instead of having this 2 here, you can have 1 and make this an absolute value. So there's a way you can actually modify this formula from the XML code to exactly make Neo-Hookean. I don't know. Well, it depends on how you define the new, what's going to be the function, but yeah, but there are ways to go about it. Okay. Yes, and what else were I saying? Okay, and this neighbor order is just how close the cells have to be before you actually create a link. And a neighbor order equals 1 usually means that the cells have actually touched. Okay, that's all it means. Okay, but you guys will not have to worry about that. Okay, so let's now start to do an exercise, and this exercise will be, let's create a simple model with links. Okay, so everybody go to your Nano Hub or to your desktop, open Tweedit, and let's create a new CompuCell3D project. Okay, I'm gonna go with the next exercise a little bit quick, because by now you should all be very familiar with how CompuCell works by create a new file. Okay. Oh, before you do that, I just want to emphasize that these links can only work, okay, when you make those links, you are basically linking a specific point in space to another point in space. Okay, So they need to be anchored somewhere. So usually when we talk about a force between the cells, right, or constraints between the two cells, we are linking the center of mass of one cell to the center of mass of the other cell. Okay, that's mostly what we'll be doing in this in this module. That's everything that we're doing in this module. But you can also define a link between the center of mass on a cell and a fixed point in space. Okay, so you can actually create what is called an anchor, and you can use that. Okay, so that basically be saying, okay, maybe the cell is attached to the basal membrane behind it that you're not explicitly modeling, but you can, so it'll be hard to move the cell from its original position, although the cell is deformable. So that would be one way to actually anchor the cell to a specific point in your space, and that can be in 2 dimensions or 3 dimensions. So this distance does not necessarily need to be from one cell to another cell, but it can also be from one cell to a fixed point in space. Okay. So James is just adding a comment in the chat that he have used linkers to model elastic micropillars, for example. Okay. Okay, so let's go back to our simple exercise. So let's create our first model with links. Please go to CompuCell3D, menu, project. Let's call this module 52_examples. Okay. Give this name of the model. On this, let's just leave everything as it is and hit continue. Okay. No modifications here. You have to add a cell type as always. So give our preferred cell name. It can be your own name. It can be the name of your dog. It can be something more relevant. You can call it mesenchyme or epithelium or fibroblast or something like this. Okay. So here I just gave my cell because it's a short name. Okay. And I like it. Then we ignore the chemical fields, and here we have to select what we want to modify, and that will be contact, focal point plasticity, and volume flex. The layer estimation we just use the same as given, 256, 256. Okay. Do you want neighbors? Yes, we want neighbors. You can select that. So please select the cell neighbors. I don't think we need for this example, but feel free to select. That's it doesn't hurt. Okay. And after we select all these plugins, we can just hit complete and do the simulation. Okay. So once you do that, you can again open in the player. So right click or double click the module 5 on the name of the module on the under CompuCell3D project, ask it to open in player, and once you play simulation, you should see something that looks like that. Okay. Okay. So are you seeing something that looks like the figure in my slide? Yes or no? Yes. Okay. If you're not seeing, it may be because you don't have the FP visualization turned on. Okay. If you want to see the FP links, you actually have to go to in the CompuCell player visualization and select FP links, and then maybe you should be able to see it. Okay. So here, all those white lines here is actually the links between cell pairs. Okay. Maybe by your default configurations, your cell boundaries are also white. Okay. So you can fix that by going to click on the tools button, going to the cell color and change the color of the cell to be a darker color, and make the cell borders to be black, for example, or you make the cell colors to to be white, and then you make the FPP link here to be dark. Okay, so choose whatever configuration you like to better visualize the links. So we want to see the links in the simulations. Okay, so are you all seeing now, after you pull up the configurations, something that looks like my screen here? I got 3S, 4. Okay, so you may notice that not all the cells are linked together, just a few of them, and the cells only connect at most one pair. Do you know why? Well, okay, so what's going on here is that we are just using the basic configuration of the FTP links as automatically generated by the wizard, right? And it should be exactly as you see here, as well as in your code. So we, by default, CompuCell just gives a maximum number of junctions equals to 1. So what happens is that as the cells start to interact with one another through the pixel copy attempts, they basically make the link with the first cell it interacts with. Okay, and then you have the formation of all those links. Of course, because the pixel copy attempts is a random process, the order of which cells create their links is also random, and there is no guarantee that all those cells you have a available cell to link to, because this maximum number is checked both for the cell that's make the link and as well as the cell that's receiving the link. So you do have things like this lonely cell here that did not find any partner. Okay, so the first exercise that you can do, you can try to change the maximum number of junctions from 1 to 2 to 3 and see what happens. Okay, and while you can do that in the code, I suggest that you try to do that in the player, which is more fun. So go one by one, 1, 2, 3, 4, 5, and then keep going and tell me what you see. Okay, good question from Cicely. I don't know if that's the way to pronounce your name. What's the difference between parameters and internal parameters? Yes, they are both there. It's just that your cells can either be represented by a single domain or by a collection of domains. Okay, so remember the exercise with the cell migration, where the cell was made of a cytoplasm, a nucleus, and a front. Okay, that is a cell that has 3 compartments. Okay, and you can specify the links to be either between cell compartments that belong to the same cell or for compartments that belong to different cells. Okay, so these internal links or internal parameters is for links that of cell domains that belongs to the same cell or the same cluster of cells. Okay. Okay, so we will be doing that later in the module 5.3. We'll be seeing that again in module 5.3 as you make compartmentalized cells. Trying to change color white one has crashed. Oh, was not supposed to. Maybe can stop simulation, change the color, and then restart again. You can try that. Okay. Okay. So what you have seen is that you can keep increase the number of links, and as you increase that number, you of course get more links being formed between the cells. And but because it's a two-dimensional simulation with all the cells the same size, the cells tend to organize themselves in a hexagonal lattice. So having connections after 6 does not really add you more connections to your system. Okay. So did you all get things that look similar to those pictures here in my slide? I hope you did. Of course, the way the cells pack between themselves is not perfect, so you can still have cells with not all the connections that they wish. Okay, but as the cells move around, they should be able to connect all the neighbors. Okay. So of course, we only change one parameter of the FPP plugin. We can now also try to play with other ones. And one that's very nice and very relevant to play is actually the target distance that between those cells. Okay, so that should be that target distance here. You can either change here from the code or from the player itself. Let's change it and make it lower and then make it higher. Just choose a connectivity level, can be 1 or 6 or higher, and just try yourselves and tell me what you see. Okay, there's a question here. If you have a 3D tissue in CompuCell, would the connectivity level max out higher than 6? Yes. Yes. Then in 3D, well, in 3D, if all the cells have the same number of, same size, the tissue usually pack with how many neighbors? Does anyone know the answer? I think it would be 9, 12 to 14, I believe, right? Well, as far as I knew, the best packing that minimizes energy in 3D for it's an unsolved problem, but it'll be something between 12 and 14. Okay, Juliano should know. His master thesis was on foams, right? Okay, James Gray said 14. Of course, if you have some cells that are bigger, some cells that are smaller, right? Then some cells will have more connections, some cells will have less connections, and then you may want to use a higher number or lower number. Okay, so if you just put 30, then as you make as much connection as you can. But of course, when you have cells of different sizes, right, the typical distance between one cell and the other cell will be the sum of the radius of those two cells, right? So now we cannot specify a target distance that will be valid for all the cell connections because some cells are bigger, some are smaller. So that's something that, so some cells, the bigger cells will be under compression, right? So that's something that if it's the case, if the cells of the same type can have different sizes, you probably have to update those parameters for each one of those cells or each one of those cells pairs, and then you have to go into the Python code. Okay. Yes. Okay. So what happened? So what you guys saw? Okay. So let me show. Oops. Let me show here. So here's the tissue with a connectivity of 4. Okay, maximum number of junction of 4, and then I make the target is to be equal to 1. That led to a slight compression of the whole tissue. Now you're basically having two energy transfers competing with one another. You have the links try to bring all the cells very close, but you have the volume exclusion from the volume plugin that does not allow the cells to be brought that close. Okay, but that leads to a stiffening of the tissue as a whole. Okay. If you use a target distance that's bigger than the typical center to center distance of your cells, which in this case is 7, well, you're trying to make those cells now move apart. Okay. And you have this kind of crossed links that you see here, and I think that's what you're referring to, Farnush. Yes, this happens, and as you increase this distance, your tissue becomes rougher and rougher, and of course at the limit where you just make this unrealistic high, right, you start to get those those very weird shapes here that I don't believe there's any biological counterpart, but yeah, this can happen. Okay, so I'm just usually if the cells stop touching each other, right, that's when you basically want the link to break. Okay, so you can control that by setting the target distance to be a little bit lower than what it has by default. So by default, the maximum distance is set very high to 20. You can make this maximum distance equal to 8, and if you do that, as soon as you have a target distance that's higher than 8, the cells will move apart and then break the links. Okay, so having a maximum distance very close to the target distance in the XML code is a way to make the links very dynamic. So they are constantly forming and breaking. Okay, and then you don't have this crossing links as you see here on this picture. So do you all got those kind of outputs? Yes, okay. Okay, so you can see that now you have another tool to play in your simulations. Okay, how you get the output like the last picture? Okay, in the last picture I just make the target distance equals to 20. If you do that, eventually you start to get this kind of stuff. You can try and you should see something that looks similar. The first thing you see is that some cells start to evaporate, right? They're kind of expel from the system, and eventually you start to get those fingering kind of shapes there. It looks interesting. Maybe there is a biological system that behaves just like that, and this would be a good model, but I don't know any that does this. Any more questions? Can we proceed? Okay, so now let's do a second exercise. Let's do an exercise where you actually be dealing with 2 cell types instead of 1. Okay. Oh, Daphne said that make target distance 20 and 8 junctions and look fabulous. That's great. Okay, maybe you can share that in the Slack channel for us. Okay, so exercise 5.2, let's make a model with 2 cell types. Okay, I trust that by now you know how to create a CompuCell simulation without me going through all the steps. But let's create now 2 cell types. You can call them green and purple, as you can see here. I'll ask you, the first thing is to not allow the cells of different types to have links between themselves. Okay, so you should get something that looks like this. Only the cells of the same type have links. You can do that by either commenting that line of code where they have the same that specify the links between cells of the same type, or setting its maximum number of junctions to 0. Okay. After you do this very first one, there are those 3 other challenges that I give to you. Can you make the cell actually actively mix with links? And can you play with the FP link properties to induce a little bit of cell sorting. So Lucas asked a question: can we set the maximum distance less than the target so that they automatically break? Yes, you can. Yes. Then the cells will be constantly, maybe they'll never form a link, or maybe they'll form and break very, very, very soon. It depends on what is the typical distance of the cells. If you may target to be higher, but the breaking language be a little before, you form the link, the cells start to move apart, and then it breaks before the. So in this simulation, all the internal parameters do not matter. Okay, we will not be using them, so you can delete them or comment them out. Okay. So this is a good time to make a pool. I should ask my colleagues if it's okay to erase the previous pool and do a new one. Okay. So for those of you that finished the exercise, how did you go about inducing cell type mixing using links? Can anyone unmute yourself and talk, or write in the chat? No volunteer? Oh, your simulation crashed. Okay. So what do you guys think it should be done to make those cells mix with FPP links? Which type of pair should be connected? My cell 1 to my cell 2. Okay. Oh yeah, of course. I forgot to say, if you guys need help, please raise your hand or contact. Just ask, and we will send you to the breakout rooms. Okay. Forgot to give this reminder. Yes, Ajay, if you make the links to my cell 1 and my cell 2, and what about links with the cells at the same time? Did you turn on or did you turn off? You turn off, right? And was that enough to drive mixing? And you probably want to make their target length to be a little bit shorter, so the cells of the different types actually pull on one another. How many junctions you allow for them, for those cells? 2, 3, 4, 8. Okay, in the interest of time, I just go ahead and show what I got. So on the left is the simulation snapshot and the parameters that I chose to drive the mixing or this checkerboard-like pattern, okay, between cells using links. And on the right is a set of parameters that I chose to actually drive a sorting of those two cell types. Okay, so on the left one, I set the links between green cells and purple cells to be 0. If you just set the maximum number of names to be 0, the CompuCell3D will not create any links of that type of cell pair. And I just allow the links between the cells of different types, like Ajay suggested. And I made those target distance to be very low, 1, so the cells really pull on those. And by this pulling mechanism, you kind of generate some kind of movement of the cells and induce the mixing of the two different cell types. Okay, and on the right, I try to obtain the reverse effect, sorting those cells. So here, what I did is I just set the maximum number of junctions between green and purple to be 0, so cells of different type do not interact via links. And I set very low energies between target distance between cells of the same type, and I made one of them to be a little bit more stronger and have more connections in the hopes to maybe make all those green cells to go inside. That happens a little bit, but it's a little bit limited, the amount of cell sort that you can have through this mechanism. It's not as efficient as driving cell sorting to the additional energies or the contact energies of the cells on their surface. Okay, so okay, there are 2 comments here. Okay, on day 1 or 2, we discussed modeling sorting using difference in contact energies. Yes, that's usually what we do. Here we're doing that with links, but contact energy also plays a role. Yes, in fact, using contact energies is better to drive this kind of process than links. The links are not very good, not very efficient. How do you balance this mechanism if you were to incorporate something like this in our model? Well, you have to go back to your biological system that you're trying to model and ask yourself, okay, what's really happening, right? Maybe as the cells are sorting to the contact energies, they start to make tight connections between cells of the same type. So maybe your model has a mixture of this. The cells first sort out by contact energies, or what would be the biological equivalent to that, and then they make tight connections. Maybe, or in other words, the cells kind of start to create an epithelium, and then you can start to create the links between the cells of the same type. So that would be one way to add those 2 mechanisms together in the same model of cell sorting. And that will allow the cells, once they connect, to not break apart, or at least the cells that are more cohesive. What are the units of target distance and maximum distance? The units are in pixel. Oh, I should have said that. Yes, is in distance measured in pixels, right? And of course, if you know what's the pixel conversion into micrometers, that you have your unit in real units. Okay. Someone is asking for a breakout room. Please, if someone in the team could help, I appreciate. So I do not know if I answer your question. Or if someone in the course has something to add. The other reason that maybe it's not a good idea to use links to drive cell sorting is that usually the cells that undergo cell sorting, they are very mesenchymal and they are very in a fluid-like state, okay? And links is usually used when the cells are solid. At least this kind of passive links where the cells are connected and they make a strong connection by adding a link. That's usually a behavior when the cells have a very tight junction connection. So do for interesting pattern, are the parameters relevant for experiment? Well, that is a question, which parameters are relevant for each experiment or each specific system that you're studying. It's a job of the model themselves that has to choose, right? So in the case where the cells are actually crawling one another, you can see, okay, if this is the center of the cell and this is the center of the cell, how far they can actually extend the protrusion of Philippoid to bring the other cell. So you can set that to be the maximum range of the cell. So that is where it is a question that we ourselves cannot answer beforehand. It really depends on the system that you're modeling and what you're trying to capture and what you're trying to represent with the specific plugins and parameters that you're using, mechanisms, techniques that you're using in your simulation. Can those parameters play enable discovery? Okay. Like do a parameter scan and try to find for the set of parameters combination that gives rise to the desired shape. If that is what you're asking, they well, any parameters here in all the plugins can be scanned and varied to try to match desired output. There are ways to do that. CompuCell has a built-in parameter scan. You'll be learn about that later this afternoon. Okay, and you can also use more fancy algorithms with parameter scan. Not sure if parameter, but you can do other kind of searches of parameters to find optimal set of parameters that actually match your experiments. Okay. I believe Sluka has something like that. He has done that in the past. Does that answer your question?
Yes, definitely. Just very exciting.
Okay. Yeah, but we do have, yes, parameter scans built in CompuCell. Okay, so maybe can go move on. Okay, so as I said at the beginning, there are ways to control the links from the Python code. Okay, so all these examples that you have seen, you set them up at XML and let the CompuCell manage them. But sometimes you want to manage those links yourself. Okay, and to do this, you have to go to the Python code but before you can add any kind of manipulation of the links in the Python, you have to tell the XML that you want to do that, so it can let you do that. And this is done in the XML by uncommenting the local /tag in the XML code. So if you look at your XML code and go to the focal point plasticity plugin, you will see this local here comment out, a comment. You can uncomment, and once you do that, you allow your all those links that are being generated by CompuCell to actually be modified from at the Python level. Okay. Usually, when you are doing this local change in the links, you want to turn off all the links that the XML will generate to not have conflicts between what you want to do and what the CompuCell will automatically do otherwise. So usually when you add this local tag, you want to set all the maximum number of adjuncts for all the possible cell pairs to 0. So if there's any link that appears, it's because you told the mod to create them, okay, and not it was not created by the machine itself. Okay. So when you are when you are controlling the links from the Python code, tweak it, make your life easy for you. And there is this CompuCell3D Python option there in the menu, which you have probably already seen in the last few days. And there is a focal point plasticity option there. And when you click there, you should see all this list of all the fun stuff that you can do with links. You can create new links between two cells. You can update existing links, delete existing links, get information about links, visit all the links of a cell, as you see here on the bottom. You can also create anchors, which is going to be attach a link from a cell center of mass to a specific point in space. So you have all those different options there to play with. We do not have time to explore all of them, but you'll be exploring some in the next exercise. Okay. So one of the options that you'll be using a lot in this module and the next is to create an FP link between two cells and delete an FP link between two cells. So this is how those comments look from at the Python level when you use link management create FP link. It gives spit out this code for you, where the first entry is the cell that is making a link. The initiated here is the second cell at which you link the first cell to. And here the lambda distance and target distance are those two parameters of your FP link energy term. And the maximum distance, as you have already seen, is how far you can push the link and before it breaks. And the other thing that you can do, that you'll be doing, is choose this option, delete links attached to a cell. So when you call this option, you are basically erasing all the links of that this specific cell has. Okay, does this make sense? Okay, so with this, I will now set you the last exercise of the module as promised. Let's create a simple model of convergent extension. Okay, so to do this model, you have to create a simulation with a group of cells the same type. So you can go back there and say create a simulation, and when you add the cells, you just need one cell type. It can be mesenchyme, the name of the cell type, and you need to select volume, contact, FPP links, and enable tracker. Okay, so that first part is easy. The challenge here is actually doing the two other parts. And what I want to do is that every given Monte Carlo steps, after some initial time, the cells should delete all their current FPP links and create new ones. Okay, and the rules for creation new FPP links should be the links must be between cell neighbors. Okay, so for each cell, we have to go over all the neighbors of the cell and create links between them. But the neighbors have to be to the left or to the right of the cell. Okay, so links, the cells that are above or below the current cell, you should not make the link. You only should make links to the cells on the left or to the right. Okay, So to do this, you may, well, you have to calculate the distance and the angle that those cells are making with respect to the horizontal. So there are those two functions here that you can use. XYZ equals self-invariant distance vector between the cells. So as you go over your neighbors, you can actually get those coordinates, distance between the two cells. And then there is also this function from the math library in Python, where you can actually, given this y and an x coordinates, get the angle that this forms, and this function a tan2 returns a number between minus pi and pi. Okay, and then you can see, okay, to check if you're on the left, if your neighbor is to your left or to the right, and then if it is, then you create a link between them. So I'll give you a few minutes to set up the simulation. And if you need help, please, just ask for help from the team, and we can meet you on the breakout room. Okay, in the wizard should be in the second to last panel. If you go back here, should be on this one, cell neighbor, just below center of mass. If you miss that from this, there is a way to add that back in the XML code. If you go that to XML code, you can click in some place that is not inside any plugin, and then from the XML CML, you go to plugins, and then there is the neighbor tracker. You can select that, and you add that to the code afterwards. So most people, of course, need more time in this one. This was a hard exercise. So maybe we can do the following. I can walk you through my code to see how this is done. How about that? Okay, that'd be good. So I'll stop sharing and then I'll share. My tweeted. Can you all see my screen here? Yes, okay. So here's the, well, I call it tutorial convergent extension, right? So let's look at the XML code. So in XML code, I have all the basic things, okay? So this is by default. I just have one cell and I gave the name type 1 and I kind of regret that. But it has the volume plugin, it has the center of mass here, it has the neighbor tracker. Let's suppose you forgot to add the neighbor tracker in your code, okay? Oh, I forgot to click that, what do I do? Yes, have a question? Okay, so suppose you forgot to select the neighbor tracker, so I just delete the neighbor tracker here from this code. One thing that you can do, you can go to, you're not really seeing, new share, desktop, okay. You can go here to CompuCell3D right, plugins, and there's the neighbor tracker here, okay, and you can click and then you place the neighbor tracker there for you in case you missed, okay. So you have this, so if you miss something in the wizard, do not panic, you can go back and through this XML add back what you missed. The wizard just makes things easier for you, okay, but you can place it back later. Okay, so you have all these plugins, and then here you have the focal point plasticity plugin that we're talking about. So because we want to manipulate the links from the Python level, one thing that you may have missed is that we have to uncomment the local tag. Okay, so the local tag is uncomment, and that allows us to go to the XML, to the Python code, and actually change the links. The other thing is that I don't want the CompuCell to be making links for me. I want to have full control of the links. So I made those two parameters here for the parameters for the cell pair connections and for different cell pair connections to be 0. So the CompuCell will not do any links for me. I'll have to tell them through the Python code to the links themselves. So that's the first part that you have to do in your code. You have to make untag this, okay, and make those two parameters to be 0. Everybody follow? Is the should increase the font of my code or not? It is fine. Okay, if this finds is legible, I'll keep like this because that you can see more code at the same time. Okay, so this is the XML code, so that's all you need to do. You just have to allow your Python code to happen. Okay, now let's go to the steppables. Okay, so of course there are many ways to accomplish what you want to do. Okay, so what you want to do, this thing here that's on the back, right? So every given Monte Carlo step, delete all FP links and create new ones, okay? And then I want to follow these orders. So there are many solutions. This is my solution to how to implement this, okay? So I just went to the, I just used the steppable that the wizard already creates for me. And here I'm not using the start function, so I deleted. And here on the step function, I first check that after a given amount of time, so I allow myself to kind of move around a little bit. And then after, let's say 300 Monte Carlo steps, I tell all myself to delete the current links and I create those new ones, okay? So I could have everything done right there, right, inside this if statement, but instead I've created those two functions, okay, that I call here, which is one is delete links and the other is create links, okay? And what I do here is that in this create links, I go over all the cells in my simulation. So there is a loop here, and I choose the option remove all cell FP links. Okay, so I'm going over all the cells and deleting all the current links. So those, you don't need to memorize how to write those things. You can simply do, remember, use your CompuCell3D Python, visit all cells, okay, and you automatically get this for loop of over all the cells. And to the code to actually delete all the links of a given cell, you can just retrieve by going here to CompuCell3D ML Python, focal point plasticity, and then delete FP links attached to a cell, and that you delete all those links, okay? So it's right here. So I just repeat it, I'm gonna remove, okay? So this function delete all those links. And then I create another function that we're gonna create those links. So the function create links, notice that when you create those functions inside your CompuCell steppable, you have to, you call them by using the self in front of it. And in the function itself, you have to pass the self parameter, which is basically stating that it has access to everything that that CompuCell has. Okay, so in this self create links, I again go over all the cells in my simulation. Then, oh, actually, I'm not using this. Let me delete. I go over all the neighbors. Oh, sorry. And here I'm creating another function that returns all the neighbors of my cells. You can either use this or use directly this loop over for neighbors, okay? And basically, so this creates a collection of neighbors that you can use. And I'm going over all the neighbors, and then I'm adding to my list. So basically, I'm going over all the cells that are neighbor to my cell. Let me do this in a more normal way. Okay, this is a fancy way to do it, but let me do this the straightforward way. You just go here, go over all the cell neighbors. Okay, you get this link, and then you want to check first if your neighbor is a cell and it's not the medium, and if it's a cell, you also want to check. And well, if it is a neighbor, that's already enough. Okay, and if it is a neighbor, what you next want to do is you want to calculate the angle between the cell and the neighbor, right? So you get this function, get the distance from one cell to the other as a vector, right? And then once you have those x, y, and z components of this vector, you can calculate what's the angle that's made with the horizontal axis, okay? And here, I'm just to make things easier, I'm converting this angle into degrees, okay? So instead of having to be a number between minus pi and pi, it's going to be a number between minus 180 to 180, okay? And now that I have this angle, I can check whatever it is to the right or to the left, okay? If it's to the right, then that means that my angle is something that is a small angle between 0 and some maximum neighbor that I define. So here, on the top outside the function, I define those global variables called strain, target distance, maximum angle, maximum number of neighbors. Okay, we'll not be using all of them, but basically I'm checking, okay, if my angle or the absolute value of my angle is between this angle to the top or this angle to the bottom, okay, I create a link basically, but this will only make the links to the right, okay. You can also have those a good angle to the other side, right, where it's between, which is around 180 degrees up and down from it. So what I do a transformation of this angle back to center it back to 0 and check the same thing, okay. Is my angle on the other side within this angle range? So if it is within this angle range, either to the right here or to the left as indicated here, then I create a link between that cell, which I'm looping over, and the neighbor, which I just found, and with this given strength or this lambda, this target distance between the cells, and this maximum distance before the link breaks, okay. So, and when I do this, I can just run the code, and you should see the tissue conversion and extending, okay. So for how many of you did this work? No, the code's not on the drive yet. I'll put at the end of this module. Okay. But you can copy what you see here. You got conversion but not extension. Okay, that's interesting. Or maybe you have to wait a little bit and then you see the extension. We need to set target volume. Yes, if you don't have target volume, then this will just squeeze the cells, and yeah, then you have conversion but not necessarily extension. The extension part is a consequence of the conversion. So the conversion is the active mechanism. The cells are trying to converge, but because they also have a volume, they squeeze and then they escape into the other direction, okay? So if you're missing the volume plugin, you can go back here, and where's the volume? You can add it back. So if you missed the volume plugin at the beginning, Kato, you can go here again, CompuCell3D ML, plugins, and select volume, and automatically finds which cell types you have in your simulation and place the volume right there. So if you, hopefully when you place back the volume, it will work. Okay, so yeah, the conversion extension only happens because you have the conversion mechanisms, which are the links but that cells create, right? plus the volume exclusion of the cells, which force the extension part as a result. So yeah, so here's again the create the link code. Okay. So for this A10 to work, you actually have to go to import the math library from Python. Otherwise, it does not work. Okay. Oh, the line 38, the later part, yes. Let me extend this here. Is all this. Okay. Yeah, it's a very long command because you have to specify the source cell, the target cell, the strength, the target distance, and the maximum distance. So there are many arguments to this function. So in this case, you see that I made those links really strong because I set the target distance to be 0, but you can set a different target distance. Of course, if you make your target distance to be something that's higher than 7, then you'll be doing the opposite. You'll be drive things apart in one direction. That's something that you can guys try to do and see what happens, right? But usually you want the target is to be 0 or something, a small number, so you can actually be driving those two cells together. Okay, so for how many of you did this code work? Okay, it worked for you. Great, it works. That's great. Okay, let me run the simulation here. Opening player. Okay, so this is my simulation. Okay. Actually, I'm driving the links on the other direction. Oh no, I know what happens. Okay. So one thing that I asked you guys to do was to do this at every given frequency, a set up frequency to call to delete out links and create new ones. Okay. So you actually want to do a specific time frames. Okay. So I am. Okay, so the way you can do this is by setting the frequency here. So this steppable is only called every 100 Monte Carlo steps. So there was a question from don't you have to hold that for a few times Monte Carlo steps to actually have time to put those cells? Well, in this case, even if you have a frequency of 1, because you're constantly making those links again and again and again, you are basically keeping the same links forever. So every Monte Carlo step there is a link inactive, so it kind of works. So let me just run simulation. So here I'm running simulation. I'm allowing some time for the cells to relax their boundaries, and I'm creating those links. So let me make this zoom out. And you can adjust the parameters. I can make this angle a little bit higher. Instead of having 45 degrees, 55 degrees. That usually works better. So simulation, play again. So for this simulation, 55 works a little bit better. You can see the links forming and breaking apart. Okay, but over time you should see the cells to converging and extending. Okay, maybe I can make this even this target is to not be that strong. Oh, I didn't change that. Oh, okay, I changed the wrong parameter. Here, let me go back to my simulation. Okay, that should have worked better. If you start with a rectangle, that should still work. The only problem is that you have to maybe add a little bit more of time before you actually start the links to allow the cells to rearrange, and then it should be able to work. But it should still work with a initial rectangular shape. Okay. So this is the simulation working. I'll leave it here and allow you guys to see the code again. Okay, so did it work for everyone? Yes, yes, okay. So we already, again, over time, so you have to stop here. But things that you can do later, you can, besides have this simulation that always happen at the same direction, right? You can modify your code that you have some global direction at which the cells converge and extend. Okay, so maybe I can show you guys very quickly how you do this, because that is very fun. So the way to do this, where you can set up a angle here to be, let's say, 65, and then here, when you are actually, after you convert your angle to degrees, you can add this modification. Your angle actually be the, I can just copy here, right? You just add this global angle to the current angle between those two cells, right? And then you divide by 180, and you have to use the division with a percentage, which is the module division, and that aligns the two cells with respect to the global orientation. And then you can have a convergent extension instead of happening only the vertical direction. You can have at any angle that you wish. So let's say if you want to happening at 45 degrees, you just set this global parameter here, 45, and you add this code here. And when you do this, let's say you should see your cells not convergent extension in this direction, but convergent extension in the diagonal. Okay, please try this and tell me if it works. So all we need to do is define this angle here outside, and then add this line here where you actually rotate the actual angle of the cells and do the and check it like this. Okay, Ian, it works for you. That's great to hear. Okay, so I will try to see if it's working for me as well, right? I'll stop here, play again, and when the links start to form, okay, now they start at angle 45 degrees, okay, and now the tissue should now extend diagonally. Okay, so you can choose which direction it is. Convergent extension, converting, extending. And the other very fun thing that you guys can do is that instead of defining a global angle to converge and extend, you can define a cell-specific angle to converge and extend. So each cell is trying to put their neighbors at a specific angle. And then you can have regions of your tissue that converge in one direction and other regions that converge in other direction. Okay, so this is basically done here in this part of the code. I just define a start function where each cell receives a kind of vector, right, and it has an angle based on this vector. And here's the same trick, but instead of rotating the angle calculation by some global number, it rotates by the actual cell global number. And with this, you can have each cell to have a different rotation angle, and then you can have you can try to get different shapes in response. Like all the cells on the right try to convert extending this direction, and all the cells on the left try to convert and extend in the other direction. Then you can form a T, or you can form a star, or you can form many kind of very fun shapes if you try to do this. Okay. Okay, I know this went a little bit over, but I will stop here. I'll share this code with you to see how I went about coding this all the things, but I hope that the basics of how to manipulate links is clear, and you can actually try by yourselves later. Well, that's already the end of the module. I'll be with you again in the next module, where you're gonna try to use links to drive tissue folding. Okay, so I'll stop here. If there okay, we'll start back at 2:50 in 25 minutes, right? And I'll stop sharing, and I'll upload all this code to the student material so you can have it and check by yourselves. Okay, thank you for your attention, and well, we should have the lunch break now, right? It's already late. Sorry for taking too long.
I actually have a question about this
Yes.
about this convergent extension model. So, because this model, I know this model is kind of like a simple version of convergent extension, but I'm wondering if, because I mean, like the tissue is doing a global convergent extension, which is the right way that it elongates along one axis and narrows along the other. But I'm wondering, because the cells doesn't look right, because during the convergent extension, the cells is actually polarized, not like along the elongation axis, but like perpendicular along the elongation axis. I'm wondering, is there a way to do that?
Okay. Okay. Yeah, that's a good question. Usually, well, you can add the cell elongation plugin, right, to make the cells be elongated. But when you do that on top of this, you usually get the cells to be elongated in the wrong direction, because you are basically putting two things together that are elongated, and then what they do, they should try to do this, right? So
Okay.
you get the wrong shape, right? So so when I used this model, I was most interested in, okay, what's the large scale effect of the cells trying to circulate in one direction. So for me, when I try to do that, you do the lean bud paper. I didn't care much about the cell shapes because the cell shapes are super complicated in those limbs to begin with. So maybe I can share my screen again. Sorry, it's kind of hard to find where Zoom is when you're trying to screen share and have a. Okay, so for example here, those cells. Look at the shape of those cells. There's no way I can do that in CompuCell if I want to model the whole tissue, right? So what I did, we do not care about the specific cell shapes, but we care about what's drive the whole tissue to converge and extend and elongate one direction. So we create a model which each cell has a polarization vector, and they use this polarization vector to put those cells in their neighborhood, given some rules, and then we only inquire about the net effect on the whole tissue, and not and we didn't care about reproduce the cell shape, the specific cell shape. So in some situations, you want to converge and extend your cells, and you want your cells to actually be elongated in the correct direction. Okay, that will be a little more challenging. There are probably ways to do that, but that will require more work. But you also have to ask yourself, okay, do I really need this? And maybe you have some application in mind, but it's absolutely essential that the cells have the correct shape as they do this. Okay. Maybe we need to add compartments to the cells. Okay, that would be one way to do it. And then we have a compartment cell on each side that actually these are the compartments that actually throw Filipodia, right, and have the links and pulling the other cells together. Okay, so that would be one way to do it.
Yeah, that makes sense. Thank you so much.
Okay. So those cells are intercalating, right? So if you look at this over time, those cells are exchanging neighbors and intercalating and interdigitating, and then so there is cell intercalation in this model, or maybe you need something else or you mean to bring together those two models of the cells that throw Filipode and pull each other, and also shrinking their junctions. Is that what you meant? So if you meant that, okay, but well, the question that you did not make, it's also a good one. Let me go back here. So I told you that John Wallingford from University of Texas. Okay, I don't know exactly which university, but he's in Texas. He said that some xenopus cells, tissues, actually undergo intercalation with using both mechanisms. But the catch is that this mechanism is happening at the basal side, and this mechanism is happening at the apical side. So they can happen in two. The same cells can be doing the both things at the same time, but they usually happen at a different level. Some things happen at the bottom, and these other mechanisms happen at the top. Okay. So if you want to do that CompuCell, you first need to do a three-dimensional epithelial tissue, and then you can add one mechanism that happens at the bottom and another mechanism happening at the top. Okay. Yes, in both cases, if you look closely, the cells are exchanging the spatial locations, right? So here they are maybe a little bit 4x4, and now this one maybe went to the top, this one went to in between those two, and this one went to the bottom, right? This something like this more or less happened here a little bit differently. So, but they in both cases they are intercalating in some sense. It's just that on the right, the T1 transition is more clearly visible, and the one on the left is kind of hard to follow because the cell shapes are very regular.
Hi, Julio. Is it Julio or Julio? Sorry.
Julio. But it's okay.
Julio. Okay, great. Yeah, so in the model of intercalation I'm studying in zebrafish, you have this convergent extension, as you know, but usually you end up with like cell layers which are about 2 or 3 cells thick, and you can see intercalation as in, but not cells simply pushing together, like in both these cases. For example, the cell that's on the, let's look at the one on the right here. The cell that's on the far left stays on the far left, right?
Ah, yes, that's
and
true.
in the other picture, like let's say the one on the top left, or in fact, all of the cells come into the middle, but none of the cells on the left end up on the right. But in the model that I'm studying, you have cells that start on the left and simply switch with cells on the right.
Oh.
They seem to have some intrinsic understanding that they should be in some order, if you see what I mean.
Oh, I see.
I don't know how they do that, but they do seem to do it. And I wondered if you'd ever seen that happen, where cells literally just move from one side to the other.
So they not only interdigitate, but they pass each other.
Yeah, exactly. Yeah.
Okay, that sounds very interesting. You see that in zebrafish, you said?
I can send you a paper if you like.
Okay. Oh, that's very interesting. Yeah, please do. I'd like to see that. Yeah, that's for me. So just that's something more like on the bee, right? That the cells are calling each other and going to the other side.
Yeah.
Usually,
well, it seems that they exhibit this convergent extension to some extent, and then they also just interplay with each other once they're there.
Okay, there are probably other mechanisms at play, right? Because if you just have this convergent extension, the cells over time they maybe end up in a different side, but it should be a little bit random, right? You do not know a priori if they're just pulling randomly from their neighbors where they're going to end up being. So maybe there's some kind of chemotaxis, maybe there's some kind of addition, difference in addition that makes those cells to, once they found that boundary on the other side, they like to stick there, so they go there. So there's some kind of sinking mechanism, or maybe those cells here do not like those whatever is on that side, and then as they intercalate and move away, they don't go back.
Yeah, so you think there could be some way to sort of
drive
some sort of superposition of mechanisms.
That'd be my guess.
And you can do that in CompuCell3D, no problem.
Oh yes, you this is one plugin, right? One mechanism. You can
Yeah,
also have the
yeah.
contact energies on top of it. You can have the chemotaxis on top of it.
Okay.
You have the cell can switch type in the middle of the process, right?
Mhm.
So you have all those tools in CompuCell. It's up to you to mix and match them to fit your model. But
Yeah,
that
great.
sounds like an interesting thing to try to study.
Great. I'm just gonna see if I can grab that paper for you.
Okay, good.
Oh, I found it actually.
Okay, you can send to my email. I think.
Oh, okay. Sure.
Put it in the chat, please.
Or put the link in the chat. I think other people are interested to see.
Yeah, I wish you could send papers on Zoom. I'm just gonna find the link for it.
Yeah.
I've got the DOI here if that helps.
Oh, DOI should be enough. Yeah. The mechanism on the right, the T1 transitions usually it happens once and that's it. Okay.
Yeah.
Cells undergo one round of this and that's it. And that's the end of it. At least in the Drosophila extension, that's the case. They only go one round.
Right.
Because usually when you get rid of this, you only end up with links that are either this or in the diagonals. Okay, so then you have to add another signal mechanism on top of that to redefine other boundaries to intercalate. Okay, to shrink.
Yeah, that makes sense.
Okay, so we'll be back in 10
10
minutes. I'll myself take a break.
Yeah.
And I'll be again with all you guys in about 30 minutes to talk about other things that you can do with links. Okay?
Great. Thanks, Julia. Bye for now.
So hello. Let's go back to our next module, module 5.3, cell compartments and tissue folding with links. Okay. As people get back, I remind you that this course is being recorded and is being live streamed to YouTube at the same time. I wonder if there's any leftovers questions from the, I think, are we going to cover parameter scanning today, or will that topic be dropped? We hopefully will cover it today. Okay. We still have one module after this one, and Jim Sluka will talk about this and some other things as well. Okay. Can you visualize cell strain in these models? Yes, you can. Yeah, well, your question assumes that the cells are being pulled, right, in a specific direction, and I assume that they are being extended that direction. There is a tracker called moment of inertia tracker where you can get it, you call, and then you can get the major and minor axes of inertia of their cell, so you can get a sense of how long the cell is stretched in a given direction. So you can divide that by what the cell was supposed to have, elongate in that direction. So you get this number, and you can feed this number right into the scalar field tracker plugin, and then you should be able to see the strain of the cell. So the link for this presentation is already on the student materials. Okay. You can also add strain as pressure, right? So you can plot. You can also, well, that's not strain, right? You can also get the pressure of the cell by making the difference between the current volume of the cell and its target volume. Okay, and that times the lambda. So that should be the pressure of the cell, which either be compressible or it cannot be positive or negative. Okay. The problem with the model inertia is that basically is approximating your cell shape to ellipse, right? That may be not what you want to do. What you want to do is actually the cell shape, but then you have to kind of go over all the pixel locations of your cell and see the one that's most to the right, the most that's one or to the left, and then get the actual length of your cell in a specific direction. Yeah. Okay, that's a good question. Answered by Pedro. Okay. Ellipse is good enough. Okay, then it makes your life easier. Okay. I believe I'm sharing screen, am I not? Okay, is there any other questions before we start?
Can see your screen.
You cannot see my screen. Can you guys see my screen or not?
Yeah, we can
We
see.
can see a PowerPoint presentation.
Yeah, it should be a PowerPoint presentation, and this is the Zoom logistics.
Yep, that's right.
Well, now I see it. Yeah, thank you.
Okay. Okay. All right.
You are muted, Julio.
Oh, how do I mute myself? Okay. So
Alt
so
A,
here,
maybe. Maybe you pressed Alt A. Alt A.
okay. It can be. Can you hear me now?
Yes, yes.
Okay, good. So the objective of this model is to make a simple model on epithelium, and then make the epithelium apically constrict and induce a furrow formation. Okay, not completely fold. Well, I know that I said fold at the beginning, but we're gonna just grate a furrow. Okay, to make it actually fold inside, you have to add more stuff. You can try that later. Okay, So usually you have an epithelium, right? So the first thing you have to do is build an epithelium, and an epithelial cell is usually made of have an apical side, a basal side, this lateral. So to build this epithelium that you are trying to fold, we have to actually use the notion of compartmentalized cells. Okay, you have already seen a little bit that in the cell migration, but I'm not sure if you actually have seen 2 or 3 or 4 cells that are compartmentalized together. When you do, you have to be careful about how to set up your contact energies. Okay. So these are some examples of epithelial tissues undergoing morphogenesis. So some structures that, well, their cells are compartmentalized, have an apical basal side, and sometimes those tissues can undergo changes like this folding process that you see on, for example, neurotial development. On that one, you have many folding events actually going on here, and or something a little bit simpler, which is drosophic gastrulation, where those cells at the ventral mesoderm apically enrich their ectomizing network, and that leads to contraction of that size and furrow formation and further invagination of those tissues. Okay. Is there a role of curvature in the fold? Probably there is. Okay, so those are topics under investigation, and that really depends on which system you're talking to. Okay, and of course, then different people have different opinions what's going on. Okay, but here we'll talk about tissue furrow formation or folding in general terms, right? It's up to you guys to adapt to your specific scientific question. Okay. So if you want to model epithelial tissues, if you not only care about that the cells should be elongated, right, as you can use with length string, but the different sides of the cell should have different properties, then you want to actually think harder about the apical side of the cell, the basal side of the cell, and what's not either apical or basal, which is the lateral side of the cell. So epithelial tissues are apically basal polarized, as we talked before in the first day of the course. I talked a little bit about this. So maybe you want to set our cells, subdivide our cells into compartments, and call one apical, one basal, and one in the middle, which you just call lateral, and assign different properties of each one of those compartments. Okay, so usually the apical side faces the lumen or some cavity or some fluid. The lateral side of the cells is actually in contact with the neighboring cells in the epithelia, and they have tight junctions. And the basal side cell is in contact with the ECM or other cells, that if you are talking about stratified epithelia. So, but still, it has different characteristics than the side of the cells or the apical side of the cell, right? So here you want to do 2 things. First, create an epithelium, a stable epithelium, and then once you find a stable epithelium, which is actually some kind of work, you want to instruct this epithelium to actually apically constrict along some of the cells, apically constrict and deform the tissue. So the objective of the model is to end up with a simulation that looks like this. Okay. So what you can see here, you have a line of cells, right? That each one of those cells has 3 compartments. Okay, so it has an apical side in red on the top, a lateral compartment in the middle, and a basal compartment in blue at the bottom. The apical side sees the medium or nothing, and the basal side see this whole grayish thing, which you call just the ECM. Okay, it's kind of very thick ECM in this case, but this is a toy model. We just want to make something very simple for that you guys can play with. Okay, and what you're not seeing here in this movie is that there are actually links between all those red compartments, which are the apical compartments of those cells, and all those cells here within this region outlined here. At some point, they decide to, we assign different properties to these links, which drives the tissue to, right now, to those cells to constrict and induce that folding. Okay. So we'll have to using two concepts in CompuCell, the concept of the compartments, how to compartmentalize a cell and make cells of different compartments, and we have to be careful to not the cell, this compartment has to know that this is the other compartment that belongs to it, and you should see this other grain here very different than it is its own grain. Okay, so you have to, when you create compartmentalized cells, you have to duplicate many of the parameters that we have, that we would normally have. Okay, we will be playing with the contact energies to make this stable, and then we will play with the links to actually connect the cells and actually drives those chains. Okay, does it sound good? Okay, so let's continue. So the first thing you want to do is to create a new project where you're gonna create this epithelium, right? So this is the first exercise of this module. So I'll go with this a little bit quick. Let's go to CompuCell3D project, new CompuCell3D project. Let's give the name of our model, module 5_3_ tissue folding. Okay. Hit continue. And now in this general generation properties, you actually want to change those numbers here. So please, you usually have X and Y as 256. I want you to make the X 350 and the Y 100. Okay. And the other thing I want you guys to change is to change the periodic conditions or the boundary conditions on the X direction to be periodic. So the idea is that you have a simulation space that's very long on the X, and in this X direction you want the cell here on the end here to actually be seeing the cell on the bottom end. So it's periodic boundary conditions on in the horizontal direction. Okay. And we can leave everything else as it is. Okay. So did everybody do this change? 350 here and 100 here, okay? And the X has to be periodic. So I'll continue. Now the next page, the cell types. You want to add 4 cell types to the simulation. Because our cells will be made of 3 types, and you want to differentiate between what the cell sees on the top and what the cell sees on the bottom. So on the top, they will see, above them, they'll see the medium, and below that, they will see the ECM. So you want to add apical, lateral, basal side, and also another cell type which we'll call the ECM. Okay, so make sure that you type all those things and hit add to populate the table there. And once you have all your 5 cell types, right, the 4 you just add plus medium, then you can hit continue. Everybody with me? So we will ignore the chemical fields, right? And here on the cell properties and behaviors window, we'll select contact internal, focal point plasticity, volume flex, and cell neighbors. And this will be an example where it's absolutely necessary that you guys have the center of mass selected. Okay. So have you seen the contact internal before? Or will this be the first time? maybe this is the first time. No problems. So did you all select this? Oh, maybe seeing the cell migration model. Okay. We'll be seeing again here. Okay. These slides assume that you have never seen those before. So after you select all this, you can configuration complete. And now we have all the basic elements of simulation, and it's your turn now. Now the first task I'll ask you is go back to the initializer plugin, delete what you have there, and which should be around line 255. And I want you to create 4 lines, 4 rows. The first row of only made of apical cells, the second row only made of lateral cells, the third row only made of basal cells, and a last row made of big ECM cells. Okay. So to do that, you have to, in the uniform initializer, you have this region inside. You have to copy and paste this region 4 times to create each one of those rows. Okay. And just remembering the origin of coordinates in CompuCell simulations is on the bottom left. So this is the 0, and then you go up here. Okay. So, and here you already see the code I'm using to draw the apical line. Okay, line of cells. When you draw the ECM, it does not need to be those big cells, but it can be also a bunch of cells, which is 7 by 7. So that's your first task. Please go on the code on the uniform initializer plugin and try to create this initial condition. Okay. Okay. That's not enough. We're still far away to get a stable epithelium, but this will be the first step. Yes, yes. I'll walk you through. I want you to try a little bit first. Okay. So you basically have to repeat that 4 times, one for the apical, the other for the lateral, that for the basal, right? And your cells are 7 by 7, so you have to do that in steps of 7. So for this one, I made the minimum and max boxes reached. Well, the X goes from 0 to 350, which is the size of our simulation, and then the Y I made from 0 to 35, and I make the width of the ECM cell to be 35, and then from 35 to 42, 42 to 49, and 49 to 56. So this is one possible solution to actually accomplish this. Okay, so here in my initializer, I define these 4 regions, one for each row. Okay, and the first one is it just goes as in this order: apical, lateral, basal, and ECM. and these are the coordinates that I use to draw it. Okay. So when after you do this, maybe you have already done by yourselves, you can play the simulation to check if you get the correct initial condition. If you just hit play, it's gonna play the simulation. It's bad if you just hit the step function, so you can see the very first frame, right? The 0 Monte Carlo step to see how it looks like, and just to check if you got it as you want it. So is this making sense? Was this the first time that you use multiple regions in the initializers? No, okay, you have already used that before. Okay. All right, so if you have already all have done this, I'll proceed. Okay, so this is the solution. So that was your first task. But if you just play the simulation, you see that things look very ugly, right? So you can try to play the simulation as it is right now, and you should get something that looks like this, right? So what's happening here? Can anyone now tell me? So this is definitely not an stable epithelium, right? What's happening is that, well, the simulations just use the default parameters, and use the default parameters. We did select FPP plugins, so it's just making connections with FPP links through any cell that it sees. Okay, those ECM cells, which I set up them to be very big at the beginning, they still have the target area that CompuCell3D assigns as a default, which is 50. So the first thing they do, they shrink, and those cells do not know who they are. They have all the same contact energies, so they will just randomly mix with one another. Okay, so what we need to do now is, okay, the initial condition is right, but everything that goes after is wrong. So we have to fix all those parameters, right? So first, let's adjust the links, okay? So as I said, by default, the CompuCell3D is going to create a template code with all the possible pair type combinations, right? So here we have how many cell types? We have 4, right? So you can do the calculation how many cell pairs it is, and they are all trying to do something. They are all active. Okay, so in this model we only care about the connections between neighboring apical sites. Okay, so the first thing you have to do is if we only interested in the links between apical sites between adjacent cells, we can leave that on and turn off all the other ones. So you can either delete or comment all the other FPP links that you see under the focal point plasticity plugin in the XML code, or you can just set the maximum number of junctions to be 0. Okay. And as you have seen before, CompuCell also creates a template for internal contacts between cells that belong to the same cell. We will not be using that today, so we have to also turn all those off, okay, including the internal parameters for between apical and apical sites. Okay, so the apical we're gonna leave that on, and usually each apical site cell see 2 neighboring apical sites, one to the left, one to the right. So we want to have our maximum number of junctions to be 2. And but that's between cells of different types, not between cells of the same type. Okay, each cell has only one apical compartment, so we can ignore the apical pair for internal compartments. Paulo Burke has a very good question. Would not work the lateral sides? Why? So if the cells are connected by the lateral sides, shouldn't that be turned on in our model? Okay, so that's a good question, but the region of the cell which actually has the additional junctional belt, yes, is technically on the lateral side of the cells, but is very close to the apical side. So this is one case that our terms do not completely match the biology, because the lateral junction is very close to the apical side of the cell, right? Is right there. Is better, our model be more realistic to have those links from the apical compartments in our model than from the lateral compartments for our model. Does this make sense? So this is a case where our terms don't completely match the biology, but we have to make it, we want the apical side of the cells to the side junction of the cell to be between the apical side, because that is the it will be more closely, it better matched what's seen in vivo, and it will make a big difference. Would it hurt leaving both? You can leave both, and you can tell us what happens. Okay, that's okay. Because usually the very strong complex junctions, the ones that actually transmit the force between the cells to one another, the form is happening right at the top. But it's true that you also have other types of junctions all the way to the lateral membrane of the cell. You have the desmosomes, you have other kind of junctions. So it's justifiable to have links between those lateral compartments as well. Okay. So turn off all the links except apical, and if you want, you can also leave the lateral as well. And so that's one fix, our simulation. And the other fix that we need is, if you're using very big ECM cells, you have to adjust their target volume. So it should be 35 by 35. So the target volume should be 1225 pixels. Okay. One thing that may help your simulation, you may want to set the lambda volume of the ECM cells to be very low. Instead of 2, you can make 0.2. That will make your simulation look a little bit better than what I'm showing here. Okay. All right. So is everybody on board here? If you're all fixed, you should get something that looks like this. Okay. So yes, did you got it? Did you guys got something that looks like this? Now the ECM cells do not shrink, and all your apical cells, they're not compartments yet, are linked with FPP links.
Excuse me, can you show the previous slide, because
yes.
Okay,
yes.
It shrinks for me, the ECM.
Okay, so you have to go to the volume plugin and fix the target volume. And of course, you have to make sure that you save the simulation before you go to the player. Can I go back?
Yeah, sure.
Okay, great. Okay, are your initial ECM cells very big, or they are 7 by 7 initially? If they are 7 by 7 initially, you leave the target volume at 50. Maybe that's what happened. So, do you need help? If you need help, someone can assist you in the breakout room. Formatting error. Okay, so maybe something was not set up right in the plugin tags. That's something like this. Okay. So if anyone could help Ajay in the breakout room and tag him, I would appreciate. Okay, Juliano can help you both. Thanks, Juliano. Okay, so I hope you all got something that the rest of you got something that looks like this. Okay, but as you play the simulation, you see that those cells start to move around. Okay, you can even tell which ones started together. They're just moving around, and they do not know that they belong to the same cell, basically. Okay, so the next thing that we need to do is go to the Python code and instruct those cells that, okay, you three one that are on top of each other, you actually belong to the same cell. Okay, so you have to do that on the Python level. Okay, so you basically have to find all the cells that are stuck together, okay, and tell them you belong to the same cell. So usually each one of those cells that you create, they have an ID, but they also have something that's called a cluster ID. Okay, so if you want to group a bunch of cells together to belong to the same cell, you have to all assign them to the same cluster ID. And there is a function to do this in CompuCell. There is self.reassign underscore cluster underscore ID, and then the name of the cell that you're reassigning and the new cluster ID that it has. So in this case, cell 2 is getting the cluster ID of cell. Okay, so I'm reading the cluster ID of my original cell and assigning it to the cell 2.
Excuse me, what is cell 2?
Cell 2, okay, that's code specific. Okay, is the name of your cell. It can be the neighbor, it can be another loop over other cell type. So, but before you get there, we have to first identify all the cells that are on top of each other, right? So how you identify all the cells that are on top of each other? Does anyone has an idea? Is there any suggestion? You can look through a type and look for neighbors or the other type. Very good, Paulo. The other complemented, same, XCOM, excellent, get COM. With the angle like last module, that can be done. But maybe look at the center of mass a little bit easier, right? So that's actually what I suggest. Do looper over one cell type. Inside that loop, add another loop for the other cell type, and check if both x coordinate center of mass are equal. Okay. If you'll be doing that on the start function, then the cells have not moved yet, so they should match the center of mass if they're on top of each other. And then if they are, you just use the function that you have at the bottom there. Okay, so try to do that and see if you can, if it works. Okay, that's true what Pedro said checking the neighbors, close neighbor of certain type would work for any rotation. Yes. So if your epithelium was like this, right, that would be a very general way to do it. But here it's oriented in the x direction, so it's easier. Okay, so let me show this is one possible solution. Okay, so I'm just implementing what I did. I chose to start with the apical side. I'll go over the list of all the cells that are of type apical side. And inside I do 2 loops actually. The first loop for all the cells that are of the lateral side. I compare their center of masses, and if they are exactly equal, I link those 2 cells. Well, that's a good question, Arhan, because the cells will wiggle. But this has been done in the start function, okay? And in the start function, the cells have not, no pixel competence have happened. So you can count on the center of mass to be exactly equal. Okay, but yeah, but if it was something a little bit different, then yes, you have to check there within a interval. That's true. Okay. So I'm doing a loop for the first cell type, and then I'm doing a loop for the another cell type. If you just pull up those loops from the CompuCell menu, you're gonna give you for cell and for cell. So you have to make sure that the second cell type does not get, the second loop does not get mixed with the first cell loop. So that's why I call this one cell and this other one cell 2. Okay, so they have to be different. Okay, and then I check cell 1 if equal to cell 2, the x center mass. Once I found that, I can stop the loop because all the other cells will be different. Okay, so there's no, so this break here takes you out for the innermost for loop that you are in. So it's gonna take you out from this loop, but you keep you still in this outer loop. Okay, and then I link the apical with the basal, with the lateral, but I still have to link the apical with the basal side. Okay, so I do another loop here with looking for all the basal cells and do the same code again. And when I do this, this is what I have. For this simulation, I only have instruct the player to draw the cluster borders and not the cell borders. So here you can see that the cells are actually made, all those cells here belong to the same cluster. And this one belongs to the same cluster and this one belongs to the same cluster because I have a different kind of boundary drawing to show them.
So in the reassign cluster ID, the second parameter, I mean the cell cluster ID is copied to the first parameter.
Exactly.
Okay.
Exactly. Yes. All right. Okay, so now if you do your simulation, they should look a little bit better at the beginning, but everything that you have seen on the first one and the one before, it will still happen. The cells now know that they belong to the same cell. Those compartments know that they belong to which compartments belong to the same cell, which parts is from the other cells. But we didn't, besides this, we didn't change any of the parameters of the energy or the links or so far. So the cells will still mix over time because they don't have any strong attachment to the cells of the other type. Okay, so what you have to do now, you have to adjust their contact interactions in order to prevent this mixing. Okay, and that's the key step to make sure that your epithelium will be stable. Does it make sense? And now it comes to the part that may sounds very complicated. So because now we have cells that may be of the same type but belong to, so we now have this also cell types, apical, lateral, basal, right? But at each point in time, one of these compartments are seen compartments that belong to the same cluster and compartments that belong to a different cluster, okay? So they should have different contact energies between them, okay? So this means that when you look at our contact energy table, we actually have two contact energy tables. One for all the pairs of cell types that belong to different clusters, and another for all the cell pair types that belong to the same cluster. Okay, so you have all these different numbers to specify. Does this make sense? So it's one thing this interaction of this green with this red, and a different thing the interaction of this green with this red or the apical side that belongs to the same cell. Okay, so all your contact energies suddenly got double, and you need to know which values to put there. Does it make sense? So, in other words, okay, now we are dealing with a lot of parameters, okay? And that seems to be, we're gonna spend the rest next 5 days trying to fix this, but we can reason out what those values should be, okay? So let's go now step by step and think about it.
Can you say again, what's the problem here with the energies?
Well, the problem is here that now you have a lot of parameters, basically. If you just run simulation as it is, you know that all those numbers here are equal to the same value, which is 10, and that means that the cells are just mixing. So now we have to correct those numbers to actually have different contact energies between cells of different types and between cells that belong to, and you have to differentiate those contacts of compartments that belong to the same cluster and contacts that belong to different clusters. So that's why now we have two tables of contact energies. Okay, so let's now go step by step with what those energies should be. So we can start with the internal compartments, right? So one thing that we know for sure is that all those cells, all those compartments that belong to the same cell, they should be more closely in contact with one another, right? They should prefer to be, you want to ensure that our cell cluster that's made of different compartments, it's well connected together. So we should prefer to be in contact with the compartments of the same type, of the same cluster, than with contact with other clusters. So one thing that you know for sure is that probably when you choose your contact energies, you want those values to be low. So to ensure that the cells remains a single thing, okay? So you probably want all those values here to be low. Does it make sense? Okay. Yes, okay. Lucas brought a very good point. All those cells here, they should only have one copy of each compartment, right? So we do not care about all those values in the diagonal. Okay, so that already simplifies our life, because this one here, and this one here, they will never be used. So you can just set it to 0 or don't bother changing it. Okay, so you can set all those values to 0, just like you never had two mediums in the same simulation. So that first value of your contacts, either external or internal, which is medium, you can just ignore. Okay, so now we solve. Okay, so we know that this has to be low because we want to ensure that they like each other, and those here in the diagonal, we don't care. Okay, so we already solve one, and then Yeah, if you have a value there, but it's not being used, it's no problem. The other thing is that there are no cells with a medium compartment or cells with an ECM compartment, right? The ECM is a cell by itself that is not part of any cluster. So one thing that you can do, you can basically ignore all the values that are in the contact internal that relates to the ECM. So that, together with what Lucas said, makes our life already very easy. So in the contact internal table, we only care about three values: lateral apical, basal apical, and basal lateral. These are all the three values that we have to specify because, and we don't, you know that they should be low, okay? You know that they should be low. Does it make sense? So here you have to be using all your what you learned on the first day, okay? Okay, so okay, we solved the internal content of energy values, right? Or we know more or less how they should look like. They should be a low value there. Now you have to leave the other energy elements there. I think that if you just delete those entries, you get an error, okay? You just set all of them to 0 or just ignore them. Okay, so remember this black this matrix is symmetric, so that's why I blacked out all the off diagonal elements, okay, in the bottom. Okay, now let's go to the other table of contact energies, and now this one's going to be a little bit more complicated than you have before. So let's start by, so what's the first thing you can reason out? What about the contact? Let's talk about the diagonal. Okay, the diagonal, which sets the addition between apical, lateral, and basal. Okay, there's a question: why low energy between apical basal? Okay, I was trying to keep the story simple here and say that because they belong to the same cell, you want them all to be low. But yes, you want the apical to be one side and the basal to be on the opposite side with the lateral in between. So one thing that you can do is can make the energy between apical and basal to be a little bit higher to ensure that this happens. Yes, you can do that. It's a good question. Yes. Especially when you have situations where your epithelial cells are moving a lot for some reason, or they are going into and out of the epithelium because they're that's what the cells do when they divide. So the cells in the epithelium, when they divide, they lose their connections. They all go to the basal side, round up, divide, and then go back in. Okay. So when you have the situation, you probably want to make sure that the apical and the basal sides will sort out as the cell elongates. Okay, yeah. But here the cells are not moving; they're just kind of relative to one another. So it's okay to just leave the apical base to be as low as the others. Okay. Okay, but let's go back to now the cross cluster to cluster interactions. So let's see what should be the energy between apical and apical, lateral and lateral, and basal and basal. Okay, should they be high or low? Or another question: do they like to be with one another? Okay, does everybody agrees with Paulo, or do you have something to add? Okay, so should they be low, as low Ah, very good, Lucas. Yes, it has to be low, but slightly higher than the internal, right? You don't want it, otherwise you may start having mixing, right? Otherwise those two can swipe to other. So we want them to be low, but we want them to be low but a little bit higher than what you had before. Okay. If they're low, if they're high and everything else is low, they'll be detached. Yes. So everything here is irrespective, is relative to what the other energies have been chosen, right? So, but let's go by side by side. Okay. So the cells like to align their apical with the lateral and the basal. So you know that they should like this. Okay. So that also should be low, but maybe a little bit higher than what you had for the internal. So I'll put these things here as low plus, okay? Something like this. Does it make sense? I mean, what I'm basically saying is that, okay, those cells like to align themselves apical, basal, right? But they should not like that so much that they would want the compartment to enter the inside the other cell. So that's why they have to be low but higher than what you have internally. But the basic answer to your question is yes, they will move apart. Okay, so what else we can say? What about the adhesion between the apical and the lateral, or these cross terms here? And basal and lateral, should they be high or low? So would the cells like to match the basal with the lateral, or the apical to the other one? Or what should be the adhesion energy between apical and basal? Should they like to be close to one another, or should they hate each other? Okay, should be higher. Okay, so yes, all these crossings, they should be high energies. So they do not like to have, so this kind of contact here that you see the red from this side entering contact with the lateral of this side should be penalized. So this may still happen through the fluctuations, but you quickly go back to what you have here on the right, which is just apical and basal. Okay. So when you have this, so all this now is ensuring this kind of shape. Does it make sense to everyone so far? What else we can tell? Okay. So what about the apical interaction with the medium and the ECM? Okay. Now we will get to XML later. Let's first figure out qualitatively what's going on. Okay. So we have to reason out what should be. Yeah. Of course, we could go to the XML code directly and just try numbers randomly, but it's better if you think about what should be what's the behavior should be, and based on what the behavior should be, what should be the relative values of those things, and then we go to the code and try to implement that. So let's talk about now the apical side. So the apical side is mostly done, but you still have the apical with the medium and the apical with the CM. One of them should be this high and the other one should be low. Which ones? Okay.
Apical with the medium should be the same as the basal with the ECM.
Yes, very good. By the same logic. And that
they would be
like
lower.
that, or should they hate that?
they should love them, like
Yes.
lower and
Love
apical.
them. Yes.
But the vice versa, like the apical with the ECM or the basal with the medium would be much higher.
Yes. Yes. So other people already went to gave the same answer. So apical should be low here, and apical ECM should be very high. Okay, to make sure that the cell never flips. Okay, you probably don't want to be as low as the internal one, maybe just as low as the apical to apical. How about that? Okay, and now the same thing for the basal side. We have the same logic but inverted. Okay, basal with medium should be high. They should never like to be here, but they should like to be here. Okay, like this. Okay, and now we have those other 4 missing. What they should be. So, well, ECM and medium should be super high. They should never touch each other, right? We want to ensure that you always have cells in between. Okay. ECM does not make much sense, right? The ECM, we subdivide the same into different units, but they should be the same tissue. So the ECM itself should be 0. Okay, so this value here should be 0. And the lateral side with the medium and the lateral side with the ECM, maybe should assign the same value as with the basal and apical side of the neighboring cells, something that's also high. So like this and this. And with this, we set up qualitatively what those relationships should be. Okay? So is this all making sense? Or do you disagree with one of those parameters? So I tried to reason out what all those hierarchies should be. Okay. Now that we reason out all those hierarchies should be, and I hope you kind of agree. You said basal ECM low, but they are merging. Okay, now it goes to the other question, right? Now that you have this qualitative table of things, let's try to put some numbers that reflect that and see what happens. Okay. So you can do increase of 5. Okay. Well, if you just if the difference between additions energies that you put there is just like they're all 10 at the beginning, and just make 11, 9, 8. That's not enough of a difference to see an effect. Yeah, the difference has to be a little bit higher. Okay. So, well, in the interest of time, I will just show the values that I used. Okay. So the content internal I just set to 2, a very low value, so they will like to be close to one another. This low plus that we defined before, I just set it to 5, so it's still low, but as before. The high values or the higher values that we set, we decide to just leave it, left it to be 10, and those very high values I just left it to be 20. And I can guarantee you that if you make those choices of numbers, you'll get something that looks like in my picture. Okay. So,
sorry, about the internal other parameter that you put dash. So we are going to delete them, or what should we do with them? Or
I think I'm pretty sure that if you delete them, you get an error. You just leave them wherever they are. They're never used, so that's why you put a dash, because they're never used. So You can either set all of those to 0 or leave them to the default value, which is 10.
The default value is 10, so it may have an effect on the simulation.
You can leave there because they're never used. Those pairs never happen, right? So people are saying that the basal ECM at 5 is not working. Okay. Maybe you can make the well, I instruct you to set the ECM to be 0, right? If it's not working, maybe you can increase a little bit. In principle, should work for. It's fine now. Okay, good. Okay, so is this working for everyone? If yes, you just said yes in the chat. Or maybe do you need a little bit more time to type in those values? Okay, some people are saying that the basal layer has been ruptured. Okay, that did not happen in my simulation. Double check if you have the correct values. Maybe this is not a good solution, but so do you have a basal that just starts float around? Make sure that the contact internal between basal and lateral is set to 2, because this value here should prevent the basals from detaching. Okay, if for everyone the base is starting to mix with ECM, maybe you can increase this contact here from 5 to 10. Maybe that will fix. Yeah, as James says, you can also try different energies. In principle, all that hierarchy that we have here should be enough. You can improve on that to make it better. You can make arguments that, well, all those low plus should be a little bit different from one another, but that's the simplest things I can present to you, propose to you guys. Okay. So now we have hopefully a stable epithelium. If your basal ECM is wandering around, make sure that you have increase its value with the ECM. Yes, ECM has to be 0. Maybe that's what's missing. Fixed. Okay, excellent. And now let's actually add some active movement to this thing. Let's start to follow the tissue. So you can see that only make this thing was already a lot of work, right? Even though it was doing nothing. But now we have a stable epithelium. And in order to get a stable epithelium in CompuCell, we have to really think through, okay, what those energies should be, what they should be the contact energies. And so it takes some time. You have to know what's going on, right? You have to understand what the contact energies work. So that's what I try to emphasize on the first day of this CompuCell workshop. Okay, so now we have our stable epithelium with a nicely connected apical side by side on the top. What you want to do now is actually make that fold. Okay. So are you all ready to do that? If you are, then let's let's do that. And to change those links properties has to be done inside the Python code. But before you go to the Python code, you have to remember tell the plugin that you'll be changing then from the Python code. So in other words, you have to go under focal point plasticity and uncomment the local tag. So the change you'll be doing from the Python level will be accepted by the CompuCell3D player. Okay. Good question, Jay. To make the cells, okay, what if you want to control of how tall the cells are or short the cells are? Okay, there is a way to do this. You can actually use those links to make the cells shorter or bigger. Okay, you can go back to the focal point plasticity settings in the XML code, and you can draw links between the apical side and the lateral side, the basal side, and also add a link between the basal and the apical side, but within the same cell. Okay, so remember all those FP links that will have internal parameters, they all refer to the compartments that belong to the same cell. So you can add a set of links between those compartments and change the target distance of those links to make your cell either longer or shorter. That will do that artificially, but how about as a consequence of cell adhesion? It probably can work. I never tried that because the other way gives us more control, but that's something that you can try. Definitely. Maybe decrease the adhesion between green and green would help that. You can also change the target volume of those compartments, right? You can also do that. Right here, you're making the apical side 7x7, the lateral side compartment 7x7, and the basal side 7x7, but the lateral side probably should be bigger and the other ones smaller, right? Well, just increase the volume will also do the trick. Okay. So there are probably many ways to do it. Okay. So very good questions. Okay, so we turn on the local tag to allow changes in the Python level of coding. And what you want to do now, you want to do this not in the start function, but in the step function. You want that some specific, you want to create epithelium and let it be there for some time, and a specific time tell all the cells at the center to apical constrict. Okay. Let's say that, so this is something that you want to do at the step function, right? So what I want you guys to try by yourselves is at specific time, let's say at Monte Carlo step equals 500, find a group of apical compartments inside the epithelium, right? You can choose where it's going to be and how wide it's going to be. Okay, find all of them. And for all of them, you delete all the links that they have and create new links with their apical neighbors. Okay, and for that you'll be using those two drop down menu codes, the delete FPP links, right? So you find the cell that you want to change, delete the FP links, and then After delete, you go over the neighbors, find the neighbors, and make a new link with those neighbors. If someone could help Cicely to, I will appreciate. So as James says, the reason that the cell elongates or is partially because of the microtubule structure inside the cell become polarized. And so these are long pipes inside the cell basically that they're very rigid and they are centrally organized in the centrosome, very close to the nucleus of the cell, and then they, in an elongated cell, all those microtubules, they make some kind of hourglass shape, right? And there's some that goes to the apical side, some that go to the basal side, and they help keep the cell elongated. So in some way, that's using links as a surrogate for the elongating or as a surrogate for the underlying microtubule structure is very justifiable. Okay, so try to make your code for yourselves. I will give you 1 minute and then I will show my solution. In many cases that will be true. The microtubules drive the elongation of the cell. In other cases, that's not true at all. Sometimes the cells become elongated because of external factors, and the microtubules are still aligned inside the cells, but they just align as a result of that elongation. So that's happening from the epithelial cell. Well, in Drosophila dorsal closure, you have a group of cells that contract and drive all the epithelial side to elongate. And if you look at the epithelial side, they're very elongated, and the microtubules are aligned in the direction of elongation, but that happens as a consequence, not as a driver. So, well, the thing is that, well, in biology, you always can have find exceptions if you look at half those things. Someone propose a model or a mechanism, and then people find many other instances that that's not the case. But usually we can make the argument, yes, the microtubules are helping keep the cells elongated. Okay, so did anyone got this working already? Yes, no? Let me try the chat. So are you done with the exercise? Okay, the lambda distance value. So that is how strong it is. You can just keep at 10. Oh, that's a good question. Try 10. If nothing works, keep setting higher. You can start trying with 20 or 30. Okay. The target distance has to be something that's lower than 7 for in order to drive contraction. A change. You can just set the target distance to 0. If anyone got the tissue to fold or get some indentation, please paste that on the Slack. How to justify this plugin biologically? Okay, so biologically, when the cells undergo invagination or this kind of folding, is because 90% of the time is because there is an actin cytoskeleton, which is a set of filaments, okay, that are present at the apical side of the cell that becomes activated, and they start to contract the apical cytoskeleton. Okay, the axon cell contract. But this apical cytoskeleton on the top of the cells is connected to the tight junctions around it. So when they contract, they pull the neighbors. So what's happening is that the cell, the top of the cell, the apical cytoskeleton start to constrict, and as it constricts, it's pull its cell neighbors. And when many cells are doing this, then this causes the whole, each one of those cells to become a wedge shape, because you constrict here. And when you have many of those cells in the same region trying to get this wedge shape, you end up driving the curvature of the tissue. So that's what's happening biologically. In the model, you cannot directly model the actomyosin cytoskeleton in the apical sites, but you can model its downstream effect, which is pulling the other cells together. Okay, so when the cell apical sig is pulling the other ones towards it, right, exerting forces. So you are just modeling the net downstream effect. Okay, we are pulling the other cells, and then when you make this, you drive tissue folding. Yeah, so after you delete the cells, you have to do a loop over the cell neighbors, find the neighbor that's an apical compartment as well, and then do the link. Okay. So let me show my code. Okay, so again, this is one way to implement it. Your code may look very different than what I have here. So yes, you have also checked if the neighbor is not medium. Good. So let me walk you through my code. So I check what is the Monte Carlo step. If it's equal 500, I do a loop over all the apical cells, right? These are the ones that have the links. And then if the cells are between the center of mass, the x direction center of mass is between this range, and you can make this bigger or smaller, right? You first remove all the FP links of the cells. And once you remove the alt FP links of the cells, you have to reestablish those links. So I do a loop over my neighbors. Okay, that's this familiar one. And if this is a neighbor and not a medium, and if this neighbor is of type self-apical, then I create a new link. And here I define the lambda distance to be 40, for example, the target distance to be 4, so it has to be smaller than 7, otherwise things will look funny. And I kept the maximum distance to be 20 pixels as I had before, and I used those 3 numbers here as arguments of this function, and then the first entry here is the cell, the second entry is the neighbor. Okay, and I promise that if you do this code here, you get something that looks like this. You get some kind of indentation here and the cells kind of moving inside. Okay. Yes, yes, Ajay. Yes, I think that would be a way to accomplish it. This. I'm not sure I understand your question. Membrane is tight domino structure. Yeah, of course.
Anything that happens in a domino line can affect all the next ones. But this is so dense that anything that happens anywhere should have some effect on the whole line, no?
Yes, that's true. That's true.
Just an observation. I know it's out of the programming here.
No, it's true. We do have a closed system here, right?
Yes.
I made those
Too close. It's very tight.
Yeah, I made those cells here to be seeing those cells here, right?
Yeah.
Yeah, but all those cells feel this thing happening here. Yeah.
Right.
It takes some time for them to feel that, right? As I said, to pull, but you can see in simulation that it takes some time for the cells to actually move, but they will be sensing that thing.
Also, the edge effects are, there are some kind of walls here, no, on the right and left?
No, because we set periodic boundary conditions, right?
Oh,
So
okay.
it continues on the other side.
Okay,
But
fine.
still, all those cells are things. So that's why I tried to make this very big in the horizontal direction to kind of reduce a little bit. Because if the simulation was just centered between 100 and 250, right? Those cells that are not constrict, they will have to stretch a lot, right? And there is always this tug of war. Okay, for some cells to constrict, the other cells have to give way a little bit. So if you go back to the XML code and make those all those cells to be very high lambda volume, lambda strength, right? To begin with, those cells that said they would not be able to constrict. We have to even increase more their lambda. How can you do a full invagination? Okay, that's a good question. First, this model is a little bit simplified, right? One thing that you probably want to do, you want to make the apical compartment a little bit smaller on those cells that are undergoing constriction, right? Otherwise, you have some volume exclusion mechanism there getting in the way. Okay. Some people, at least for Drosophila gastrulation, some people said that there should be tensions, transverse tensions happening on the cells for this to happen. So the cells either have, not forgot the way it goes. You probably have to try to force those cells to shorten a little bit, and that will help it drive it even down further. Okay. You also have some kind of volume exclusion because of the ECM below it, right? So you can try to make the lambda volume of the ECM cells that you're modeling below it to be very low. So if you make from that, right now it's 2, you make it 0.2, so they are very compressible. That should help a little bit the tissue to go inside more. You can also make those initial cell links from the other side, they're not seem to be a little bit weaker, so they can stretch more. That should also facilitate the process. Okay, what else you can do? This is a good question. How you can get a full vagination, right? So some people say that there must be things happening on the transversal side. Okay. So you can add those links from the other internal links to make the cells longer or shorter. Only the cells that are undergoing vagination, see if it helps. So think I stopped screen share, but that's basically everything that I had for this module. Are there more questions? So how to get full imagination with this very simple model is it's a very good question. I just suggest a bunch of things you can, guys. You are free to try yourselves. What else you can you can do. Okay. So So I know that there was some interest in those PCP kind of simulations. Maybe I can set a time tomorrow to show you guys. I didn't really prepare a lecture about that, but I can show a code that I have and walk you through what's going on. But it's basically using all those concepts that you have seen before here. You're using compartment cells and you are making educated guesses of how the energies should be in order to drive kind of compartments of different size to be in touch with others. Okay, so if there's interest, I can set a parallel section tomorrow to just go over the code and tell you what's going on. I know that says something like this for other topics, and we can set up a time tomorrow. Okay. Yes, I said I will be make an exercise about cells using the anchors to drive the cells and make another anchor and drive the cells. That would be the idea. It's just that, well, I didn't get into doing that, and we already running out of time anyway. Okay. So, but in principle, tomorrow there's no lectures. Is the hackathon, right? So you're gonna put all this in practice for those different projects. And I heard all your projects presentations on yesterday or Thursday, and they all sound very exciting. But there's still one more module to go that Sluka is gonna show how you do other things with CompuCell. I don't want to take too much of his time, and I will stop here and let him take over, or maybe I think probably need a break in between. Okay.
Hey, Julio, can you see my screen?
Yes.
You've got a typo on the number of the,
So I
of the
had,
module. Jim, you've got
Nope.
a typo in the number
Nope,
of
it's
the
not.
module.
No, I don't. That's the correct number.
Oh, you want it to be 3, okay, it's 3/4 rather than, okay, got it.
The file name is 534, so I'm not
I
going to
get
change
it. No worries.
it. I had absolutely no luck moving any of these files onto NanoHub. If you're writing files, you can't transfer the CompuCell jobs from your local machine to NanoHub and have them work. We do not know what the problem is, but NanoHub has just totally hoses up all the paths. So none of these demos actually work on NanoHub. Which is unfortunate. Did we ever ask him, people how many have CompuCell running locally? Okay, I assume everybody is back. You got a stretch and some more coffee. Everybody needs more coffee. We're running a bit behind. It's 4:30 and we're scheduled to go to 5:30 or so. So I think this is going to have to be a bit quicker than it was supposed to have been. We're going to end up covering a lot of ground, I think. A lot of the stuff that we cover is not going to transfer terribly well to NanoHub. NanoHub does not like file IO because of protection problems, probably. But in any case, we'll see how far we can get. Remember, this is being recorded and may show up on the internet somewhere someday. So let's see, where should we start? Hopefully you can see my screen here of my slides. Here's a brief outline. We start off with writing data to files, and if you can write the data to files, it's pretty easy to read it back. We'll talk a little bit about running parameter scans and running replicates, a little bit about sensitivity analysis, and then try to finish with parameter files and some best practices ideas. There's a zip of zip in the today's folder online. I will put this link into chat if you want to download this zip. So that's a zip of zip. So it's got this PowerPoint in it. It also has these 5 files. These are zip files of CompuCell projects. If you want to follow along or check back and kind of catch up, this middle set here is meant to be sequential, so that if you get behind, you can just load up the next one in the series and get caught up. Okay, so we're going to start with writing data to files. You might have seen a little bit of this earlier this week. Twedit does have some tools to help you do it. You can also use standard Python code. There should be a C there. It's like any other Python program. You can open and close files, control where they're at. Usually you'll want to open a file in your start function, write some stuff into the file in your step function, and then close the file and finish. And the demo and the working set is writing two files, excuse me. So as I said, there's some helpers in Twedit, and I will show you some of them. For instance, so here's my little demo of just writing the files. It's pretty much straightforward. It's just a cell sort. So normally you would open a file in your start function, do some calculations, and then write some stuff in your step. In this case, we're only going to write it once, just at step 50. And then just to be a nice citizen, close all your files with the finish. Also make sure you don't lose any data. So as using the Twedit shortcuts in CC3D Python, there's Python utilities, and you see these are almost all related to dealing with files. Get the steppable class name and so forth. In general, or one common thing to do is open a file in the output directory, which on NanoHub is a little weird, exactly where the output directory is, but on most desktop systems, Linux, Mac, and Windows, that would be in your standard CC3D workspace or whatever folder you specify in your player settings. We'll talk about some of these other ones in a minute, but if you go ahead and click on Open File in Simulation Output Directory, you get this little insert, which is pretty straightforward. The only 2 things you really need to change is you have to give it a file name. It should be quoted. And you have to decide if the file should be read, write, or append. So W for write, A for append, and R for read. This returns 2 variables. The actual file object, which is what you generally refer to, and also, just to be nice, it returns the file path. But in the future, you'll refer to this particular file as this file object. If you're going to be using this file object in any other class, for instance, open and start, but you want to use it in step, you'll probably need to include the self tag onto it so that it can be used in other places. So I'm going to take out my insert that it did. Just delete that. There we go. And here is my edited version of that little helper script. So my file name, I define up here, logfile.txt, and this is a file being opened for write, and it's self object. So this is writing to your standard output directory. If you want to write to your project directory, it would be the directory where this actual CompuCell code is located. It's a little trickier, not too much trickier, but you need to import the OS and then open up the file using that. Import OS is actually in the Python CC3D Python utilities, import OS. And so that once you do that, you can get the directory of this actual file. So here's the script to do it. So this is where this actual Python steppable file lives, is now in FileDir. FileDir up one just goes up one. You can look at the code later if you ever need these little widgets. So this goes up one directory. So now instead of being in the simulation folder, this is in the project's main folder. And that gives you a path, and then you can append a file name to it and use standard Python open to open that file name for append. So this is a handy way to do it if every time you run your job, you want it to append to the same log file. So just so we have a complete thing, we're going to define our cell target volumes. We're going to start running the simulation at step 50. We're just going to count how many of our two cells we have, A cells and B cells, or condensing and non-condensing cells, usually in cell sort, but here I named them different. And we're going to build a text string to write to the output files. Writing to a file, so you need to write strings and not numbers in general. So we have to convert our numeric values with string. We can get the count of cells just using the same list we use if we're going to visit all cells of a given type. So the len of self cell list by type, self A cell and B cell gives us our cell counts for the two cell types. So we create a string, and then our self object file write, output text, which is that string, and then there's another one, because I opened two files, remember, one's in the normal output directory, and one's actually in our working directory. And that we're just doing that at step 50. And then at finish, we're going to write a little closing message in each one of those two files, and then close them both. So if we run this, where'd my player go? It's going to be a little slow to start up here. The slide deck has basically everything I've said on slide. So import the widget, edit your file name. Don't forget to close the file. How to write to the project directory. Again, how to insert the import OS and edit it so that it's writing to a file where you know where it's at. So here's the sort, and it's already done because it is such a tiny little project. And now if I go to, if I remember that, so this is my CC3D workspace, and by default, CompuCell writes my job name and then a timestamp, and here's my log file. Get it to open. So there's everything I had written to the log file. How many cells I had at that particular step. I think it's always a good idea to write a starting message in a file and write a closing message to the file. That way, if you go back later and look at the file, you can tell the file was closed normally because it's got your closing tag in it. So I know I got to the finish statement that officially closed the file, which means I got to the end of the run. So that is the one that's in the standard output directory. Yeah, see if I can find my other output. Now I'm lost track of what file I'm in. But in the top left, wherever this file was loaded from, actually in Twit. Okay, it's in my CompuCell file. There it goes. So this is the other log file that was written to the top of the file. And you notice in this case, I didn't write a header and closer, which is not a good idea. You should definitely write a header and closer to those files. So this is actually probably the wrong file. But in any case, in the top level directory, there it is. So this is the new one. So this is my regular working directory for the simulation. And here's where it has written my log file. And that one looks correct. So I got my starting message, how many cells were at that particular step, and then my closed. So that's two ways of writing either to the standard output directory or writing back up into your main working directory. So this is my regular old working directory. It was set up so that this one will get appended to if I run my simulation again. It's a very short simulation, so it won't take very long. Now it's done. It says this editor is telling me my file has changed. So there's the result from this new run. The other write would be written into the our standard output for CompuCell directory, which I won't show. So that since I did a new run, I got a new output directory, and it'll have just a single entry file in it as well. So that's the easiest way to read and write. If you're actually going to read a file, it just changes the mode. And you can do a quick Google search on Python reading from a file to figure out what the command is. So that's reading and writing to files. Appending to files, writing them in different places. Reading and writing files is one of the things that tends to not transfer very well from platform to platform. Especially if you have hard-coded paths. Obviously, if it's got my username and my path somewhere, that's going to be a problem to transfer it. The way I've done it here, though, I have nothing hard-coded in the Python about the path. It's retrieving the path from CompuCell. So the file should move from machine to machine without any problem. Okay. So reading and writing the files. Next, we're going to go to parameter scans. So a parameter scan is an attempt to exhaustively walk over one or more parameters in your file and automatically run the simulation as you change those parameters. So with the parameter scan, you explore a range of values in an automated fashion. You want to know what your cell lambda volume should be, and you'd like to try 3 or 4 values, but you don't want to sit there and have to change your script in Twedit and then run it and then change the script again and run it. So parameter scans will let you set it up and say, I want to do 5 runs with these particular sets of values. You have to be careful about parameter scans, though, because it is a combinatorial problem. If you do 3 values for 1 parameter, that's 3 runs. If you do 3 parameters for each of 2 values, that's 6 runs. If you do 3 values for 3 parameters, is 20. Excuse me, 3 of 3 was 9, and the next one up, 3 parameters, 3 values is 27 runs. So real quickly, you can get into a combinatorial explosion where you just way too many things to scan. So I had this example here. If you wanted to scan 10 parameters with 5 values each, It's a 10-digit base 5 number. That's 5 to the 10th. That's a lot of simulations. You're not going to be able to do it. So there's a limit on how many you can do, what's practical, and that depends on how many parameter scans you want and how long it takes to do one simulation. The simulation we're going to be using runs really fast, so we could actually do quite a few. In general, your simulations won't run in the 10 or 20 seconds at this demo that we're going to be using. So we'll move on. One thing about parameter scans is once you put a parameter scan into a project, it's kind of difficult to get it out again. So it's always a good idea to get your project working the way you want it to, and then make a copy of it and use that one to do your parameter scan. Like I said, it's not easy to get rid of. One thing you definitely want to make sure is before you add the parameter scan, if you're going to save any screenshots, you want to make sure that in your preliminary version project, you've got screenshots saved on, you've got your windows open, you've clicked on the camera for any other screenshots you want. Because once you move into the parameter scan, you're not going to have access to those functions because it's going to snap through all the stuff automatically. And you won't have time to click on the camera icon and stuff like that. So we have another model. If you're playing along at home and you want to open up cell sorting base, we'll go get it. Take me a moment to get there. So this is the thing we're going to, this is, again, another cell sorting. This one's written a little different. I'm actually using condensing and non-condensing cells. It's very straightforward. It's very similar to what you've seen several times. If we run it, take a moment to start up. So our window of our cell sorting, we've also got a metric being calculated, the hetero contact area, and it's being plotted here, and we'll talk about that in a minute. So we'll go back over, let me stop that. So like I said, regular old condensing, non-condensing, very straightforward. Now, if you're going to do a parameter scan, it's a lot like model fitting, is that you need some output that you can judge how good the simulation was. In this particular case, what we're trying to do is get the cell sorting to work best. So we need some metric that describes how well cell sorting works. And that's not necessarily, this case, it's not too complicated to come up with a definition of what is a good simulation. In other cases, it's actually kind of tricky to figure out what a single numeric value is that you can assign to the output of your simulation. If you're fitting data, usually that metric would be your residual error. But a lot of times you don't have data to fit, and so you need to come up with some metric. In this case, we're just going to come up with this very simple metric of just counting how much surface area there is between the condensing cells and all other cell types. And that's going to be our metric. And in the run here, that's what's being plotted. And in this sample, it's pretty straightforward. It's this part. We are going to visit all cells of self-condensing, all the condensing cells. So we're going to visit all of the neighbors of that cell. And then if the neighbor is not of the self-condensing, it's a different cell type, we're just going to sum the heterosurface area. There's also a possibility that the neighbor is actually not a cell, but a media instead, and that's what this second catch will catch. So this will sum up non-condensing and media surface. And then we plot it to our plot. At the end of the run, we'll also write this. We're just going to output it to the screen. So that's going to be our metric. Pretty straightforward, but if you're going to do a parameter scan, you definitely need a metric. It's possible you could also write screenshots and just go through all of your outputs and look at all your screenshots and make some judgment call. I think it's usually better to have a numerical value. Okay, so now we have some metric. Now we can actually start thinking about how you run a parameter scan, or what you would parameter scan, what you're trying to optimize in a cell sorting experiment. So this is basically the core of what really makes the cell sort work is your J values. That's what's driving sort. So presumably one of the best things to adjust to get the best cell sort would be the J values. And just kind of by gestalt and practice, I've got a pretty good set of J values there. The cells sort pretty well. You can see a demonstration down here, starting with a random set of random field of cells. It's doing a pretty good job of sorting. It's not perfect, though it's probably because the simulation is not going long enough. But this is probably where you want to do your parameter scanning. So to start a parameter scan, we will go ahead and use this file here that I have open, this demo, but I'm going to fork the project. I'm going to make a copy of it so that my original file stays put. Did I miss it? There we go. Then give your quick name, just so I'm not working on my original file. I'll just get back to it if I need it. So as I said, this is probably where you want to be fiddling if you're trying to get a better cell sort. It's in the J values. And so now we're going to add a parameter scan to it. And there is tools, a bunch of tools. I got ahead of myself, as I say here, to remind myself. But first, before we can add the parameter scan, we need to add that metric into it. So there's another file that actually does that. And basically, what we're going to do is too far back. Add a new steppable, which is the Python CC3D Python option to add a new steppable. And in that steppable, we're going to calculate that metric of contact surface area, heterologous contact surface area. So the new file will have that in it. For the time being, I'm going to kind of skip over that. So that's the cell sorting base, actually the one I started with anyway. But now, so now that we actually have the metric, we can go back and say, okay, now we can really add the parameter scan. So fork the scan. I just make a copy of your folder so that you're not editing your original, because like I said, it's hard to take a scan back out. It can be done, but it's a lot of work. And now we're finally ready to actually add our scan. So here's our project we're going to add a scan to. These are our values we probably want to be manipulating. So you come over here to the file project list and do a right click and say add parameter scan. And now we get a new entry here, parameter scan, or I can open it up and it's a JSON file. And to start with, it's basically just has a header in it. It doesn't actually say anything of interest. But that's where our scan information is going to go. We go back over here. And now we need to decide what we're going to scan. I think medium to condensing J value. Let's explore that one a little bit. What you do is you highlight the numerical value and then right click and pick add to scan.
There was a question in the chat about how do you open the parameter JSON file.
Okay, so once you've done add parameter scan, it adds this new entry into your file list. And by default, that one doesn't automatically open. So you'll need to click next to it, which it'll show you the file. Double click the file and that'll open it up here. So see, now I have 4 files, my base XML, my registration file, my Python steppable, and my JSON spec. Okay, so now we're going to add this value to it. Select the whole value, right click, add to scan, and we get a dialog. First thing it wants is a name, something that we can call this thing that we're changing. it's nice to be a name that's relevant. This is the J value from medium to condensing. So we can just call it JMC. So J value, medium to condensing. Once a start value, our value was 10. We don't want to do too many values here. So let's do, so you might do something like 8 as a low and 12 as a high. So we're going to scan from 8 to 12. We're going to do as a number of steps, just 3. There's an option for whether it's a linear or a log or a random number. We're going to do linear. There's an option for whether it's a float or an int or a string. In this case, we're going to use a float. And then you click on generate and it will show you the list. And if you get it wrong, 4 steps, that's not what I don't want these fractional numbers. So you can change that back and forth until you get it. What I want to do is 8, 10, and 12. 10 is my starting point, and I want to do 2 below and 2 above. Hit OK. It warns you that something has been changed, and just say yes. And now I'm looking at my JSON file, and it's added in this new chunk. I'm changing a parameter called JMC. The original value was 10. It's going to do 8, 10, and 12. Usually you want to scan more than one thing. So let's also do condensing. Same thing, highlight the value, add to scan. This one's going to be JCC, J value for condensing to condensing. My value started off at 5, so maybe we'll do 3, 7, with 3 steps, linear, float, floating point number, generate 3, 5, 7. So we started off at 5, we're going to go 2 above and 2 below. Click OK. Another warning about the JSON getting changed. That's OK. And now it's added in my new value into this JSON list. This list is editable. You have to be careful that you don't corrupt it. But if you ever decide, you know what, I really don't want that line, you can delete that line, or you could add more lines. The only thing that's tricky about this particular number list is it can't end in a comma. Your last value can't have a comma on it. Okay, so that's how you add values in your XML. What if you wanted to add something in your Python? What if you wanted to say something like, well, I don't actually know how much lambda volume is going to affect the sort. So let's do a parameter scan of that as well. Again, you want to highlight the numerical value, not the name of the value, but the numerical value. Right click, add scan. And this is lambda volume, call it lambv. Starting off at 4, we'll just do a little above it, a little below it. So we'll do 2 and 6, say, with 3 steps, linear, float, generate. So we started at 4, we're also going to look at 2, 4, and 6. Another warning that it changed to JSON, which is okay. Now it's in my JSON. So this is going to do 3 times 3 times 3. It's going to do 27 simulations. And as you can see, we're not actually doing real fine steps, and we're only doing 3 parameter values, but we're already up to 27 simulations, which would be a big problem if each simulation takes 2 days. This simulation doesn't, but it is something you have to keep in mind, that eventually your simulations will get too big. Now we've got everything in there. That was a good idea. Save your file, save all your files with the safe icon here, the multi-disk icon. Now we're ready to run. Now there's a problem. I'm going to run it, but there's actually a problem. And I'll explain what the problem is in a moment. To run it, you run it just like you run any other CompuCell simulation from Twedit. Open player. I'm just going to think about it for a moment. Or I've lost a window. Yeah, there it is. It just took it a while. I had to think about it for a moment. Instead of immediately going to player like normal, since this has got a pscan specified, it opens this new window. This little dialog window. It should be properly filled out with the parameter scan simulation. So that's my full name to this all the way down from C all the way down to my test name that I gave it. There should be an output directory, and by default, if you're working on your local machine, it'll be in your CompuCell workspace with your name of your simulation, test in my case, with parameter scan appended to it. So this is going to create a new directory in this output workspace. There are some options for whether or not you're going to output screenshots, which right now is a bit buggy because it actually depends on whether or not the original simulation had screenshots turned on or not. Just for the heck of it, in this case, I'm going to use the GUI, so it's actually see every simulation run. This, by default, the output, the CC3D installation duration probably filled out already. One handy thing is this display command line button. So there's the command that you could issue in a command window. A DOS window on a Windows machine or any command window in a Linux or Unix machine. And that will run the simulation. You'll want to think about the platform a little bit. You might want to do things like NoHup or some of the other capabilities that let you simulate or launch a run without having to sit there and watch it. But that is the command that you would use. In this case, we're not going to use that command. We're going to go ahead and run it live, so to speak. Again, this will take a moment to start up. And my CompuCell window, and there it goes, it's running. And it says, see at the top of the window here, it says iteration 0. And I'm going to let at least one simulation run. I think I have this set for 5,000 Monte Carlo steps. See, my hetero contact's doing pretty good. This sort is doing pretty good. You see, it's still got some stuff out here on the periphery. It's sort of embedding the blue condensing cells. Certainly my hetero contact area has gone way down, and it's about to hit an end here. You know, I guess I had it set at 10,000 steps. That's unfortunate. Yep, I had it set for 10,000 steps. I'm going to stop it in a moment anyway. You see the top of the window that says this is iteration 0. I'm going to let it run for a moment. Here is my standard CC3D workspace folder. And see, it's created a file called the name I said, my root name test plus added parameter scan to it. So that simulation just ended, and in a moment it'll start up another one. There it goes. And now it says it's on iteration number 1. We start off at iteration 0, now it's on iteration number 1. So I'm going to kill this, which is actually kind of tricky to do. I don't know if that's actually going to stop it by closing that window. We'll have to wait and see if it comes back. And I'll go and look at my output directory. Yeah, see, it continued on. So about the only way to get it to kill is to kill the TWEDIT and the simulation both. I'll go back to that in a moment. This is my output directory. So there's the results for scan iteration 0, scan iteration 1. And if I had screenshots, there would be screenshots in there. And as you can see, it started up another simulation. It's not going to let me, it doesn't want me to be able to stop it, which is actually kind of what it's supposed to do. So what I'm going to have to do is to kill all my Pythons. So it's hard to kill one of those runs. If you accidentally kill a run, you can go back to TWEDIT and just relaunch it, and it'll pick up where it was. We'll restart TWEDIT. So we're back to where we were. Now, if I do open in player, it'll go through the dialogue again, and it will pick up wherever it was when I killed it. So we'll keep on running. Another handy thing that you can do is if you're doing it in the command line and you figured out no hup and how to actually, you don't even need no hup, you could open two command windows and paste this command into both of them and have two simulations running, well, two streams running at the same time. And pscan will keep track of what's going on, and each one of those two jobs will just alternate about which one they're doing. On a single machine, that doesn't make any sense to do that because it's just going to take twice as long for each simulation. You're doing two simulations at once, but it'll take twice as long to do one simulation. If you're on a cluster, you can issue this command as many times as you want. And if you've got multiple CPUs and multiple threads, the system will just fork that all out over whatever you have. We have a small 32 node cluster, and we can issue this command 32 times and have 32 simulations running simultaneously. And pscan will make sure that they don't collide and they don't do any duplicates, and that everything actually gets done. So that's all hunky-dory. I'm going to cancel. I'm not going to run it here. Whoops. Uh-oh. Hopefully it didn't start up. Now, I said there was a problem with this. I'm going to get my PowerPoint back. So there's slides in the deck about it, what I just went through, if you ever want to look at it. There's the final values. And NanoHub, if you set the scan up on NanoHub, I think it works. But if you set the scan up somewhere else, you can't transfer to NanoHub. Same kind of thing in your output directory, you'll have all your files. Again, as I said, where all the output files are. If you want to redo a parameter scan looks at that output file and sees if it exists. And if it does, that's how it decides if it's done or not. If you want to do new scan, you have to either delete that old directory or rename it. So for example, just add save to it or something, so the pscan will run again from the beginning. Okay, so this is where we want to be now. So what we did just worked great. It ran a parameter scan. The only problem is we don't actually have any output because we didn't tell it to write anything. What we'd like to be able to do is write to a single file, appending it every time with the values of our parameters and our nice metric. So we'll have to go back and add writing to a log file. So there's another project file that's already set up to do that, and it's in that zip file. It's cell sorting base pscan write output. And that just goes by the things we started off with today about how you write to a file. You can use the twedit helper for Python utilities, open an output file and open it for append. So it just keeps getting, every time you run, it gets updated. And let me open that one. Get rid of this one. And that will be this one, cell sorting base pscan write output. It's the same basic simulation. It's the cell sort. It's got the graph in it. But now what we're going to do is write the output to a file. This also has the parameter scans already defined, so it's still in there. And remember, this is how it marks the things that are being changed. It's the double curly brackets and then the name we gave it. So in this particular file, I was changing 3 things. To capture that, well, when the pscan Python script runs, it just replaces all occurrences of curly brace, JMC, curly brace with whatever the current value of that is. So you can refer to this structure anywhere you want in your code, including in your little chunk of code where we're going to write to the output file. So we've opened a file. It's for append. We're going to write a line that's tab delimited of our headers, but they're going to be the MC, CC, and CN J values plus our header surface contact. And then another line that converts those values to strings. And again, the pscan Python will replace this with the proper number when this particular job is running. And we're going to write that all to the outline. We're going to print it to the terminal, but then we're going to write it to our file.
Hey, Jim, there are a couple of questions in the chat.
Okay.
I think,
Okay, I'll back up a little bit.
yeah, I mean, I think maybe you went through it very well, but maybe it's worth actually just walking back and going through the steps again. You don't have to do all of them, but just walk people through the workflow when you have a chance.
Okay.
And there were some questions about, some of them are technical and maybe not appropriate to answer here, but you might want to look at the chat and just ask a couple.
Okay, so at this point in the development process, we have a scan developed or defined. And we have a way of writing the results to a single file. So at the end of the day, we know how well each simulation did. But if we want to go back to the very, very beginning, remember we start off with the simulation that we have that's working. And we're going to make a copy of it because we're going to make quite a few changes in that. We want to keep our original file. Over here in the CC3D project menu, if we right click, you get this drop down and there's add a parameter scan. And when you click on that, that will add this new JSON file. And you can open it up and it'll start off closed. You open it up and then double click the file to actually see it. And it'll start off empty, basically empty. Now we want to go through and add all of the parameters that we actually want to scan. And we do that by, for instance, if I want to change the medium to condensing J value, make sure you select the numerical value, not the name of the variable. Numerical value, including all of its, make sure you get the trailing 0 and stuff like that. Right click and add to scan. Now, as you can see, there's another thing here, and I'll just do it try it real quick. There's also, select that, right click, remove from scan. And it'll ask you if you want it for sure. What it'll do is it'll get rid of the tag and put the value back in. I'm going to say no. But it'll put the value back in that it had saved. So it had saved the original value when we defined the pscan. So we can't undo it. And if I do that undo, for instance, if I undo the JCC, it'll just take this chunk out of the JSON file. And in this place here, it'll just put that value of 5 back in. I'm not exactly sure what it would do to a situation like down here in our output where we refer to it again. I think it might just replace this down here with that initial value of 5 as well. But normally you're putting things in, so highlight, right click, add to scan, give it a name. That's the string that'll put back into the file inside curly braces. Tell it your starting value and ending value. Say perhaps 4. I'm going to go 4 to 6. And how many steps you want to do. You can do a lot of steps if you want. And whether or not it's linear or log, whether it's an int or a float. In some cases, it would matter if it was an int or a float. In general, in Python, it would not. However, if you had something like an iteration counter, you wanted to do 10 iterations, you want to make sure that those are integers. And then hit OK, and it'll go ahead and do the changes to the file. So once you got it all set up, make sure you save your file and start it the way you normally do. Open in player. And this is one of the places that changes from the normal behavior in Twedit. Instead of actually going right to player, it gives you this dialog. Now, if I was going to run from a command line, I can just display the command line, copy it, and then cancel. And this guy should not have come up. Hopefully it's not actually going to start to run. Doesn't look like it's gonna. That's good. But now I have in my copy paste buffer command, the whole command. If you want to run on a cluster, let us know. We don't have any really great write-ups, but we've done it a lot, and we can give you the no-hub commands and all that kind of stuff. It's reasonably straightforward, but you want to make sure everything is working really well before you go to a cluster and launch 10,000 jobs, each of which are going to take 3 hours. You definitely want to make sure everything is set up to work right. Okay, so in this particular version, yes, this has the parameter scan in it. It has a little chunk of code that calculates the quality of the cell sort just by summing up the heterologous contact area for condensing cells to all of the condensing cells. And it has a little chunk of code in the finish to open a file, write the value to it, and then close the file. At the beginning of this session, I talked about using the start to open a file and then write it in step and then close it in finish. But in this case, all we want to write is how well the simulation worked in our parameters. So in the finish, we do everything. We open the file, write to it, and close it. So down here is an open, put the our output string into it, and then close the file, and we're done. Now I'm going to try to run this one, but since we don't want to watch it for too long, I'm gonna make it go even faster. Let's say we'll just do 2,000 steps per simulation. And I just did open in player, and it's going to intercept me and not go to the normal place, which is okay, just fine. We'll go ahead and use the GUI again. Go run. Yes, James made a good point in the chat. I will go and find my output directory. And I'm getting too many windows open here. Yep. Here is my my command box, my typical log that you get from CompuCell, and it says, wait a minute, the parameter scan is completed down here at the bottom. The reason it says it's completed is because I'm trying to write to the same output file that I used before. And that file has all the scans in it. And as far as it's concerned, it was done. And James's comment was in each one of these files, so scan duration O, there's my JSON file, there's my CC3 dial. I can keep drilling down. There's all of my stepables and everything else that goes with that job. So this scan iteration directory has everything that was done for that particular simulation. So in any case, so the problem is it won't run another parameter scan because it's pointing at the same output directory. Here I have a couple of choices. I could just rename this. Yeah, I want to keep this one that's got a different name so CompuCell won't see it, or I could just delete it. Whatever seems to make sense at the particular time. So let's go back. I've hidden that file. Open in player. Think about it for a moment. There it is. Really nothing I need to do. I'm going to hit run. And I got the same message again. It still thinks that that parameter scan is alive someplace. So I need to go figure out where that guy is hiding. This is one of the things that gets really difficult on NanoHub is because the file structure is so arcane and you don't have access to all of the different file structures. I'm just going to delete a whole bunch of my old ones. Back to Twedit, back to open in player. And I got the same message. So I'm going to do something a little different. Open in player. It's the output directory. Gonna nuke them all. This is interesting that I'm getting this clash. It should not be getting this clash. This file shouldn't be there anymore. So that's enough. Back to open in player. This is cell sorting base, pscan write output. Cancel. I had slide one unopened. There's back. So there's the output directory. That's the thing that has to be non-existent or only partly done. And actually right now we want it to be non-existent. We want to create it. And there we go. Looks like it's going to start. And I forgot to turn on the GUI, so it's running without the GUI. Well, that's okay. It'll actually be a bit faster without the GUI. And this is our standard CompuCell CC3D workspace output, cell sorting, parameter base, pscan. And you can see it's updating. So 0 scan, 1 scan, 2 scan. And we were writing this scan file, this log file. I double clicked on it and get it to open up here. You see there's another one file. So there's my log file. I remember we wrote a little header that was the 3 things we were changing, and then the header surface. This is tab delimited. I don't want it in Excel, automatically opened in Excel. And then here's the values for one run, and it's appending to this file. So if I reload the file, it's continuing to add lines to it. Now you might say, well, it'd be nice not to have all these duplicates of the header line, but eventually what I'm going to do is take this whole thing into Excel, and I can just sort it and delete all these extra headers. But so you can see, so it did JMC was 8 and JCC was 3 and JCCN was 5, and I got 668 as my heterologous contact area. The next run it was 650 and then another 650 and then a 902, and it's just going to keep chewing through all of these possibles. And this guy's doing 27 simulations, so it actually isn't going to take it that long. It'll take it another 5 minutes or something. But we probably don't want to wait for it. Okay. So this guy's running. I'm just going to let him run in the background. Here's my command log. So it just it'll count up to 5. Yeah, we're only doing 2,000 steps. So that finished, and now it's doing another one. So I'm just going to keep chewing through it. So let's go back to our slide deck. So we had changed our parameter scans with writing files, writing a log. That's all in the slides if you want to go back and look at it in more detail if you forget how to do it. And this is what this job is going to eventually create. Is that file I just showed you. This is our log file, and there's 27 lines total. Take that and paste it into Excel. Sort it. And in this case, I've sorted by this heterologous surface area. This is the number we're trying to minimize. So the best one I found was 278. Here's a graph of the blue line is just in order of runs of that heterologous surface area, our metric. So it's oscillating up and down from the periodicity of the parameter scan. If you take all these things and sort it like they are here and plot that our quality metric, see there's looks like 6 or 7 simulations that basically give the same result in terms of our quality metric, and then a bunch of simulations that give much worse results. So we have it sorted here. Here's the best run, 278, and our 3 values that were 278. Just looking at it, you can make some conclusions of this on this sort, that JCC was always 3 or 5. JCN was either 7 or 9. But JMC had all 3 of its values, 8, 10, and 12. So it looks like these two, JCC and JCN, are doing more than JMC. And you can, of course, start playing with all the numbers and do, well, what's the correlation between each of these J values and the quality of the fit? Well, here's the correlations. JMC to the hetero surface area correlation is 0.1. JCC is 0.4, 0.5, basically. JCN is 0.75. So JCC and JCN are the one are really driving how good this sort is. And you could just say, well, these are the best values here, 12, 3, and 7, which are pretty close to what we did before. You could also do a nonlinear regression with parabolas and try to interpolate what the optimal values are. But that's downstream. So that's the basics of a parameter scan. It's relatively straightforward. It's a little tedious sometimes because of deleting files and making sure you've got file data being written somewhere you can use it. If your simulations take a couple hours, your parameter scan could take a very, very long time. And so you'll want to be so much selective about what things you're scanning. It's also entirely reasonable to do a very coarse scan over a few parameters. And when you get a better idea what those parameters should be, doing a finer scan separately, taking smaller steps. Okay. So as everyone knows from the past week, CompuCell is stochastic, and depending on exactly the characteristics of your model, you can get anything from very small run-to-run variations just from the stochasticity to very big changes from the stochasticity, from run-to-run. So it's often very useful to be able to do replicate runs, do the same simulation 5 times, 10 times, however many times you think are necessary. And it turns out that the parameter scan is actually a really handy way of doing that, since it will take care of spawning the jobs, how many it's done, how many it's supposed to do. If it gets interrupted, you can restart it and it'll pick up where it left off. And if you've already built into your code a metric function and the ability to write that metric out to a file, you can collect all your simulation, your metric from across a large number of simulations, even if you're not actually scanning a variable. So it's easy enough to do. One way to do it is we'll just add a dummy variable in our Python, like replicate count. And originally it had some value, and I highlighted it and right-clicked on it and said, add to parameter scan. And in our output, we're going to print that replicate count as well, just so we have it in our output. And our JSON file just now looks like we just have a new variable with some values. So this particular job looks like it's doing 2 values of JMC, a single value for JCC. There's nothing wrong with doing a single value in a parameter scan. It's just do it once. Same thing with JCN, and then it was going to replicate it 3 times. So this then launches exactly the same way. Right-click in Twedit and click Run, and you'll get the dialog, and you can copy the command line or launch it and go ahead and run it locally and immediately. So that's how you do replicates. You don't have to have any other scans, right? You could do a parameter scan that did nothing but the exact same simulation 10 times. So we have that. There's a version of it in the zip file called, I keep getting my name longer and longer and longer, cell sorting base, pscan, write output replicates. I think I'll go and open that one. So this is exactly the same as the ones we've been looking at, except for now in our parameter scan file, we're going to do 3 replicates. And now I'm not going to run this because this is 27 times 3, 75-ish simulations, and it'll take a while. But it's still going to write this an output file very similar to the one we had before. Let's find it. This one. The output file looks exactly like this one, except there's one more column, and that column is iteration count. So you'll find 3 rows where I had these 3 values, and each one will have its own score, because the cell sorting is actually quite sensitive to, it varies a lot from run to run. So you'll have 3 runs, same J values, 3 different outputs. You could take the average of those outputs as a measure of how good the parameter set is. Or you could say how good the parameter set is the best value. It's kind of tricky to figure out what the best way of deciding what the best value would be, whether you use the average or the minimum for a set. So that's, again, fairly straightforward if you're doing, I lost my spot here, replicate runs. So if you want to do 50 runs, you can do it. And the pscan is a nice handy way of doing it. And you can, of course, do as here, replicate runs as you're doing the parameter scan. Okay, so getting kind of late, 5:30, we still have some time. I'll go a little bit farther. Parameter sensitivity. Parameter sensitivity is how much the output of a model changes if you change an input parameter by some fixed amount. This is a good approach to deciding what parameters in your model actually make the most difference. Sometimes you'll have parameters in your model, and the model is really quite insensitive to exactly what that value is. You really love those parameters, though, because that means you don't need to know what they are, or at least you don't need to know what they are accurately. In the parameter scan we did earlier, I think it was the medium to non-condensing, the metric was pretty insensitive to that value, which is great. That means I don't need to know that value to great precision. It just has very little effect on the model. I still need the value in the model, but I don't need to know it with high accuracy. Parameter sensitivity, on the other hand, will tell you that this parameter just does not affect the model very much. Parameter sensitivity will also tell you which parameters actually make a difference. Parameter sensitivity is reasonably straightforward. You can get lost in the details because there's a number of ways of defining what you're trying to do. What we're going to do here is just talk a little bit about it. You could use pscan to do it. basically just give every parameter two values, the reference value and a change value. It's not the most efficient way to do it. For a small number of parameters, say 10 or less, it's probably easiest just to do it manually. Change the parameter, make a run, write down the metric, then go on to the next one. So a little bit more about parameter sensitivity. What you do is you have a reference set of values. So here we have three values in three parameters in our model, and we're going to do a run at the reference value for those three parameters. And then for each parameter, we're going to do one run where we change that parameter by some amount. And if you look in the literature, frequently people do 0.1%. They change their parameter by 0.1%. You might have noticed in CompuCell that CompuCell is not going to respond to small changes like that. It's going to get lost in the stochastic noise. So you need to do pretty big changes, much bigger than a hard ODE model might use. So maybe we'll do 20% changes. So we'll take our value and we'll do run with P1 value changed by 20%. And then we change P1 back to where it was and do the same thing for our parameter number 2. Then we change it back to where it was, same thing for parameter number 3. The nice thing about sensitivity is if you have 10 parameters, it just takes 10 plus 1 simulations. So you do sensitivity a lot faster than you do a parameter scan. So that's the overall plan. No reason you can't do 20 or 30 parameters, even if you're doing it by hand. So what is the sensitivity actually defined as? The sensitivity is defined as the change in output divided by the change in that parameter. So each parameter has its own sensitivity. This number is the standard way of doing it. The problem with doing it this way is that your parameters have a wide range of values. You'll have some parameters that are 10,000 and some parameters are 10 minus 6. And so if you just go by the change in the delta parameter, you've got a difference of orders of magnitude in how big this number is. So the usual way of doing this is to normalize. It's the change in the parameter divided by the reference parameter, and it's the change in the output divided by the reference output. Basically just making fractions of both of these values. If you do that, you can now compare the sensitivities for different parameters, even if those parameters have radically different numerical values. You'll be able to say that if I change this parameter 10%, the model changes by 20%. If I change that parameter over there by 10%, the model changes by 5%. Therefore, my first parameter is more sensitive than my second parameter, without getting lost in the fact that the parameters themselves have different magnitudes. So in practice, it's just if all of your delta parameters are the same, that your fractional parameters are all the same, the sensitivity is just how much the output changes divided by the normalized or the normal output or the reference output. It probably sounds more complicated than it really is. It's pretty straightforward. So the thing though is that you're doing a sensitivity at a fixed point. So back here, our fixed point is these three values, whatever they happen to be. And we're looking at sensitivity as you change around this fixed point. If our fixed point is different, our sensitivities may well be different. In general, our sensitivities will be different. So when you're doing a sensitivity, you want to make sure you're at a fixed point that makes sense. Typically, that fixed point is at a set of parameters that either generates a reasonable output as far as you're concerned, or perhaps a set of parameters that reproduces an experimental result. And of course, that would be where you'd be most interested. You want what's the sensitivity of my model to the parameters at a set of parameters that gives me a biologically relevant result. So the first thing, if you're going to do sensitivity, is you need a reasonable fixed point. The second thing to consider is exactly what is the output you're using. In the cell sorting, we've been using the heterologous contact area. But in other simulations, you might have multiple metrics that all make sense. In the cell sorting, you might say how many cells are left at the end of the simulation. If you crank up adhesion too much, you might start squishing cells out of existence. And that would be a bad thing. So you might want to do a sensitivity of what's the best set of parameters that sorts the cells without squishing any out of existence. But that's 2 metrics. And so each one of those metrics would have its own sensitivity. So things can be complicated. The more complicated your model is, the more outputs you might be interested in, which means more sensitivities you would have. I think I'll skip that. That's just the numerics. So we can do sensitivity with the parameter scan. It's not the most elegant way of doing it, but it is one way of doing it. Basically by saying, here's our reference value for this one J value is 10, and I'm going to change it by 5%. So my second value is just the first value plus 5%. This one was 3, and if I add 5%, it's 3.15. 9, if I add 5%, 9.45, so on. And I want replicates. So this P-scan will do a sensitivity analysis, and it will do 3 replicates, so you have some way of dealing with the stochasticity of the CompuCell simulation. Reasonably straightforward. The problem is with this approach is it's going to do exhaustive sensitivity, which is every single parameter gets tested once. But it also tests every situation where 2 parameters are changed and every situation where 3 parameters are changed. Usually you want single parameters. Sometimes you want pairwise parameters because that tells you if parameters interact with each other. You almost never want these higher order ones. They're too complicated and too hard to interpret. So in using the P-scan, it's kind of overkill and we're getting a bunch of values we don't actually need. But again, it's an easy way of doing it, and it's an easy way of building in replicates. It just means you'll have to do some post-processing when you're done to kind of sort out all of your various values. You'll need to find the one simulation that has all your reference values, and then the individual simulations where exactly one other parameter is changed. It's doable. It's just a little fiddling in Excel. Again, it's getting pretty late. So we can do sensitivity analysis, very handy to be able to come back into your model and say what sensitivity is. This is the result of doing the sensitivity analysis on this cell sorting model. So here we have the reference simulation with our 3 J values and a single run. I'm pretty sure I did this as a single run. The metric, quality metric was 301. And then what happens if you change this guy by 5%, change this guy by 5%, change this guy by 5%, use the formula that we had earlier, you find the sensitivity for JMC is very low, 0.2. Sensitivity for JCC is much higher, 4. What the 4 means is if I make a 5% change in that J value, I get a 20% change in the quality metric. So these 2 parameters are quite sensitive, and this parameter is quite insensitive, and these 2 are quite sensitive. The other thing you can do, if you actually work through all the numbers and do the pairwise sensitivities, you can determine if parameters interact with each other. If the parameters don't interact, their effects sum. You can just add up the effects for the individual changes and predict what happens when you do the double change. And the exhaustive scan will give you both of all 3 of those numbers. If the parameters do interact, then you can't just add the individual effects. And there are a lot of places where parameters interact. A common one that you might have is if you're using Michaelis-Menten equation anywhere, the Km and the Vmax are highly correlated with each other, and they strongly interact with each other. And depending on your system, you may or may not be able to determine values for Vmax and Km. It's not surprising that they interact because they occur in the same equation. So the more interesting ones are when you get interactions between parameters that are not in the same equation. So situations like in our cell sorting, where an adhesion is interacting strongly with, say, your lambda volume for the cell type. It's warning you that there's something more complicated going on there. Not necessarily wrong, but it's just that there's something more complicated going on. And it's getting even later. Some quick caveats on this thing that That was for demonstration purposes only, because you're only doing 5,000 steps, and it really should have been done for a lot more steps. I will skip to conclusions, and that's just a list of all our files. I wanted to real quickly go over, as you've been working this week, you probably noticed it's easy to get lots of sequential changes in a file and starting to lose track of what files are what, and not having a file that you can back up to if your simulation gets broken in some way and you can't figure out the problem. So for best practices, you really should develop some routine for versioning your projects. If you're a programmer, programmers love GitHub and SourceForge. It's a great system. It's got a really steep learning curve. Alternatively, you can just put the files onto something like Google Drive or Dropbox, which automatically versions all your files, since Windows and Mac don't usually, and Linux don't usually version your files. You put it on Google Drive, you can scroll back 72 variable or 72 versions of your file if you want to. That's okay. Problem is you get all these old versions, you don't actually know which one to scroll back to. Kind of a simple and easy one to do is just periodically just version. Actually, I'm going to skip one more. Is just version your project every so often using the zip it in Twedit. You get the point, you've got all the bugs out, it's printing the right thing, it's not throwing errors because you're missing a quote mark or something like that. And it's doing something useful, you've added a new function. In Twedit, just make a zip of it and write the date on it, rename it with the date. So you just have this sequence of old zip files. You know, this is where I got cell sorting to work. This is where I got writing the metric to the file working. This is where I got the pscan working, and just version it that way. So you have old versions. Very straightforward, very easy, non-programmer, not sharing it with anybody routinely. It's a great way to do it. Much better than just ending up with only the most recent file of your most recent version of your simulation. It's also a good idea in your simulation folder to just keep a Word document or something of the basic description of what you're doing. What are your cell types? You know, this is a wound healing model, and I got 6 cell types, and I've got 2 diffusing species. Just a word description, a prose description of what you're trying to do. You can expand and contract that description as you go on. You find out you have to add something to your model, take something out. But just keep that nice little biological description. You can also use it as your elevator pitch when you're showing it to someone else. And quickly describe what you're trying to do. Next thing is parameters. As you've noticed, you have parameters in your file, and they tend to be spread out. They're in different places. Some of them are in your Python file, some of them are in your XML file, some of them are in some other file someplace else. It's difficult to keep track of them. It's difficult to make sure you're not making mistakes like mixing units or not handling units correctly. So though we're getting really late, and so you can look at these slides at your convenience. One thing you can do is just at the top of your Python file, just define some parameters. Like here I have converting seconds to MCS. That's my conversion factor. If I want to convert days to MCS, that's my conversion factor. And my cell diameter is going to be 5. I can then refer to these things anywhere in this Python file as just S to MCS. I can't change it in other places, but all of my steppables and things like that will inherit this variable. So this is one approach. It's a beginning. It's better than doing nothing, but it's not the greatest. A little better way is to create a parameter file and put your parameters there. And you can do that as just a Python file that you're going to add into your simulation. There's a demo here. I think people have noticed this little checkbox that you haven't used yet that says Python only. So this will create a simulation where that XML file doesn't exist. And instead, what you get is all that XML stuff as Python. The nice thing about it being Python is you can do things like put a variable in how many Monte Carlo steps you want to do, for example, or the X dimension of the model. That can be a variable now, and you can change that variable in code. Whereas in the XML version, you can't do that. So that's a handy way if you combine that with a parameter file of doing just that. You're having a parameter file that says my model width is 50 by 50 instead of having it hard coded into the XML. There's a demo, the CC parameters example that's in that zip file that does a pretty exhaustive set of parameters. I just lost my spot. To use a parameter file, you basically, well, actually, let's actually do it. It's probably more use if I actually go through doing it. So over here in my left thing, I can say, we had add parameter scale before. We don't want to do that now. What we want to do is add a resource. So you could add a resource and it says, what do you want to call it? We'll call it parameters, except I can't type. Its file type is just going to be Python. It's going to live in the simulation directory where all of our other files live. Finish. Now I have a file here called parameters. Double click it and open it up. And this is now my parameter file. Now I can do something like, say, and the volume equals 4. And I can also do calculations here. Lambda volume is something scaled by the width of the model or something else. To use it in my regular steppable file, I just need to import parameters as P. I can't type. And now I can use that value as, trying to find where I used it. So in my parameters file, I have a thing called lambda volume. In my steppables, I'm going to import my parameters as P, and I can refer to lambda volume as p.lambda volume. And I can make a change here. Unfortunately, I can't do that, this particular version. This is our old XML version. I can't do that with these values. But that's why you'd want to do a Python only simulation so that this file was in Python, and therefore you could import the J value. You could also import things like the model width and all, and you know, the dimensions, these dimensions up here, the number of steps you're going to do, the temperature, anything you want. And you can calculate all those. So this is just showing, so I import parameters as P, and then how I'd use P. Our parameter file can include conversions and equations. So if I define my model in terms of cell widths instead of pixels, and I say my cells are a certain width and my conversion for pixels to microns, I can calculate how wide my CompuCell model needs to be. And I can use that at the startup of the model. Now I can come in and change my resolution of my model by changing these values. Let's say it's 5 pixels per micron. Make it run really fast because the cells are only a couple of pixels across. The other thing you can do in this file is write down what you think the units are, or what the units are supposed to be. So this is supposed to be in microns. And it's the cell width in microns. And oh, if you have a reference, you could put a reference in. So if you had a diffusion constant that you pulled out of a paper, you could put the diffusion constant in and say that's in microns squared per second. And oh, that came out of PMID such and such a paper. It's a good way to keep track of all your parameters and things in your model. So there's an example in my set of zip files. TJ has an extensive parameter file in his viral model file as well. This is one way of doing it. There are more complicated ways of doing it using a dictionary so that you could actually keep track of the name of the variable, the value, the unit, and the source, all as a dictionary of things. We won't go into that here. It's more detail than we need. But in any case, so this is something to consider, making a parameter file and keeping all your parameters separate, not having them scattered all throughout your codes. Okay, I'll check the chat real quick. So should the parameter file have a specific structure? I kind of messed it up here, but I think it should. You should have a section for biological parameters. That's where you decide things like my cells are 10 microns across, or my model is 20 cells across. So those are purely biological descriptors, and it has biological units like microns or seconds. I would then have a domain that's your conversion factors. You decided it's 1 micron per pixel. So you can generate a conversion factor with that, or it's 3 microns per pixels. Also, what is your conversion from seconds or hours to Monte Carlo steps? What's your time conversion? You can hard code, or you should specify what that is in this file. Those values can change, but you should actually have them defined explicitly in the file. So if somebody says, what is 50 MCS in seconds, you can tell them, well, it's supposed to be 50 seconds, 50 minutes, 50 hours, whatever it happens to be. And then, so biological parameters, conversion factors, and then model parameters. And this is where I kind of messed it up. The model width in pixels is a model parameter, and that can be calculated by, the model is supposed to be 20 cells wide. Each cell is 10 microns wide, and it's 2 microns per pixel, or whatever the value is. So you might have those 3 kind of characterizations: your biological parameters, your conversion factors into CompuCell units, and then what that results in terms of the parameter in the model. So that's one way to do it. There's no real, strict definition of how to do this. It might be worthwhile sometime in the future for CompuCell to have a class for a parameter where you could keep the unit, the numerical value, the name, the unit, and some reference to it, and just generalize that into CompuCell. Okay, since it's getting so late, I think I will stop there. So we covered a lot of stuff. Some ways of writing to files, and you can go into my deck here and get the code. It's also in the demos. Parameter scans, reasonably straightforward, can be a little picky about certain characteristics. You can also use parameter scans to run replicates. We talked about sensitivity analysis. We saw a real simple example in cell sorting. Definitely something you want to consider when your model is getting more mature and you've got components in it and you think it's giving reasonable results. Sensitivity analysis is an interesting thing to look at. Tell you what you need to know precisely and what you don't. Parameter files, a better way of keeping track of things, and best practices about doing something like versioning and whatever you're comfortable in versioning, whether that's zip file or GitHub, whatever it is that you like. I know TJ et al. Use GitHub for the viral model. It's extensively versioned and tracked. And it's 6 o'clock, so I'm a half hour late. Excuse me. Okay, so if there are any other questions. So let's see, I think we are starting tomorrow morning at 10 for the hackathon. And I believe we'll probably start with just a general discussion of everybody, with everybody in the room, and then pretty quickly break out into the separate projects.
I have one question. So what is MCS in terms of biological time?
Sure. That's a tricky one. In a lot of cases, how to convert MCS to real time. In a lot of cases, it actually doesn't matter a whole lot. If you're doing, say, cell proliferation, you would probably want enough Monte Carlo steps per second so that as you tell the cell to grow, it's actually keeping up with what it's supposed to be doing. If you tell the cell to grow to double volume, for instance, and say, well, 1 MCS is 1 second, well, cell doesn't actually grow that fast. So what you would do in the case of growing cells is you might say, how many Monte Carlos does it take to actually get it to grow so that it's never too much smaller than it's supposed to be. And then go and look, well, what's the doubling time for this cell type? It's 12 hours. Well, and now I know it takes 150 Monte Carlo steps to get that cell to grow properly in CompuCell. And that's supposed to represent 12 hours biological time. So now you have your conversion. Other ones to use are things like if you're doing chemotaxis, if your Monte Carlo step represents too much time, you're trying to make the cell move too fast, and the cell will fall apart. You'll have to put too big of a force to get it to, it's supposed to chemotax 1 micron a minute, and in order to get that to do it in your model, it takes too big of a force and your cell falls apart. So you have to use smaller forces and more Monte Carlo steps to get it to move 1 pixel. And that is the speed limit, and that's how you would set your Monte Carlo to real-time conversion. Like I said at the beginning, a lot of times it actually does not matter a whole lot. If a Monte Carlo step is 1 minute or 2 minutes, might not make any difference at all. It will make a difference if you think a Monte Carlo step is 1 second, and it really should be 1 month. Cancer models, your Monte Carlo steps typically pretty big because tumors tend to grow pretty slow. And you don't want to be doing, 1 second Monte Carlo steps because it'll take you too many steps to simulate 2 years or 6 years or however long you think it takes for a tumor to grow.
Yeah, I think you can make them as small as you want, but you can't make them too big. Basically, it's like any other computation where if things change too much during one step, you have numerical issues.
Yeah.
And so you have to figure out what the fastest process is in the simulation. And that fastest process will define the time step that you have to use. You can, if the fastest processes are not cell movement, you can substep to basically subdivide that intrinsic Monte Carlo step into subunits. There's a very awkwardly named quantity called flip to dim ratio, which really makes no sense at all as a name if you don't have the history, which allows you to say, I want to call the diffusion solvers or the antimony solvers 5 times every Monte Carlo step instead of every Monte Carlo step. And so you can do some fine grain tuning that way, although it has to be done by hand.
Yeah. And on the other end of the scale is if your time step is much too small, the Monte Carlo step is a millisecond. The problem is you're trying to model a slow process, you have way too many steps, and it's just going to be too slow. So on the low end, you can't break the model. And on the high end, you don't want to spend a whole bunch of time calculating these real fine steps that don't actually do anything of use. You have to find a happy medium. And as I said earlier, usually that happy medium is a fairly broad range. It's not a real sharp minima. It tends to be, it gets better, it gets better, it gets better, and then it goes flat. And you can use anything in the flat and it works just fine. You just don't want to be on this slope where as I change my step size, the model is behaving differently.
Right.
As long as the model is, if you make small changes in your Monte Carlo step size and the model behaves the same way, then you're reasonably confident that your model is not sensitive to your step size.
I mean, it does depend. For example, in the liver simulations where we were modeling blood flow and capillaries explicitly, blood flow in capillaries is very, very fast.
Yeah.
And if you want to model the changes due to pulsatile availability of a chemical in the blood, you wind up having Monte Carlo steps of 1/100 of a second.
Yep.
And that makes the
that's right.
simulations very, very expensive.
Yeah.
If you're doing simulations where you're having immune cells move, some of the leukocytes move very quickly. Macrophages crawl quite rapidly. And so then your Monte Carlo step might be a few seconds to 15 seconds. For most developmental simulations, a Monte Carlo step can be a minute or 2 minutes. represent a minute or 2 minutes. And there definitely are simulations we do, for example, diabetic retinopathy, where things happen very slowly, and the typical time step we'll use is 15 minutes or 20 minutes. So it's really determined by the timescales of the biology that you're trying to represent.
Yeah, so when in doubt, say 1 minute, one Monte Carlo, and write that in your parameter file. And then as you work through the process of building your model, you'll find out that that number just doesn't work. It doesn't match your diffusion coefficients or your growth rates or something else. And that's fine. Then you in your parameter file, you change it. So yeah, Monte Carlo step is now 6 minutes or whatever it should be. And that's fine. But at least if you have a number written down, you know where you're going.
Right. One thing you have to remember is that if you have probabilities of transitions, you have to use the Poisson rule when you change the step size. It's not linear in the step size. But beyond that, typically we are careful about that these days. We'll have that equivalence, say 1 Monte Carlo step equals X amount of real time. And then when we change that, we have all the parameters written so that they automatically rescale appropriately when we do that.
Right.
But again, that's the kind of thing which in principle would be very nice to have automated, but in fact, every simulation is so different in terms of the way it handles it. It's very hard to think of a way of doing that automatically. So I just wanted to close up, and we're having a good discussion here. I don't want to cut it off. Tomorrow we have the hackathon. I hope we'll see a lot of you there. We'll start officially at 10 a.m. I'll try to open the Zoom at 9:30. Please, if you have a name for your hackathon team, please put it into the Google Doc so that I can have it to set up breakout rooms. I will ask people to go over one more time very quickly their projects so that people who want to join the hackathon who don't have a project yet can decide who they want to join, at least temporarily. If people decide they want to merge projects because there are two projects that are very similar, I don't see too many like that, but it's possible. Let me know. Then from 11 to 1, we'll have a work session. We'll have as many of our assistants as possible available. My guess is there'll be more people to help out Saturday than Sunday, just because everybody's getting tired. And I encourage you, if you haven't started yet on your project, to start thinking about some of the material that I provided earlier today. We'll do a very quick half-hour check-in. You don't have to do the check-in, but we'll be available for a check-in around 1 p.m. And then actually, I have a self-inconsistent timing here. Between 1 and 3, there were some requests for some informal presentations. I don't want to distract from the hackathon too much, but we'll use the main room. TJ will show how to do advanced applications, how to call CompuCell as a Python library element. So to use that, people have asked for that. He might talk a little bit about job scheduling as well, his Slurm script. Giuliano will talk about how to build NanoHub applications. We've been using the raw CompuCell NanoHub mostly, but TJ showed a NanoHub demo of the COVID model that was a packaged NanoHub app. So if you write a simulation in CompuCell and you want to have that packaged as a freestanding application, Giuliano will show you how to do that. And then Julio has agreed. There were a lot of requests to show how to do this subcellular chemical concentrations moving around in the cell. And he has this nice planar polarity and Drosophila demo, maybe also a PIP23 demo that he could show briefly. These won't be polished necessarily. They may not, demonstrations, but there'll be an opportunity for people to find out a little bit more. I hope it doesn't distract too much from people working on their own projects. We will record them and make them available. So if you miss them, you can always go back and watch them another time. But these are topics that we didn't get to during the main course. And I'd like people to have the opportunity to see that material if they want to. I would, again, encourage people to focus on their hackathon projects. If you are part of the hackathon, you don't have a team, please look over the list of possibilities. I know Priyam, who has been an instructor in previous classes, agreed to come and help us out tomorrow morning a little bit. She's an expert in using links and doing epithelial reorganization, so that should be very helpful in doing compartmental cells as well. So, for example, for the lateral line project, I think she has a lot of relevant expertise. So that would be our schedule for tomorrow. Are there any more questions before we break? I know it's been a very long week. I appreciate those of you who've stuck it out all the way. I really appreciate the ideas that people have for projects, and I hope we can make those work. I'm quite excited by the variety of projects that people have proposed. So are there any final questions? We're already over by 15 minutes, and I know for our European colleagues, it's very late.
I have one doubt regarding the hackathon. So the past day reports means this this format that you have sent that report, or what else? Like that one or anything else?
No, it's not that formal. But at the end of the day, I would like to go through all the teams and have each team report in front of everybody what they did. That could be a slide or two. It could be showing a simulation. It could be saying you got stuck. It could be a document that shows what the conceptual model is, any of these things. But we have a lot of expertise and a lot of ideas. And so I think it's worth spending 45 minutes learning from each other and seeing what worked and what didn't. So the goal of this first day report is not to have a formal report. It's not that you have to hand in a 20-page PLOS computational biology paper. It's more to have an opportunity for people to present, learn from each other, discuss problems, and help each other out. Okay.
Okay, thank you.
I actually have a specific question. Sorry, I won't be able to participate in the hackathon for the weekend, but I just want to ask this question. So is there any ways, because I know like the volume flex can make the cells freeze, but is there any way to set up like a material properties to the cells or to like the substrate materials? Because what I want to do the simulation is like make my cell adhere to a substrate which have different Young's modulus, and I don't want to apply the forces directly to the cells, but I want to apply the forces on the substrate and let the substrate stretch the cells.
Okay, so there are two possible ways of doing that. One of them would be to use a finite element package like FEBio, or one of the Python libraries that models the solid material, and then couple the solid material using the explicit force that Giuliano showed in the second second day between the solids model and CompuCell using the Python interface. That may be the most efficient way to do it. If you want to do it entirely inside of CompuCell, it's very possible to do that. You can divide your solid material into small blobs using the cells, generalized cells. You cross-link those blobs using links, and the spring constant of the link is the Young's modulus of your material. And then you can couple your cells to the subcellular extracellular material that way. The only disadvantage of doing it that way is, one, because you're on the lattice, there's a little bit of nonlinearity in the mechanical response. If the forces are very small, things get stuck. So there's a binding friction, a starting friction term. And for larger forces, it's fine, but for small forces, that can be an issue. And you have to use a fairly small lattice constant because you need to have the size of your blobs of material in the solid be multiple pixels and still much smaller than the cell. But you definitely can do that, and people have done that. Last year, there were people who built models of extracellular elastic material and then put cells on top of them and then pulled on the elastic material and had that pull on the cell. So that's definitely a possible thing to do.
Okay, I see. Thank you. So so you're saying there is a way that to implement FE-bio into the CompuCell3D?
No, I'm not saying anybody's ever done it. I'm saying it's possible to do it.
Okay.
There are Python libraries that do finite element simulations.
Okay.
TJ, are you still on the call? What is the one we were just talking about the other day?
Now I'm trying to think of it, and I'm also blanking.
Sorry, can you repeat that? I was distracted for a second.
So there are a number of solids and fluids modeling packages in Python.
Yes.
And you can use any of those inside of CompuCell if you want to implement a solid material or a liquid using those. But then you're on your own in the sense that you have to then write a description of how the objects created in those models exert forces and have forces exerted on them by CompuCell cells. It's not intrinsically difficult to do, but it does take some thought to make it work efficiently. But there's certainly no reason you can't do it. A long time ago, we had a Phoenix integration part, but the problem with Phoenix was it doesn't run on Windows, so we didn't pursue it.
Actually, today I was very impressed by the 3 sort of cell version of creating these for the apical constriction, and that together with what we learned yesterday about single cell migration, it's made me rethink how I might incorporate these ideas into a primordium which has a subset of cells that undergo apical constriction and a subset that remain more mesenchymal for collective migration. And that actually might be a good way to do it, which would not require me to go outside and actually take full advantage of many of the things we learned in a couple of days.
Yeah, I mean, the simulations you showed, the ones you showed it with NetLogo.
Yeah.
Those you should be able to replicate relatively quite straightforwardly.
Yeah, no,
if you want to include more mechanical properties, again, the issue that comes up always when you do detailed mechanics is that you have to have very fine pixel size. And so then the simulations get to be quite large in terms of voxel by voxel, because you need to have a high spatial resolution. But it works quite well. In the past, this year we didn't have time to do it. I've written simulations where you build essentially a square of a material, cross-link of blobs, cross-linked by links.
Yeah.
And then you pull on it and measure the stress-strain relation. And then you can adjust if you use crossed links, you can have Poisson ratios, different Poisson ratios, and this kind of thing. You're never going to be as good a solver as Abacus is, because they've spent billions of dollars developing code to do these things in great detail. On the other hand, it's very convenient to be able to do everything within one package rather than to have to go outside of it. And so you have to decide what the trade-offs are for your particular application.
No, considering I already got something useful out of something as simple as NetLogo, I think this is already going to allow me to do a lot of very interesting things. And so, yeah, I'm actually much more optimistic. In fact, we had this little discussion about how reasonable it is to elongate the cell. And actually, you raised a very good issue, because I think that I was too focused on getting everything done simply by cell adhesion, but in our cells that undergo apical constriction, they have actually been epithelialized, means their shape has become elongated. They have acquired that polarity. So actually a bonus to include that as a feature of how the cells change. And conversely, I might use that feature to define what a more mesenchymal cell is by changing that in the opposite direction. And it turns out the microtubules change their orientation in migratory cells versus constricting cells. So all that actually becomes biologically perfectly reasonable.
Right. Well, if you look at the simulation that Gilberto did, he didn't show the 3D version of the cell crawling this year, but that's a very mesenchymal cell.
Yes.
He's modeling essentially a
No.
keratocyte.
So I'm thinking of actually incorporating both ideas now.
Right. And if you look at Julio's simulations of segmentation or of kidney polycystic kidney disease, in that case, he has very strongly polarized cells that are very elongated. And he uses this method of defining apical basal domains and then defining essentially something that represents the extension of the cell by the microtubules. And that works quite well. Again, as you make the cells more detailed, the lattice gets more fine grained, and therefore the computation gets more expensive. But once you have a simulation that does what you want, if the simulation takes a long time, it's no problem. You throw it in the cluster and you wait. When you're fine tuning it and trying to get the parameters working, then having a slow simulation is more of an issue.
Yeah. So actually, I wanted to go back and read and look at the model where it was mentioned that the somatogenesis happens as a consequence of mechanics rather than how it's normally described. So which paper is that? I tried to look it up. There is one paper, but it seems just superficially when I looked at it, it was more like the traditional notch oscillation clock and wave front. Is there a different one which describes it much more purely on the basis of mechanics?
So Julio has a paper in Science that does it. The modeling is all in the supplemental materials, so you have to look in the supplemental materials.
I looked that up. Okay.
And then there's a recent paper with Julio and Priam on mechanical instabilities in epithelial layers that does some of that related to somatogenesis. And then Priam has some work that she did in her PhD thesis, which you can find online, that was not published, where she did three-dimensional reorganization of tissues. So most of the simulations that I've just talked about are 2D simulations, but she did a full 3D model trying to do somatogenesis. And she graduated before that all got nailed down, but it is in her thesis, and it's well worth looking at.
Thanks.
Somebody says it's too bad that the last two sessions were short and a little bit accelerated. I guess that I'm sorry we don't have more time together, but you've given us a lot of your time over the past 5 days. You've given us 30 hours of your time. So we didn't want to take too much, but please do feel free to reach out and ask for more details. There is documentation of most of these things in the online manuals. Sometimes the manuals are not so clear. And if you find that the manuals are not clear, please do put a pull request into our Reddit and say, hey, page such and such of the manual says this and I don't understand it. Because the best way that we have to go back and fix things is to have people tell us that we need to improve. If you tell us where we need to do better, we try. Okay. Anything else we want to go over now? Otherwise, I will see you. I'll see you, many of you, I hope, tomorrow morning. Lorenzo, you probably need to go to bed. It's very late. Thank you so much for all your help.
All right, thank you. Night.
Thank you. Good night.
Uh,
And Julio, I think there are going to be lots of questions. Gilberto, I think
Yep.
people would like to see your 3D cell crawling too, probably. So if you,
No?
if you want to now, no. But maybe tomorrow afternoon when we're doing the optional informal presentations, you could do show the 3D
Okay.
cell crawling model. I can add that to the list.
Okay. That's it. See you tomorrow then.
Okay, thank you. Anything else before we break? Thank you to everybody for sticking it out. We still have 2 more days to go. But I do appreciate everybody's patience with, oh, and there's a new version of CompuCell that Maciek has just released, fixing the bug for the Windows tellurium antimony integration. So I'm not sure he's pushed it to a SourceForge yet, but it should be there very soon. Okay, well, thank you then, everybody. Appreciate you being with us, and I'll see you tomorrow.
Okay, bye.